eres la jefa de esto?
No.
Sí. Tú eres la jefa. Tú eres la jefa, tú.
Good morning to everybody. Thank you for coming to this wonderful workshop. First of all, I would like to introduce you to our Vice Dean of the Faculty of Pharmacy and Nutrition, Dr. Juan Manuel Irache.
Thank you, Pilar. So, good morning, dear colleagues and friends. It's a pleasure for me to welcome you to the Faculty of Pharmacy and Nutrition. On behalf of our Dean, Professor Maria Javier Ramirez, who unfortunately could not be here today as she is currently traveling, so she's coming back to Pamplona. Nevertheless, she has asked me to express her warmest welcome and to remind you that this is your faculty, you are in your faculty, this is your home. And thank you for coming. Welcome to everyone here in the room and also to those joining us online. It appears that we are more than 350 participants in total. This figure, at least for me, gives a sense of just how much interest there is in the topics of aging, immune metabolism, and inflammation. I would like to take a moment to congratulate the organizing committee for the excellent choice of the theme for this workshop and for the outstanding selection of speakers. Well done. Our faculty is the 5th oldest in Spain, and since its foundation, it has been committed to excellence in both teaching and research. The faculty offers 2 undergraduate degrees, the degree of pharmacy and the degree of human nutrition and dietetics, as well as 1 double degree and 4 master's programs focused on pharmaceutical and food sectors. We firmly believe in fostering innovation and quality, and in training professionals who are equipped to lead in a constantly evolving world. In terms of research, we are deeply committed to advancing frontier knowledge, a commitment reflected in the work of our Nutrition Research Center, with more than 70 researchers working there, mainly on the challenges associated with obesity. In line with the theme of this workshop, we believe that exploring the frontiers of aging, immune metabolism, and inflammation is of strategic importance, not only from a scientific standpoint, but also due to its implications for public health. Understanding the mechanisms behind chronic inflammation and immune and metabolic dysregulation may lead to the development of more effective therapeutic strategies for highly prevalent diseases, such as atherosclerosis, type 2 diabetes, cancer, and neurodegenerative disorders. All of these issues, to a greater or lesser extent, are reflected in the workshop program. Moreover, research in these areas could pave the way for the discovery of biological aging biomarkers and for early intervention strategies, even before the onset of clinical symptoms. Beyond the science, I would also like to encourage you to enjoy Pamplona if you haven't already. I invite you to stroll through its streets, discover its history, enjoy its people, and of course its cuisine. I hope you will take a moment to explore the city and allow yourself to be surprised by its charms and hospitality. Sometimes a short walk through the old town or a taste of the local food can offer a fresh perspective after an intense day of sessions and, I sincerely hope, fruitful discussions. And finally, let me thank you once again for attending this workshop. You are most welcome in this faculty, and I wish you a highly productive day, both in terms of knowledge and professional connections. Thank you.
Thank you, Juan Manuel. Good morning to everybody. My name is Pilar Lestao, member of the Scientific and Organizing Committee of the workshop Frontiers in Aging, Immune Metabolism and Inflammation, together with Dr. Maria Jesus Moreno and Dr. Elena Puerta, sit in this table as well. I would like to welcome, first of all, to our speakers. Thank you for sharing with us your science and for your availability to come to this conference face-to-face. This is very valuable these days. And coming to the main campus of the University of Navarre in Pamplona. Also, I would like to welcome to the attendants who are present in this auditorium. And of course, welcome to more than 250 attendants who are joining us through streaming. Welcome to all of you behind the screen. This is the 4th edition of a series of workshops under the general title Frontiers in, through which we would like to bring together experts in the cutting-edge topics in physiology. This workshop is organized by the Journal of Physiology and Biochemistry and the Center for Nutrition Research of the University of Navarre, with the collaboration of CIBER-OBN and IDISNA. The Journal of Physiology and Biochemistry is an official journal of the University of Navarre, published by Springer Nature, and precisely in this 4th edition of the workshop is special since we are celebrating the 80th anniversary of the foundation of the journal. For those who are not familiar with this journal, the Journal of Physiology and Biochemistry publishes original research articles and reviews describing relevant new observations on molecular, biochemical, and cellular mechanisms involved in human physiology. Fruit of this workshop, the intention will be to publish in the near future a special issue dedicated to the topic of aging, immune metabolism, and inflammation. I would like now to give the floor to Professor Maria Jesus Moreno Leaga, who will present the content of the workshop.
Thank you, Pilar. I also want to welcome all the speakers and the on-site and online attenders to the workshop. All of us are here together because of our common interest in understanding the complexity of aging. As you well know, the world's population is aging rapidly, and we face a future where the number of elderly people will exceed that of children, and there will be more older people than ever before. And aging is a complex process in which progressive, time-dependent physiological changes result in a deterioration of biological functions and a decrease in the quality of life. This loss of physiological integrity increases the risk to develop multiple pathologies, including obesity, type 2 diabetes, fatty liver, cardiovascular disorder, neurodegenerative diseases, and cancer. Moreover, there is a complex overlap between several metabolic disorders and aging. For example, it has been observed that obesity may accelerate the aging process. In this complex context, it is important to focus on the understanding of the mechanisms driving aging and age-related disorders. Aging is associated with metabolic changes in the immune system, which leads to an impaired immune response and makes older adults more susceptible to infections, autoimmune diseases, and cancer. As reflected by the title of the workshop, aging is also characterized by persistent chronic inflammation, called inflammaging, which has emerged as a critical factor linked to age-related diseases, including the cardiovascular diseases, the metabolic disorders, and the cognitive decline. Therefore, the understanding of the mechanisms underlying the resolution of inflammation during aging is critical to tackle these diseases. And in this context, we have organized this workshop with the purpose to discuss the current knowledge about the molecular, cellular, and systemic processes mechanistically accounting for aging and age-related diseases, Research in these topics is necessary to develop strategies to promote a healthy aging. We are very honored for having here some of the most recognized international experts in the study of aging and age-related diseases, metabolism, and inflammation. We would like to thank you, all of them, for accepting our invitation to participate in the workshop and sharing their novel findings with us. We also want to thank you, all the assistants, for their interest in this meeting, and we hope that you enjoy and learn with the program and discussions of the speakers. Thank you.
Well, good morning. Just a small practical issue. After each conference, there will be 10 minutes for questions. So those here in the public, just ask when, raise your hands, and we'll give you a microphone. And those online, you can use the form, the Google form that we sent you this morning. And we will receive the questions here, and after the talk, we will have 10 minutes questions with our speakers. Thank you very much. So after this introduction to the workshop, we are ready to start with the opening conference that will be given by Dr. Maria Blasco. It's my pleasure to introduce our first speaker today, Dr. Maria Blasco. Maria Blasco is a leading molecular biologist whose research has profoundly advanced our understanding of telomeres and telomerase in their role in cancer and aging. Since 2003, she has led the telomeres and telomerase group at the Spanish National Cancer Research Center, the CNIO, where she also served as scientific director from 2011 to 2025. Her work has been published in some of the most prestigious scientific journals, including Cell, Nature, and Science, and she has received numerous awards for her outstanding contributions to science. These include, among many others, the EMBO Gold Medal, the Joseph Steiner Award, the Rey Jaime I Prize for Basic Research, and the Spanish National Research Award in Biology, Santiago Ramón y Cajal. In addition to her scientific achievements, Dr. Blasco has been an active editor, advisor, and member of several national and international scientific committees. She has also received 4 doctorate honoris causa in Spain, and currently she serves on the advisory boards of institutions such as the FECID and the Fundación Valenciana Premios Rey I, and she's a full member of the Royal Spanish Academy of Pharmacy. Please join me in welcoming Dr. Maria Blasco.
Thank you. Good morning, and thank you for the invitation. It's a pleasure to be here. And I will be telling you about the work we have been doing on the role of telomeres in cancer and aging. We know that aging is at the origin of many of the diseases that kill us, and this includes many degenerative diseases as well as cancer. So it's essential to understand aging from the molecular point of view in order to prevent or to treat these diseases. So we think, as I said, aging is the cause of many diseases. The diseases would be the consequence of this molecular aging. This molecular aging can be influenced, of course, by our genes, also by our habits. But what is important is that if we know what is aging from the molecular point of view, we will be able to have, on one hand, biomarkers that will help us to find which persons are in risk of developing these diseases. But we also could, in theory, be able to modify longevity in model organisms. This would demonstrate that something is important for aging, for instance. But we could also maybe have new therapeutic strategies to be able to treat diseases of aging, which is something that still we haven't managed to do, right, to be able to cure degenerative diseases. So which are these molecular causes of aging? So a number of scientists, we put together a review in which we identify several of the, I think, very well validated molecular causes of aging. I work in one of them, which is telomere shortening. Telomeres protect our chromosomes, are essential for chromosomal stability. And when telomeres get short, they lead to other causes of aging. So that's why we think that this is a primary cause of aging. I will be telling you about telomeres. And more recently, we have extended these molecular causes of aging to new causes of aging in a more recent update of the review. And in the case of telomere shortening, I would like to tell you that there is an activity which is called telomerase. It was discovered in 1985 by Carol Grader and Liz Blackburn, which is able to compensate this telomere shortening that we know is at the origin of aging. And therefore, telomerase is an essential gene that I will be mentioning about around my talk, because it has the potential of re-elongating telomeres. And in fact, without telomerase, it's impossible to sustain life, because telomerase is necessary for the maintenance of the species. Telomerase is activated in early embryonic development, and it resets telomere length of the new individual. After birth, in our case, and in the case of many mammals and vertebrates, telomeres shorten. And this is associated to cell division, and this is due to the end replication problem, which is the ends of the chromosomes, the telomeres, are not replicated fully, and as the cells divide, telomeres become shorter and shorter, and this causes this telomere shortening. We know telomere shortening is rate limiting for longevity, because there are people with mutations in telomerase and other telomere genes that are going to be born with very short telomeres. And as a consequence of this, they are going to develop a number of diseases which are called telomere syndromes or telomeropathies that are characterized by a loss of the regenerative capacity of tissues. And one of the most prevalent telomere syndromes is pulmonary fibrosis. And I will be talking quite a bit about pulmonary fibrosis. But even if we are not mutant for telomerase, this rate of telomere shortening has been proposed to be a marker with prognostic value for different pathologies. In our lab, we actually developed a technology to measure telomeres. We funded a company that is called LifeLength that is still measuring telomeres for biomedical use. So how do we know that telomeres are essential for aging? The first evidence came from the generation of mice deficient for telomerase. This was the first time that we could demonstrate that if you remove telomerase from a mouse, telomeres shorten. So hence, telomerase is the enzyme that maintains the telomeres. And these mice are going to age prematurely. So short telomeres are sufficient to induce aging. But the other important demonstration was demonstrating that if you maintain telomeres longer in a mouse, you could extend longevity. And this was done in 2008 in our lab in Madrid. And we could show that by maintaining telomeres longer, you can actually delay aging. So if you have short telomeres, you age prematurely. If you maintain telomeres for longer, you delay aging. Therefore, telomere shortening is one of these hallmarks of aging. And this is not true only for mice and humans. It's true for many different species of animals. And here we did a collaboration with the Zoo of Madrid, and we measure the rate of telomere shortening in different species of birds and mammals, which are very distant in evolution. And we found that actually the rate of telomere shortening can be adjusted to a mathematical function that is called a power law. And just by measuring the rate of telomere shortening in a given species, you can predict the longevity of the species. And you can see, for instance, an elephant and a flamingo, they are very distant in evolution, but they have the same rate of telomere shortening and they have the same longevity. So therefore, an epigenetic factor, if you want, which is this rate of telomere shortening, is more important than genetics for determining longevity. And I think this is very relevant because this means that evolution can impinge in this rate of telomere shortening, and this can be a way of setting longevity or selecting for different longevities. So what you do, you have short telomeres. So we thought, and this was done together with the group of Fatima Bosch in the University of Barcelona, we thought that a good strategy to reactivate telomerase in the adult organism in order maybe to be able to cure degenerative diseases was to use gene therapy. We use gene therapy using adeno-associated viruses, which is a state-of-the-art for gene therapy. Now, you could also use RNA, for instance, to do the same, and it's been done, actually. But we use the AAV vectors to deliver telomerase into a mouse. So we took, in a first experiment, we just took middle-aged and old mice and gave them a single injection intravenously with a vector that carried telomerase. And then we wait and see. And we saw that the mice that had telomerase They had longer telomeres, but they have improved in all parameters that we measure related to aging, including cancer. Cancer was also delayed because cancer is also a disease associated with aging, with the exception of the pediatric tumors. And we didn't see any negative effects of this telomerase delivery into an adult mouse because, in fact, the mice live longer. The mice that were 1 year old, they live 20% longer, and the mice that were already very old, like 2 years old, they live like 13% longer. So there is not a negative effect of this activation of telomeres using gene therapy. And you may wonder then why we don't have longer telomeres if this is so good. And the way we had to answer this was with an epigenetic trick. We were studying also reprogramming because associated to cellular reprogramming, we discovered that this is an extension of telomeres. When you reprogram cells, for instance, with the Yamanaka factors, telomeres are greatly elongated, and this is mimicking what happens in the blastocyst. I told you that in the blastocyst is where telomerase is activated and telomeres are elongated. And with these telomeres, have to leave the new individual. When you reprogram cells, the same happens, so telomeres get elongated. And we notice, however, in iPS cells first, but then in ES cells, that the telomeres keep growing if you maintain these cells in culture. If you maintain these cells locked in pluripotency, the telomeres keep growing. So you can have ES cells with what we call ultra-long telomeres, much longer than the ones in the blastocyst. Then we thought, oh, this is very interesting. Now we can make a mouse derived from these ES cells with ultra-long telomeres, and we can maybe generate a mouse that is born with double the length of the telomeres of a normal mouse. What will happen? They will live more, less, more cancer, less cancer. And this is what we did. We took A cell morulas. We aggregated with ES cells with normal or ultra-long telomeres. You can follow the ultra-long telomeres by GFP. You can see here we were able to get mice, chimeric mice. You can see the eye of one of these mice. Just by eye, you can see the areas that are derived from the ultra-long telomeres in red. So they have much longer telomeres. These are the ones derived with the ESLs. And these are the normal length of the telomeres in the eye. So what happens with these mice that have much longer telomeres? When we did like 30 to 70% chimeras, they live longer. They have less spontaneous cancer. And even if we induce cancer, they have less cancer. So having long telomeres is something good. It's not bad in a mouse. We later on got 100% chimera. So 100% of the cells of the mice are derived from these cells with ultra-long telomeres. You can see this by the GFP. And then we also saw another thing. They were protected from metabolic syndrome. They had less obesity. They have improved glucose tolerance. And again, we saw less cancer, etc. So if we would have longer telomeres, we would live longer probably, and we would be more protected from aging. So what are we doing from the therapeutic point? So what we are doing is, and these are the different people in the lab that have been doing different projects related to different diseases of aging that I will be telling you about. But basically, the idea is to use this telomerase gene therapy to prove whether it has a therapeutic effect in treatment of different diseases of aging. We started with heart infarct, and we published this many years ago. And telomerase was protecting from heart infarct in mouse models. Everything is in mouse models. Then we tried the telomerase gene therapy in a mouse model of aplastic anemia associated to short telomeres that we developed in the lab. And more recently, we have been trying this telomerase gene therapy in a mouse model of pulmonary fibrosis associated to short telomeres that we also generated in our lab. Why pulmonary fibrosis? So we know now that short telomeres are at the origin of pulmonary fibrosis, at least of a high percentage of cases, because there are familial cases. And when the genes associated to this familial susceptibility to pulmonary fibrosis were found, they happened to be telomerase. This was the first one to be found. And then many different telomere binding proteins. So it's clear that when you have short or dysfunctional telomeres, this may synergize with damaging agents to the lung, probably killing some of the regenerative cells of the lung. I will show you that these cells are the alveolar type 2 cells. And this impairs, the short telomeres impair the regenerative capacity of cells. We have shown this in my lab and other labs. So if you cannot regenerate the damage, then you have inflammation, scarring of the lung, and then you have the fibrosis. The treatments which are being used in the clinic are not able to cure patients. They are anti-fibrotic, but the problem is that you still have the origin. You still have the short telomeres, you still have the inability of the regenerative cells to regenerate the lung, and that's why we think these drugs don't cure fibrosis. So what can we do? So what we did is to target the short telomeres. We generated actually, I forgot to tell you that we did something I mentioned before, but we wanted to know which cell type is the one that we would have to target, right, with telomerase. And for that, we did a collaboration with AstraZeneca in which we induced telomere dysfunction in different cell types in the lung. So this was done with mouse model, conditional mouse model, and you can see that the only cell type where when we induce telomere dysfunction by removing one of the telomere protective proteins, which is called TRF1, the only cell type is alveolar type 2 cells. It's the only instance in which we get interstitial fibrosis. So this means that we will have to target telomerase to the 82 cells, and also that these 82 cells are at the origin of pulmonary fibrosis induced by short telomeres. So we first did an experiment in which basically we developed a mouse model that had short telomeres and that we challenged with a low dose of bleomycin that does not induce fibrosis in a normal mouse, but if the mouse has short telomeres, it induces fibrosis. So it's a sort of a humanized mouse model. And you can see here, we diagnosed fibrosis by CT in the mice, and then those mice that had fibrosis, we treated either with the empty vector or the telomerase, and you can see that the ones treated with the telomerase, half of them completely removed the fibrosis, and the other half had only small patches of fibrosis compared to the controls that developed this progressive fibrosis. So, and we have seen that this is very fast. Only 3 weeks after the treatment, we decreased many pro-inflammatory cytokines. We also decreased many fibrotic pathways. We can also isolate the alveolar type 2 cells and see that these alveolar type 2 cells have more telomerase, and they have decreased DNA damage pathways and decreased immune signaling pathways. So by targeting the telomeres, the short telomeres with this telomerase gene therapy, I haven't shown you everything, but basically we rescue in this mouse model everything related to pulmonary fibrosis. So this tells that this could be an interesting treatment for humans with pulmonary fibrosis associated to short telomeres, which are estimated to be about 50% of the pulmonary fibrosis cases are thought to be associated to short telomeres, not all of them. So that's why this is important. So we wanted to do a sort of a killer experiment because I haven't mentioned, but telomerase is activated in cancers in order to maintain the cancer cell growth. I told you that telomerase gene therapy does not induce cancer. It actually delays cancer in mice because it's maintaining the mouse in a younger state with less DNA damage. But we wanted to challenge this hypothesis. And what we did here is that either at the same time that we put telomerase or before we put telomerase, we induce KRAS in the lung. KRAS is going to induce to lung cancer. So the idea is telomerase going to favor lung tumors, either if we put before the induction of the KRAS or at the time of the induction of the KRAS. So we wanted to see the safety of this in mice, of course. Humans, of course, are a different thing, but in mice at least, we challenged the mice with KRAS in the lungs, and we saw that the delivery of telomerase was not, either in the pre-treatment or the simultaneous treatment, did not increase the number of mice with tumors, the tumors per mouse, or the tumor maximal section. So telomerase was not changing the natural history of KRAS-induced tumors. So every evidence we have is that this gene therapy is not inducing more tumors, even if we challenge with the KRAS oncogene. So the idea is now, and we have generated now vectors to deliver telomerase into humans with pulmonary fibrosis. This is being done by a company that has been funded by the CNIO, by the Autónoma de Barcelona, and is participated by Invierte and a capital risk fund. And the idea is that to bring this to patients with pulmonary fibrosis. So I wanted to tell you something unpublished still, is that I told you not all cases of pulmonary fibrosis may benefit from a telomerase gene therapy, and this is an example of that. There is a recent mutation that has been found in one of the telomere binding proteins. It's called POD1, and this mutation has been associated also to cases of pulmonary fibrosis. So we decided to This is the POD1 protein, sorry, it's protection of telomeres 1, POD1. There are mutations in POD1 that lead to cancer, but this particular mutation leads to aging, to pulmonary fibrosis. So we decided to study this, so we generated a mouse model carrying this mutation in POD1, and we're very happy to see that these mice actually have shorter telomeres, like in the human patients carrying the mutation. So the mice have short telomeres, as you can see here, measured with different technologies. The telomeres are more unstable. This is a chromosomal aberration associated to dysfunctional telomeres, and they have more DNA damage. So we wonder, is this mouse model similar to the telomerase knockout? And in order to demonstrate that, we decided to generate increasing generations of these mice. And we generate first generation, second generation, third generation, and you can see that the telomeres keep shortening. This is exactly the same phenotype as the telomerase knockout. The telomeres decrease with increasing mouse generations in different tissues. And not only it mimics the telomere shortening phenotype, also the telomerase deficient mice are smaller with increasing generations, and this also becomes smaller with increasing generations. The litter size decreases with increasing generation, and you can see that it's phenocopying completely the telomerase knockout. This is the third knockout versus the POD1 knock-in, and it's phenocopying the genotype. So we were very excited to see that this mutation is like having a telomerase mutation. And in fact, the double mutant knock-in for POD1 and telomerase doesn't have an additional telomere shortening. So it's indicating that they are in the same pathway, let's say. It's not additional. You can see here that the telomerase knockout, second generation, is equivalent to the in telomere length to the POD1 knock-in second generation for third. So they have the same telomeres, short telomeres. So, and then nevertheless, we thought, well, okay, let's see if we can rescue these short telomeres in these mice by telomerase gene therapy. In this case, we use adeno TER vectors, and the answer is we cannot rescue. In the wild-type POD1, we see an increase in the telomere length, but in the POD1 mutant, telomerase is not able to rescue. So this would be a case where you cannot rescue with telomerase the short telomeres, because there are mutations in telomere binding proteins that are mimicking telomerase. Why are they mimicking telomerase? This is a very complex slide, but bottom line, I'll show you here that what we found is that this POD1 is able to bind to the telomeres, is able to bind to telomerase, to bind to its partner, TPP1, but it's not allowing telomerase to work on the telomeres, and it's acting like a dominant negative, as a matter of fact, and you can see that because it's a dominant negative because the heterozygous mutants also have a phenotype. So not only the double, but the single knock-ins have a phenotype in telomere length, and this is telling us that this is acting as a dominant negative. So there are mutants that have this dominant negative POD1 that is inhibiting telomerase, and they behave like a telomerase deficient. Okay, so the other disease that we have been interested in is kidney fibrosis. Kidney fibrosis is also a very prevalent disease. It's increasing with the demographic aging. And these patients that have mutations in telomerase also have more kidney fibrosis. So we wanted to demonstrate whether short telomeres were at the origin of kidney fibrosis, and we follow the same strategy as with the pulmonary fibrosis. The idea is that we were going to take the telomerase deficient mice with short telomeres that per se don't develop kidney fibrosis. They die before that of aplastic anemia and other things. And we challenge them with a dose of a damaging agent to the kidney that is called folic acid, in this case, in our case, that this folic acid dose is not inducing kidney disease in a wild-type mouse with long telomeres, but if the mice have short telomeres, they develop kidney fibrosis. So this is like a humanized model also. You can see this here by looking at the creatinine levels, so the blood urea nitrogen levels. You can see that when we put this low dose of folic acid into the wild-type mice, nothing. But if we put this low dose of folic acid into the telomerase deficient mice, you get kidney dysfunction. So it's being originated by the short telomeres. And you can see here a number of different markers of inflammation and fibrosis. And we also found something interesting that was not found before. It was sort of unexpected, but we knew that epithelial to mesenchymal transition problems were associated also to kidney fibrosis, and we wondered, are the short telomeres also initiating this epithelial to mesenchymal transition? And this is interesting because we know short telomeres can initiate cancer. And cancer, also there is an epithelial to mesenchymal transition. And this is exactly what we found. We found that the mice challenged with folic acid that were long telomeres, they did not express increased levels of any markers of EMT, but when the mice had short telomeres and we challenged them with folic acid, then there you have all the markers of EMT being upregulated. So we thought this was interesting because it was placing these short telomeres also maybe at the origin of this epithelial to mesenchymal transition, and then we came up with this model that short telomeres are initiating or are at the origin of kidney fibrosis and are involved in this epithelial to mesenchymal transition, which is also have been known for a while to be important for kidney fibrosis. So what we are doing now with the kidney is the same as I told you. This is still very recent work, is to see which is the cell of origin in the kidney, which cell we will have to target. And for that, we use the same approach. We are going to delete induced telomere dysfunction in different cell types. We started with the fibroblast, and we found that fibroblasts and pericytes, when we delete, when we induce fibrosis, is not leading to a dramatic fibrosis. Only in very old mice in the fibroblasts we see some signs of fibrosis, but basically this cell type, as in the lung, doesn't have a role in the origin of fibrosis. It may be a response to the defects, but it's not at the origin. However, when we do renal tubular epithelial cells or basal cells, there are very clear and strong phenotypes. These cells are important for originating kidney dysfunction by short telomeres. And in the renal tubular epithelial cells and as well as in the basal cells. So you can see here, for instance, the induction of telomere dysfunction in renal epithelial cells. Here we are not challenging with anything. The kidney is just what happens, and you have these markers of fibrosis very early on, and which are aggravated with time. This is interesting. These are different, in this case we are also looking at markers of senescence, etc. You can see that inducing telomere dysfunction in renal tubular epithelial cells is inducing aging of these cells, and this induces fibrosis. Maybe more interesting and unexpected is that when we did induce telomere dysfunction in basal cells, we found something called hydronephrosis, which is a disease that may happen in infants. This is very severe. The mice die very early with basically no kidney. And we are still trying to understand this unexpected phenotype. If the mice survive the first days, they will end up with fibrosis as well. You can see here smooth muscle actin, inflammation, etc. But they develop this hydronephrosis, which I think is interesting, could be a model for hydronephrosis. So, and to finish with the fibrosis part, I would like to mention COVID, because when this disease appeared, I was very interested to see that the patients developed pulmonary fibrosis, kidney fibrosis, and the virus was able to specifically enter, for instance, alveolar type 2 cells in the lung in humans. So we wonder, is this COVID-19, does it have a component of aging, a component of short telomeres? Because also patients were dying more if they were older than basically young people, nothing happened almost. And we were able to manage to get blood from the field hospital in Madrid, and we could see, I mean, it's a very basic study because we couldn't do many things at that time, but we saw that there was a correlation between shorter telomeres and more severe COVID phenotypes in these patients, COVID pathologies. This is blood, telomeres in the blood, and then the pathologies, but we later on, we collaborated with Hospital Valdecilla in Santander, where we could get samples from the lungs. These were areas of lung cancer patients, areas of the lung that were not cancer, they were normal lung, of patients that either had COVID or not during the early days of the pandemic. And we saw, this was also described by other groups, that the COVID patients had more markers of fibrosis compared to the non-COVID patients. But what this is important is that we saw that there was an inverse correlation between fibrosis and telomeres in the lungs, in this lung. So the more fibrosis you had in the lung, the shorter the telomeres were in this lung. So again, supporting already in the lung that short telomeres could be part of the causes of the COVID-19 upon infection with the virus. And maybe these patients could also benefit from this telomere gene therapy that we have developed for pulmonary fibrosis. Okay, and then I will, in the last, I think, 15 minutes or so, will tell you also what we do in my lab regarding cancer. I already mentioned briefly, but I'm sure you know that one of the hallmarks of cancer is the ability of cancer cells to divide indefinitely. And this is provided to cancer cells by telomerase. Telomerase is the third most mutated gene in cancer. There are mutations in the promoter, there is amplification. Telomerase has to be up in the tumors in order to maintain telomeres and in order to allow cancer cells to divide indefinitely. And, but telomerase is not the only thing that is mutated regarding telomeres. At the CNIO, we were the first ones to describe the first mutations in telomere binding proteins. Again, remember POD1, I showed you a mutation that lead to aging, but there are mutations that lead to cancer, the same protein. And we found that it was, together with Elias Campo and Carlo Pezzotin, we described the first mutations in POD1 in CLL, but then it has been described also in familial glioblastoma, melanoma. We also described these mutations for Li-Fraumeni-like syndrome, which is non-p53 mutant, leading to many different tumors. So how is this happening? How is this POD1 mutation leading to more cancer? We found that what it generates is chromosomal instability because you have the sheltering, the protective complex is disrupted, but also is favoring long telomeres. The contrary to the other POD mutation. I show you short telomeres, this one long telomeres in the same protein. And these are the different mutations. This is the Li-Fraumeni-like syndrome that we found at the CNIO, and that we generated a mouse model. And again, the mouse model, well, these are the different papers I told you about. But, sorry, this is duplicated. I wanted to, and this was, our work was validated recently in a paper also with clonal hematopoiesis, where they found this also long telomeres associated to POD1 mutations. So we generated a mouse model for this mutation, and again, the mice recapitulate the humans. And you can see here both mouse embryonic fibroblasts and iPS cells derived from these mouse embryonic fibroblasts have longer telomeres. And we saw not only in MEFs and iPS, we also saw in the tissues longer telomeres and more tumors in these mice. So somehow this mutation recapitulates the mechanism of action is that this mutation is, the other one is acting as a dominant negative, this one is acting as a processivity factor for telomerase. So I'm not going to go into detail into everything, but basically the mutation increases telomerase processivity, is able to bind telomerase, is able to bind to the telomeres, and increases telomerase processivity. And this is why this mutation is leading to longer telomeres. So anyway, you have long telomeres in cancer, and you have telomere maintenance mechanisms in cancer. What can you use this to target cancer cells, to make them die, to make them age and die? And you know probably that there have been development of telomerase inhibitors, but they have largely failed because telomerase, when you inhibit telomerase, there is what is called a phenotypic lag. So telomeres have to shorten, and this takes time. And this may be too long in order to kill a cancer cell. And also, if you inhibit telomerase, you may be killing the cells with the shortest telomeres in a tumor, but you may be selecting with cells with longer telomeres, and my group has shown that the cancer stem cells and the adult stem cells, they have longer telomeres. So we thought, okay, let's forget about telomerase, and let's try to target directly the telomere binding proteins, because we knew, because we have generated mouse models for all these proteins, for instance, for TRF1, that if you remove TRF1, you induce a persistent DNA damage response, so you basically uncap the telomeres, And this is sufficient to stop the cells. This is sufficient to arrest the cells or to kill the cells. So we thought, okay, maybe if we target TRF1, we can have a way to target telomeres in cancer, which is something that hasn't been done. I mean, it's not regularly used in the clinic yet. And we also like TRF1 versus the other shelterings, because TRF1 we have shown in the lab that is a pluripotency factor, is very important for the adult stem cells, and we reason TRF1 is also important for cancer stem cells, so it may be a good target to target TRF1 because it would not only stop the growth of cancer cells, but also the cancer-initiating cells, right? And we did that first in lung cancer, and this was published in collaboration with a group of Mariano Barbacid that has this KRAS model, and we showed that telomerase, TRF1 deletion in mice blocked the growth of KRAS-induced cancer, and more recently, we show it for glioblastoma. Glioblastoma is a very aggressive brain tumor, and we thought it could be a good model to test this hypothesis that targeting TRF1, which is a stem cell factor, and a telomere binding protein could be having a therapeutic effect, and it does. And there is a therapeutic window, because if we delete TRF1 in the adult mouse, and we did this experiment, it's also like a safety experiment if you want, so we deleted TRF1 throughout the lifespan of either tumor-prone or wild-type mice. We put tumor-prone to see whether inhibition of TRF1 may be generating chromosomal alterations that later on, down the line, will generate secondary tumors, for instance. But in this case, you can see that the longevity is normal. There are some phenotypes, like hair graying and hair loss, but no more than, or I would say clearly less than a regular chemotherapy for cancer. So there is a therapeutic window. So what can we do with this? So we have at the CNIO a drug development program, a drug discovery program, and we have been looking for molecules that are able to decrease TRF1 at the telomere. So this is a cell line that we generated that is knocking EGFP TRF1, and we look for things that decrease TRF1 fluorescence with the hope of finding TRF1 inhibitors. And one of the first series of inhibitors we found were AKT inhibitors. And from the chemistry, we discovered biology, because it was not known that AKT phosphorylated TRF1, and this phosphorylation is essential for stability. And this was discovered thanks to doing chemistry. And I think this is very, very exciting. And you can see here that TRF1 is phosphorylated by AKT in 3 residues. And if you mutate these residues to alanine, you decrease the phosphorylation. So TRF1 is a target of AKT. And these mutants of TRF1 that cannot be phosphorylated by AKT, they have lower stability, they bind less to telomeric oligos, and in vivo they can reconstitute less the telomeres of a TRF1 deficient mouse model. So this is relevant. This is the mouse TRF1. What about human TRF1? Is this conserved? So we saw that human TRF1 is also phosphorylated by AKT, and we have generated mutants in the human TRF1 residues that are AKT phosphorylated, and again, we see lower TRF1 levels, we see more DNA damage. In the case of humans, we also see shorter telomeres. If TRF1 is not, this is a stable cell line done with CRISPR-Cas, this knocking cell line for these mutants. And because we're using a cancer cell line, we wonder, well, are these cancer cell lines that have mutant for phosphorylation of TRF1 by AKT going to induce tumors the same? And the answer is no. Because TRF1 cannot be phosphorylated by AKT, these cell lines are less tumorogenic. So which was pointing to an interest in our hypothesis, right? That is, maybe we can target telomeres to treat cancer, no? By targeting TRF1. So in these papers, we also did more experiments, but bottom line, I think what we are discovering here is very interesting, because we are discovering that telomeres can be regulated by extracellular signals that can be in the food, that can be in the serum or the cells. So somehow telomeres respond to the environment, and this is what is telling us this story. And this brings me to to the fact that what we thought, if AKT is phosphorylated in TRF1, how many more things are touching TRF1? And then we did a screening with 140 FDA-approved drugs and see how many of them were actually decreasing TRF1. And we found our PI3K, AKT, mTOR pathway, validating our previous finding, but we also found the RAS MEK-ER pathway, and we are studying this now in the lab. There is a person studying this, and we are preparing our paper on that. But yeah, telomeres seem to be regulated by very important cancer pathways. And in fact, if you look at the TRF1 protein, here are all the phosphorylation sites that you can find in TRF1 for all these kinases important for cancer. So this is probably interesting to develop inhibitors for cancer. And we found a particular residue that is phosphorylated by all these kinases. And we decided, well, let's make mouse models for this. We are going to make phosphodate mouse models and phosphomimetic mouse models. This is still unpublished. This is the phosphodate. The phosphodate, and this is already MEVs or mice. I don't know why it's not appearing here. But basically, with the phosphodate, we see less TRF1 levels, even though here is not a tendency to have less levels. But we didn't see changes so far in telomere length or in body weight. These mice seem to be fine. But we wonder whether they will have more or less cancer. If we induce tumors, we will do all these experiments. We are still on the process. The phosphomimetic are being more interesting and intriguing. So in this case, we have TRF1 that is permanently phosphorylated, permanently phosphorylated at this site. There is more TRF1. Clearly, TRF1 is more stable when we do a stability assay. So it's like having a super TRF1 mouse. What happens with these mice? Well, they have longer telomeres. This is a mouse embryonic fibroblast. So the contrary to these mice are born with longer telomeres. And this telomere elongation is telomerase dependent, because if now we cross the knock-in with the telomerase deficient, we rescue the long telomere. So this mutation in TRF1 is making telomerase more active at the telomere and is making these mice to have longer telomeres. There were MEVs. These are tissues in young mice. Okay, you see in the lung, the intestine, the liver, longer telomeres. So maybe these mice are going to live longer, but maybe they will have more cancer. We don't know what happens with these mice. And here comes the surprise. The mice are developing like an obesity phenotype after a year of age. They are bigger. We know that they have more fat. And this is seen both in males and females, so it's something unexpected. Why? Maybe it's because this thing I told you about the environment. I mean, it's this ability of the telomeres to signal extracellular signals to the nucleus, to the telomeres. But even more surprising was that these mice die earlier. So they had how is possible? They have longer telomeres. They die earlier and they die of cancer, maybe because they have this hyperphosphorylated TRF1. But what we didn't expect at all is when these mice die, they have extremely short telomeres. So in utero, they have much longer telomeres. They are born with longer telomeres, but as they age, they completely lose the telomeres. Why is that? We are trying to figure out. I can tell you that TRF is being expressed higher, so there is more TRF1, but at the telomeres we have less TRF1 because the telomeres are shorter. So there is more TRF1, but this is causing this, in utero is causing long telomeres, but outside of the utero is causing this dramatic telomere shortening. What could this be? We don't know whether this is increased stem cell mobilization because TRF1, I told you, is important for adult stem cells, and maybe it's mobilizing more stem cells, and this is causing telomere shortening, recruiting more exonucleases to the telomere, the consequence of metabolic effects, we really don't know. So I will finish here. I think you have an overview of the things we do in the lab to try to understand the roles of telomeres and telomerase in cancer and aging. And most of the work I told you, I want to mention some people. Paula Martinez has been working in the telomere binding proteins and cancer and aging together with Raul. And I didn't have, majority of the work I told you has been done by them. And Sarita, that is the one that has been doing all the kidney work that I've shown you. And the other people are working on other things that I didn't have time to tell you about today. And I stop here. Thank you.
Thank you very much, Maria. It was a great talk. Thank you. Is there any question in the audience? Okay, we have, just raise your hands.
Maria Antonia, congrats, amazing work. I have a couple of questions. The first one regarding, longer telomeres, longevity, and cancer. Now, we learn at the school that, for instance, in liver cancer, the first thing you do is to mutate telomerase, and then this is the first step. How can, put together this controversy between when you overexpress telomerase, you see less tumors even if you activate KRAS, and at the same time, you
It's a very good question, but I think we have been answering it in the field with time, right? So short telomeres lead to cancer, because it are generating chromosomal instability, and there is a particular type of instability that is originated from the short telomeres. So in that regard, if you dare put more telomerase and prevent these short telomeres, like what we are doing with the gene therapy, for instance, then that's why you are decreasing tumors. Or if you make mice epigenetically without overexpressing telomerase, with long telomeres, they have less cancer. When you have a tumor, already a tumor, then you need at some point to activate telomere maintenance mechanisms. Telomerase is the favorite one of cancers. 90% of the tumors have telomerase, mutations in the promoter, amplification, you name it. But some tumors have mutations in POD1, and others have recombination. It's called the alternative mechanisms of lessening. This is happening in 10% of the tumors, mainly sarcomas and these types of tumors. So, yeah, you need to maintain the tumors of a cancer in order for the cancer cells to grow, and that's why it's interesting to target telomeres in cancer. But telomerase is not an oncogene, we have shown that. And maybe the strategy that we are using, which is gene therapy with non-integrative vectors, is allowing for this balance, right? Because we would put telomerase elongates telomeres for a couple of cell divisions because the virus is diluted, and then you will lose telomerase. And this clearly, at least in mouse models, and even in mouse models where we overexpress KRAS, is not leading to more cancer. So I think it's quite, all the evidence we have, even in the long-term longevity studies, we see less cancer with the gene therapy.
Yeah, I agree, of course, with what you say, no? But for instance, in the case of liver cancer, where the first mutation is telomerase, and then other mutations like P53 are coming along, could this be that, there is a matching between a particular oncogenes, not KRAS, but others, that in these cases, then you potentiate cancer?
I think in the liver, maybe telomerase is activated, is there, but you need the mutations in the other genes. Yes. But we published a paper with Greg Hannon many, many years ago in Cancer Cell, in which we demonstrated that it was possible to transform cells in vitro. This was post the Weinberg experiment of transforming cells with telomerase plus blah, blah, blah. In this case, we transform cells, fully transform cells without telomerase. If the cells have long telomere, you can transform a cell without telomerase. You don't need telomerase to transform a cell. You need telomerase later on to maintain telomeres, or telomerase or whatever that maintains telomeres. It could be recombination.
And my second question is regarding gene therapy. No?
Yeah.
You show very nicely that you need a cell of origin to target, but then you need to put the viral vector into that cell of origin. No? How easy is that? And what happens when you have these chimeras where, you are...
Well, it's easy if you use DNA vectors. As you know, you can play with the promoters. And we have achieved in the human vectors, we are targeting it specifically to 82 cells. So with RNA sort of Moderna business, it's going to be more difficult to target to a particular cell type. Actually, we have been collaborating with Moderna and with our models for that, for telomeres.
So, Maria, I have two questions. One is curiosity, is related to, do you think there is, the concept of aging is heterogeneous, that there are organs that are more vulnerable than others, and there is a
Certainly,
sequence?
certainly. And in all this, you know, a caveat of all this work I told you is that we do always sort of knockouts that are quite, well, no, because in the kidney and the lung, we are deleting in a particular cell type, and then the phenotypes are clear, it's produced by that. But in general, when you work with you know, whole body knockouts or knock-ins, you are missing that information. It would be, yeah, for the world of telomeres, let's say, we have done not so many experiments regarding, some experiments have been done regarding which is the important tissue, right, let's say, for aging induced by short telomeres. I think with zebrafish, some studies have been done in that regard.
And my second question is related to the link with the TRF1 and AKT and PA3 kinase and insulin.
Yep.
And the concept is in the context of insulin resistance and diabetes.
I think
do you see any changes in this?
We will have to do that. I think it's very interesting. I'm very excited about this idea that, you know, you can regulate what happens at telomeres from, in theory, from food, from the environment. And yeah, someone told me even this experiment that was done with the astronauts, the two twins, one stayed in Earth, the other one went to space, elongated the telomeres, but when it came back to Earth, they shortened again. So who knows? Maybe even microgravity is affecting telomeres. We don't know.
Okay, we may take the diabetics to the space.
Good talk. This is Jose Pichel from the CIVIR in Logroño. I have a couple of questions. The first one, something was already answered about this, but do you know how is the influence of telomere shortening in different cell types, and specifically in mesenchymal cells and fibroblasts, because of the key role that they have in the fibrosis? And the second question is, what's known about telomere shortening and normal human aging?
Well, the first one, we have induced telomere dysfunction specifically in fibroblasts in the lung, and they don't develop fibrosis. So the insult of having a dysfunctional telomere in a fibroblast is not inducing fibrosis. Only when we do that in alveolar type 2 cells, we get fibrosis, interstitial fibrosis. And in the kidney, the same. When we induce telomere dysfunction in pericytes or fibroblasts, no fibrosis. Only when we do it in regenerative cell types, like the basal cells or the renal tubular cells. So this is telling us something about the origin of these diseases. You have to look more to the epithelial compartments than to the mesenchymal, even though they may have a role later on, of course.
Yeah, about the second question, about telomere shortening and normal human aging?
I know, yeah, this has been studied. You know, there are many papers showing correlations of telomere shortening in humans with different diseases, life habits, you name it. There is a huge literature on that. To me, the clear demonstration that short telomeres matter for human aging is the telomere syndromes. You have a mutation in telomeres, you are going to live less because you are going to lose prematurely the ability to regenerate certain tissues. So, yeah, it's very limiting in humans, telomere length.
Thank you for your presentation. Are these findings meant to escalate to patients in the clinic, for example, those who want to prevent degenerative diseases?
Yeah.
And what are some ethical issues that could become a problem?
Well, that's our aim, to be able to bring this telomerase gene therapy for the treatment of fibrosis. This is our goal, really. And I think preventing, which would mean treating people before they get ill, in order for them not to develop diseases, right now is quite difficult from the regulatory point of view. You have to have a disease in order to be able to approve treatment. Treating people that are still healthy to prevent diseases is not so easy. And probably in the future we will think about that. No? Especially with the telomere syndrome people, for instance, that they know they may have a higher risk of fibrosis. You would want to treat their telomeres before they develop fibrosis. But,
Hi, Jasmine Darlik, Mayor University of London. Fantastic talk, thank you very much. I had more of a philosophical question, if I may. In aging, we know that there's a correlation, or at least there's an emerging understanding that inflammation plays a role in aging. Yeah. And we know there's a role of inflammation in telomere shortening.
Yep.
Now, my philosophical question is, which is the chicken, which is the egg?
Yeah.
Where
very
is
good,
the...
Very good question. Well, in this model of lung fibrosis, when we treat with telomerase and correct the short telomeres, we rescue inflammation, all pro-inflammatory cytokines. So certainly, if you prevent this telomere shortening, you don't get inflammation associated to fibrosis, pulmonary fibrosis. Then I understand, I'm not an expert in this, but I understand that there were clinical trials for pulmonary fibrosis based on anti-inflammatory drugs, and they had to be interrupted because they failed. The patients were not getting any better. So I would say, I don't know, probably the egg is for me, it's the telomere, I don't know. But yeah, you know, it's interconnected. Probably short telomeres are going to cause fibrosis, inflammation, I say, and for instance, obesity induces inflammation, and this induces telomere shortening in patients that are, it's very interconnected.
Thank you very much.
Difficult.
I had a little bit the same with the COVID study. Maybe those having shorter telomeres from the beginning were at higher risk of having like more severe COVID.
Yeah, that's
COVID inducing the shortening of the
that was our
telomeres?
hypothesis, because as we saw that the virus was entering the 82 cells, and we have found the 82 cells, the patients were developing fibrosis, and the virus was killing with increasing age. It was, well, it has to do with telomeres, I'm sure.
Okay. We have another question also from the online.
Thank you, Maria, for your really nice talk. I want to come back to this prevention that was told before. You said that all these inhibitors of telomerase in the context of cancer failed, maybe because they are slow. But have they tested maybe in the prevention of these recurrences that we know that we will have in many cancers?
Well, I've been too tough, because I think they are being used in multiple myeloma, the telomerase inhibitors that were developed by Geron, and I think they are used for some tumors. But the evidence that they are targeting telomeres to me is something. I don't think people think that this is the way that telomeres are going to be targeted in cancer. I hope the way is by going and destroying the telomere protection. No? And we are looking for inhibitors. I didn't mention, but of course we found this AKT, with these pathways. These are not the inhibitors we want, right? You want to have direct TRF1 only inhibitors. Maybe you can combine with other things. And this we're doing.
Okay, thank you.
We have a question, someone online asks, besides telomere length control, what is the contribution of other non-canonical functions of the telomerase for controlling the aging? Whether if in your experience, will you quantify this contribution, if it's relevant?
Yeah. Well, actually, this was a nightmare in the telomere field, I have to say, when Steve Artandi published this paper saying that TERT had non-canonical functions regulating the beta-catenin, I mean, all these pathways. We all had to do this. I had to do the experiments also with dominant negative. I had to make a mouse with telomerase and then the dominant negative telomerase. Until my ex-boss in the US, Carol Greider, demonstrated very clearly, I think she published that in Cell, that such role of TERT, independent of telomere elongation, was not something physiological or something that had an importance. I'm not saying that there is not any non-canonical role of any of the components of telomerase. For instance, for the telomerase RNA, it seems it may have other roles. But certainly the TERT, I think this was solved, but it was at some point, it was a lot of work. We had to double the work.
And last question here.
Well, thank you, Maria, for a very nice presentation. Pedro González from the University of Navarra. I would like to ask, you have shown a very nice slide about the differences within a species. I imagine there is also some research on within the species, which is the range of variability. And what are the factors that may affect this variability, and how many generations we need to get changes? For example, I mean blue zones, the people that live in blue zones, I imagine that they would have longer telomeres, but how many years do they need to live there?
Yeah, very interesting question. What is determining this intraspecies variation in the rate of telomere shortening? We published a second paper where we saw it was a correlation, but we saw also that DNA repair. You know, telomeres are DNA. If you have a stronger DNA repair mechanisms, maybe you are going to maintain your telomeres better. So I'm sure that DNA repair efficiency is one of the players. And then how many generations you need? It's a great question. I can tell you there was a paper by Anne Brunet, she's working with C. elegans, making mutants of C. elegans. She found a mutant that had longer telomeres, without more telomeres, just longer telomeres, and she saw that this trait was lost, in this case was lost, I think it was like 3 generations down the line because she didn't have telomeres, so probably, you know, this is something that ended up being lost. I don't know how it takes for stabilizing a longer telomere phenotype. Maybe it's around 3 generations, I don't know.
Thank you.
Well, thank you very much, Maria.
Thank you.
So, thank you for the talk.
Thank you.
Well, it's now my pleasure to introduce our second speaker, Dr. Michael Heneka. Dr. Heneka is a board-certified neurologist and a leading clinician scientist with over 25 years of experience in the study of neurodegenerative diseases, both at the experimental and the clinical level. His work bridges immunology and neuroscience with a primary focus on dementia and Alzheimer's disease, although he has also contributed significantly to research on Parkinson's disease and amyotrophic lateral sclerosis. He has published more than 260 peer-reviewed papers and has led pioneering work that has shaped our understanding on the complex interplay between neuroinflammation and neurodegeneration. Clinically, Dr. Heneka has established specialized outpatient units for neurodegenerative disorders at the University of Münster and Bonn from 2016 to 2021. He served as a head, and from 2016 to 2021, he served as a head of the Department of Neurodegenerative Disease and Geriatric Psychiatry in Bonn. Since January 2022,
I'll show you this little video where a macrophage is exposed to bacteria and he removes this bacteria. This is what our innate immune system does continuously. As I speak, as you listen, and if this system would not work, we probably would not survive a week. We would die by endotoxemia and organ failure. So the innate immune system principally is our first line of defense. And these cells do not only exist in the bloodstream, they exist as resident cells in various organs. In the brain, we call them microglia cells. In the lung, alveolar macrophages, copper cells. In the liver, mesangium cells. In the kidney, beneath cells within others. In the gut, and of course, bone marrow macrophages. They all contribute to stability and function of the organs, but they also protect them from infection. And if you're interested in the history of microglia cells, these cells have been at large described by Pío del Río Ortega, working in Madrid over 100 years ago. And there's a very elegant review some years back by Amanda Sierra on the historical background of his discoveries. Now, what happens in Alzheimer's disease? In a nutshell, what's the biological basis? We have a transmembrane protein, the amyloid precursor protein, which is cleaved by beta and gamma secretase in a sequential manner, and this gives the rise to amyloid beta monomers. The best known of are 1 to 40 and 1 to 42, and they should leave our brain usually by 25% into excretion into the CSF space, 25% into the lymphatic drainage pathway, and the remaining 50% are being taken care of by the local innate immune cells through secretion of degrading enzymes, including IDE, neprilysin. And if this balance gets out of control, these monomers tend to accumulate as oligomers. You can see this shift to higher molecular weight and as A beta fibrils, which then deposit in the brain as these neuritic plaques. This deposition causes a massive inflammatory reaction. I'll come to that why this is. And you see CD68 positive microglia cells and GFAP positive astrocytes at the site of amyloid beta deposition. And ultimately, the disease is characterized by the formation of these neurofibrillary tangles within neurons, which initially mark the dysfunction and ultimately the death of these cells. Now, the question is, when does that start? And so I have a lot of colleagues claiming that the biggest risk to develop Alzheimer's disease is age. And I think there is no sentence that's wronger than that sentence. And I'll prove you why that is. So the question is, 5 years ago, I would have told you that Alzheimer's disease starts the moment where first short-term memory deficits set in. We now know from sequential PET scanning of amyloid beta deposition that it starts way earlier. And this is a sequential movie now. This patient has undergone annual PET scans for beta amyloid. We start 25 years before he develops the first memory deficit. You see now here the precuneus in the brain. You have a sagittal view and a lateral view on the brain. Precuneus, the frontal brain area, the mesiotemporal lobe. We're 15 years prior to the first deficit, 10 years, 5 years. And if it turns to 0, this is the moment the patient recognizes for the first time that something is not going to be right with his memory. Now we go 10 years into disease, and because this disease is being survived in the Western Hemisphere by 10 to 12 years in mean. Now, usually we see cases arising between 60 and 65 years of age. Now, if you go 25 years back, it becomes clear that this disease biologically starts between the 3rd and the 4th decade of life. So if you put this on a whole life, here 8 decades, we now know that amyloid beta starts to deposit here around the 4th decade of life, 3rd to 4th decade of life. We have the tau coming up much later, brain atrophy sets in later, and we know the very bad part of the disease, the clinical part, very well. But it becomes clear that there is a disconnect between the neurobiological start of the disease and the clinical start of the disease. And it's right. The biggest risk for Alzheimer's disease clinical appearance is age, but not for biology. So the point is, this disease starts in midlife or even earlier, and means for everybody older than 35 years of age in this room has already been decided, okay? So we know now that these amyloid beta deposits are accompanied by massive inflammation. So every mechanism we study, every consideration about therapy, we have to take inflammatory mechanisms into account. And this is why. This is showing you a frontal cortex section of a patient suffering from Alzheimer's disease. You see Congo red stains the brain, beta sheet structured amyloids, and there are MHC class II positive immune cells, microglia cells surrounding it. And long before Congo red has been used to stain human brains, has been developed for something completely different. Has been developed to stain shirts by Bayer in Leverkusen, and because that's a very rainy region of the country, turns out that it would not stay in the shirts, would not stay in the cotton. So they put it back into a shelf until a cleaning person by accident would throw that vial to the ground, and leaving the lab floor stained in a very strange fashioned manner. And there was a very young intern, Paul Bötticher, in 1882, coming from Poland, who found out that it would, the cause of that stain would be that Congo red stains bacterial surfaces. So he took the vial, went to Berlin, and tried to license it without telling Bayer, and that sparked a massive lawsuit, and so we know that story so well. But it raises the question, what is the commonality between a bacterial surface and between a human brain? And the commonalities that took 100 years or longer to discover that. It was a science paper by Matthew Chapman showing that bacteria, very common to us, including Escherichia coli, Staph aureus, Salmonella typhimurium, and others, carry beta sheet structured amyloids on their surface. And they pretty much look the same like the beta sheet structured amyloids and have the same structure than the ones that deposit in our brain. So in other words, that means that myeloid cells, including microglia, over 1 million of years through evolution have been taught that a beta sheet structure is nothing else than a bacterial surface and an invasion. So nature has equipped these cells with so-called pattern recognition receptors on their surface to sense these products and sense the beta sheet structured amyloids and to elicit a very strong inflammatory reaction. So now the microglia cells will not distinguish whether this is a bacterial-derived beta sheet structured amyloid or whether it's an intrinsic beta sheet structured amyloid. For the pattern recognition receptor, it's just an evil thing. Now, this comes on the background of various lifestyle factors which fuel into inflammation in the brain, midlife obesity, especially in men, poor oral health, periodontitis, aging itself by secreting a senescence-associated secretory profile, brain trauma as a very direct risk factor. We know that infections or systemic inflammation is causing an acceleration of Alzheimer's and reduced physical activity. Now, over the past years, we have identified more than 96 risk variants in various genes that raise the likelihood of developing Alzheimer's disease in later life. And roughly 50% of these risk variants reside in genes that are important for innate immune signaling pathways. But it doesn't tell us in which cells that happens, right? We do not know. It could be any cell. So this has been answered by very elegant work from Kosoy and colleagues, and they used an attack sequencing transcription footprinting approach, combining that with all the GWAS information accumulated to date, and using an RNA sequencing data set from microglia and other cells isolated from more than 150 patient brains. So and I want you to focus on this red line here, just tells you in which cell type, in neurons or in microglia cells, the risk variant play their important disease contributing role. And you see in Parkinson's disease, there's no role in UN positive cells. There's a little significance for a role of risk variants in microglia cells. In ALS, the major role of risk variants is in neurons. There seems to be a role in microglia cells, doesn't become statistically significant due to the high variability, but there's a tendency. But now look to Alzheimer's disease. In Alzheimer's disease, there's even a negative signal for any role of the GWAS risk variants in neurons, but a very strong role in microglia cells. So all the risk variants identified so far, they do the evil job through altering microglia function. Now, here you can see another way of imaging the human brain, and I've already introduced to the PET, the pipette, which marks amyloid beta deposition, and there's another one which marks activated microglia cells by binding to the TSPO receptor. So you see that in the pre-symptomatic stage, as soon as you have amyloid beta deposition, you have concomitant activation of inflammation. And at a later stage, tau pathology comes to this. Now, for the first part of the presentation, we focus on the interaction between inflammation and beta amyloid deposition. So if a microglia cell senses the beta sheet structure, it becomes activated. It changes their morphology massively. It withdraws all the processes. The cells become roundly shaped. And they start to release a number of inflammatory mediators, including complement factors, cytokines, chemokines, radical oxygen species, nitric oxide, and so on and so forth. Now, what's the role of these factors? And we were interested to study these, but for us it's very important that the factors we are looking at are being conserved between mouse and man. And for the innate and the adaptive immune system, we know that roughly only 30% of the adaptive immune signaling transduction pathways and 40% of the innate immune signaling transduction pathways are being conserved. So we need to be sure that what we look at is really important for the human part of the disease as well. So this is work by Erik Bodeke, and Erik and his team identified the core signature of human microglia by combining 4 different transcriptome data sets, and in the core of the core you find the inflammasomes and their products. And we've become very interested in the NALP3 inflammasome, which is a highly regulated immune signaling transduction pathway upstream of many, many other innate immune responses. And it requires the binding of a toll-like receptor or a cytokine receptor, initiating the transcription of various proforms of cytokines and auto-inhibited NALP3. And if there is no second signal, it stops. If there's a second signal that licenses the buildup of the entire inflammasome, you have the formation of the active inflammasome, the maturation to a so-called ASC speckle, which then has the outward-directed active caspase-1, which cleaves gasdermin D, pro-IL-1 beta, and IL-18. And the gasdermin D cleavage product moves to the membrane, forming a pore complex that allows the coordinated release of these cytokines. Now, years back, we identified this in microglia cells in Alzheimer's disease. You see an ASC speck, an activated inflammasome in a microglia cell, but it didn't tell us anything about the activity. So we had to find out about the degree of activation in the brain, and we started to blot for cleaved caspase-1. If we did so, we found that age-matched non-demented controls would show almost no activation, while Alzheimer's disease cases would show massive activation. And this was already detectable at very, very early stages. Now, luckily, this was reflected in all the mouse models that had been investigated so far. This is the APP-PS1 transgenic mouse model generated by Jane Jankowski and David Borchild. And you see that in an age-dependent manner, around 8 months, we see the cleaved caspase signal coming up. And if we knock down NALP3 in these animals, it disappears completely. Now, in a nutshell, because that's published work, NALP deficiency in this mouse model prevented microglia activation, increased amyloid beta removal, reduced cerebral amyloid beta load, but most importantly, it protected from spine loss. So this is the spine density of a wild-type neuron. This is the spine density in APP mice, and it's completely protected by NALP knockout or caspase-1 knockout. And it protected synaptic plasticity. And this is LTP measured in the hippocampus, which is a very important and highly conserved mechanism that is responsible for memory formation and consolidation of information in our brain. And you see that's entirely protected by NALP knockout. And most importantly, in reflecting the protection of synaptic plasticity, there is this highly protected phenotype in the Morris water maze. So these animals are swimming in this tank where there's a hidden platform. They cannot see the platform. They only have these external cues to orient them, and they do this swimming over 8 days. You see that the wild-type and NALP knockout animals learn the task over 4 days, and the swim distance to the platform gets lower and lower, and then they consolidate that information during the next days. The APP-PS1 mice, they hardly learn anything over this 8 days, and the APP-PS1 NALP knockout animals, they highly behave like the background unaffected animals. At day 9, we remove the platform and we interrogate whether these animals have been followed a spatial navigation search strategy. And so what you see is that wild-type and NALP knockout animals, they confine their search to quadrant 1, where the platform had been located at the 8 previous days, while the APP-PS1 animal has a random search strategy, and the APP-PS1 NALP knockout animals, they're not leaving the quadrant 1. their memory function is entirely preserved. So just knocking out a single inflammatory molecule preserves the entire memory phenotype in these animals. Now, at the plaque site, there is more than such inflammation and inflammatory mediator production. There is also the death of these cells. The encounter of these inflammatory products and the continued fight against the beta sheet structured amyloid will lead to the cell death of these microglia cells. And this is one experiment where we expose microglia cells to amyloid beta. You see interleukin-1 beta goes up as a sign of inflammasome activation. And then we have the breakdown of mitochondrial respiration of the microglia cells. You see there's a strong reduction of respiration and energy breakdown. And these cells stop taking up beta amyloid in this case. So they're unable to clear and degrade amyloid beta due to the compromise of their energy metabolism. But ultimately, the compromise of the energy metabolism is leading to the cell death and to the release of the ASC spec. So the inflammasome is being released, and you will see here in this movie, this cell releases a spec which is becoming yellow, and it's becoming yellow because we put TAMRA-labeled amyloid beta in the supernatant, and you see that there's halo formation very quickly. So obviously, the ASC spec is binding very, very rapidly to beta amyloid, and that was entirely new. So that leads to the question whether the amyloid beta deposits, which we see in the patient brain, is there in a random fashion, or whether there's a reason to it. And so we hypothesized at that time that a microglia cell that would undergo an activation from the homeostatic stage to an activated stage undergo microglial pyroptosis, so cell death, releases the ASC spec. And the ASC spec then obviously is an ideal matrix for amyloid beta oligomerization and eventually amyloid deposition. So the claim here was that an amyloid beta deposit would not occur at a random site, but would require a death of a microglia cell before. Now, this is showing you the first assay we then developed to look at this phenomenon, and we have now a cell culture dish here with 4 amyloid beta deposits, all of which contain an ASC spec in the core. And you see that the beta sheet structured amyloid labeled with TAMRA forms the halo again. And this is the first cell culture approach to study amyloid beta deposition now. And you see at day 4, we just have this very tiny ring of beta sheet structured amyloid surrounding the ASC spec. At day 8, it has grown substantially. At day 9, there is only a tiny bit to see, but there's a whole kind of basket of beta sheet structured amyloids surrounding the ASC spec. Now, this led to experiments where we tried to prove this, and this is work in collaboration with Jochen Walter and Satish Kumar. And what they did is they exposed A-beta 1 to 40 or 42 alone or with ASC specs or ASC alone to a time course experiment at time point 0. Everything is in the supernatant, and you see after 6 hours, everything has went into the pellet. So this is depicted here. The ASC spec would drag the amyloid beta into a plaque-like structure. And these results prompted experiments in a mouse model where we showed that in an age-dependent manner, only in the transgenic mouse model, we will see that by co-immunoprecipitation, ASC would bind to amyloid beta. But most importantly, when we looked at the immunohistochemistry of these animals, we would see that the core of each of these mouse plaques would be made of ASC, and the A-beta would form a halo surrounding it. Now, we were not absolutely convinced. We thought this is, we should exclude that this is wishful thinking, so we developed another mouse model, and this is APP mice, CX3, CR1, EGFP animals, which have all the microglia cells in green, and we had a knock-in ASC M-cherry mouse crossed into this. So we hate to kill animals in my lab, so we stopped doing this, and we now basically, animals live a normal mouse life. They get a cranial window, and we put them under the microscope. They walk the treadmill here. They see a spatial navigation task, so we can visualize the brain during a task. We even can visualize the hippocampus during memory formation. And what we get then is a movie that looks like this. And here you see the microglia cell movement. You see the ASC specks in yellow in the microglia cells. You see the released ASC specks in red at different positions. You can see the release of the ASC spec due to the microglia death. And when we inject methoxy-XO4, we can visualize the amyloid beta in the brain under living conditions. So we don't have to kill the mouse. We just do videos of a certain part of the brain, and we can do this every day, every week, every month throughout the entire mouse life. And we can test memory function along with it, as an example. Now, you already see that some of the aspects are surrounded by a bluish stain here. And to better see this, we removed the green filter. And now you see this next movie, and you will see that each of the blue amyloid beta accumulations contains an ASC core. There is not a single amyloid beta deposit which would not have an ASC cherry core, proving our previous observation that the foundation of a microglia deposit is the death of a microglia cell. Now, this is a time-lapse experiment, and again, you can see you don't have a single plaque which does not contain an ASC core, but we wanted to further substantiate our claim, and so we said, well, if we are right, then initially the ratio here is closer than at the end of the experiment, and the ratio of the core to the surroundings would drop over time. And this is exactly what's not being displaying here because it's a Mac program. But what you see is here is the mean of the second observation. So the ratio of ASC volume to A beta drops massively down over the experiment, proving that once the ASC has seeded the deposition, the ASC would not change, but the A beta would grow. We now developed a program where we can analyze these movies. It's we can't count these. There are too many plaques, and over many months of mouse life, we developed this program that depicts single positions in the brain and can then follow up the ASC spec release, the microglia paroptosis, and A beta deposition. And this is if any one of you is doing such experiments with in vivo mouse brain imaging, the program is free to access at fairsharing.org, and you're happy to use the code. Now, we don't stop here. This is a human amyloid beta deposit, and we sectioned more than 60 patient brains. And what you see is each core of a human amyloid beta deposit is made of ASC. And you see the 6E10 positive amyloid beta surrounding it. So we started to develop humanized antibodies targeting the interaction between ASC and A beta, because we believe that this is a better target than A beta itself. So what you see here is that A beta aggregates. If you compare the green surfaces, you see there's aggregation is much lower than here, especially after 4 and 8 hours. And then you see that M-cherry itself has no function, so it's the ASC which is driving the aggregation. You also see that not all the antibodies work. Antibody 1, developed by us, was a clear failure, but antibody 2 was pretty good in suppressing the formation of oligomers and fibrils, and we're moving this now into clinics. Now, ultimately, there is proliferation. If you have death at the plaque site in the immune encounter, you also stimulate proliferation. And so microglia cell proliferation is a long debated problem, but it has been massively advanced by this very nice experiment by Jonas Friesen. And Jonas initially had a totally different target. This is experiments that build on the fact that the land of the free, has donated to human mankind supraterrestrial nuclear bombing in the 50s, destroying the Bikini Atoll. And each of these nuclear detonations was leading to a C-14 fallout, which was covering the entire planet. But you have to know that each nuclear bomb has a very own C-14 fingerprint. And each of the proliferating cells in our body had incorporated that nuclear dust. So Jonas went to the Karolinska Institute, and they detected in proliferating cells in the body, in various cells, the time points when the C14 fallout was incorporated into the body. And the initial aim of this experiment was to test whether neuronal precursors would be turned into intact neurons. And the neuron experiment didn't come out great for him. But he had a great deal with microglia cells, and so he was able to determine that human microglia renew at the median rate of 28% per year. So imagine 1/4 of the microglia cells in your brain is exchanging during a year. And they live, on average, 4.2 years, and most of the microglia population will renew throughout your entire life. Now, this is a fundamental change, because now imagine a microglia cell that would proliferate into a healthy brain and a microglia cell that would proliferate into a degenerating, inflamed brain. This is a different thing. The microglia cells that proliferate into this degenerating brain is like a kid born into a war zone. It will behave differently. It gets different cues, different signaling. It will most likely develop a different response. Now, this was what we tested, and we found that already at 6 months, we have a massive change of the entire transcriptome of the proliferating cell pool. And you see here that we regulate cellular responses to LPS, regulation of phagocytosis, regulation of neuron death, antigen processing and presentation via MHC class II, and regulation of leukocyte differentiation. All major pathways important for immune function and host defense in the brain. At 12 months, this effect was even stronger, and the network was much about TARA-BP and TREM2 and catepsins here, so lysosomal pathways became important as well. Now, turns out that over this time, if we compared the APP mice and the versus the proliferating cells in wild type, they would have a tolerogenic phenotype that would get lost over time. So the same comparison at 12 months would show that the tolerogenic genes were downregulated, while the genes related to neurodegeneration would be massively increased. And some years back, in a paper, in a seminal paper by Karen Schall and Ido Amit in Cell, there was the so-called DAM signature identified, which stands for degeneration-associated microglia signature. And you see that at the beginning, already at 6 months, we have roughly 20% of these cells which are identical between the DAM signature and the proliferation signature. And it increases massively to 50% at 12 months. So what we believe is that the DAM signature is actually driven by proliferation. So it's not the single microglia cell which will change over time, it's the population as whole through generating different generations. And so we then tested the function of these cells. We were interested, would a microglia cell which would have undergone proliferation, would the daughter generation be equally well in taking down and degrading beta amyloid peptides? And they don't. And interestingly, you see here that there's a massive difference. The cells that proliferate, the key I67 positive cells, they hardly take up any methoxy-XO4 material, while the cells that have not proliferating still do. It becomes more obvious at 12 months. And then the interesting part is here, we see that these cells, which are compromised in the phagocytic clearance, they start to express the entire inflammasome pathway. So all the members we had been interested before. So we knock down ASC in these cells, and we gain back a part of the proliferation function. So, which means that we target inflammation, we suppress the inflammatory response in these cells, we gain back some of the protective function of these cells. Now, what about this second thing? I told you this is the beginning, but now we go into tau pathology. And tau pathology is the pathology which is more relevant to the decrease of neuronal function. It's highly correlated with cognitive decline. So to address that, we adopted a model that had been introduced by Jürgen Götz and published in Science over almost a quarter of a century ago. And we adopted, Christina used that model and injected either solvent control or APP-PS1 brain lysate into opposing hippocampi. And these was done in tau mutant mice. And what we wanted to see whether we see a spread of pathology in response to APP-PS1 lysate injection. And the question here was, would amyloid beta drive tau pathology, yes or no? And if so, would there be a role for inflammation? And what we found was that yes, the wild-type brain hemisphere lysate receiving hippocampus would show only a bit of pathology, while this was massively increased in the hemisphere that received the APP-PS1 brain lysate containing beta amyloid. And when we knocked down ASC or NALP3, this increase was entirely gone. That's the quantification. The red bars compare the control to the APP in the tau 22 mice, and then you see the ASC and NALP knockout animals which do not show such an increase. So this experiment suggested that the innate immune system is not only responsible for the seeding of the initial pathology, it's also responsible for driving the pathology in the tau mutant part of the disease. And this aligns very nicely with a paper by Darren Baker, who has published in 2018 that senescence and release of interleukin-1 beta by senescent microglia cells would drive pathology in the neurons. Now, I summarized that paper for time reasons, but what we showed is that we have this immune activation of the microglia cells in response to pattern recognition ligation and drive interleukin-1 beta release, and in an interleukin-1 receptor-dependent pathway via MIDI88, IREC4, MAC1-2, we get phosphorylation of CAM kinase 2 alpha, which hyperphosphorylates tau, which leads to the loss of tau binding to the microtubule, formation of paired helical filaments, and neurofibrillary tangle formation. Now, what I haven't told you initially, I told you the view of Alzheimer's into the microscope. And what I haven't told you, that these gentlemen, Alois Alzheimer and Franz Nissel, a neuropathologist in Munich at that time, have depicted like the beta sheet-structured amyloids and the tau on one page. The remaining 9 pages of this publication contain all these beautiful pictures about immune cells in the brain. And you see the astrocytic reaction, and you see here, he called the microglia cells at that time amoeboid glial cells. You see that the amoeboid glial cells interact with the neurons. You also see that they depicted the suffering of these cells very, very nicely. So none of that is new. I think we just forgot about it for 100 years and a bit more. But it will become highly relevant. So in a nutshell, I tried to convince you that the NALP inflammasome is a key innate immune signaling pathway which is activated in Alzheimer's disease, has been reproduced now by many. NALP3 knockout improves amyloid beta clearance, improves hippocampal long-term potentiation and spatial learning and memory performance in mouse models. It's been shown by various other groups by now, and more recently with the first NALP inflammasome inhibitor. ASC-specs seed A-beta deposition in vitro and in vivo, and I think this is important because that tells us that the true target is not beta amyloid. The true target is the ASC-spec. Now, ASC A-beta can be targeted by antibodies, and those are already humanized. I've shown you not everyone works. A-beta-1 was a failure, A-beta-2 was great. NAB3-derived interleukin-1 beta drives tau phosphorylation in neurons, and several pharmaceutical companies, Sanofi, Merck, others, are preparing clinical phase 2A trials, which will start next year in the AD arena. I'm not very happy about that because I think it's still a bit too early. I told you that the disease trajectory encompasses 40 years, 4 decades. I'm not sure at which time point we have the ideal window of initiating such a treatment. I'm afraid that the pharmaceutical companies want to jump on this too quickly and make a good target worse by, proving or disapproving their function already. So I believe that we still need some more insights there. Co-development of anti-ASC A-beta antibodies with AC Immune and Mabilon may be a different kind of strategy to target the deposition, which is at the beginning of the disease. Now, these are the people of my former department in Bonn who contributed to the work, all our great collaborators and the people funding this work. This is the new team in Luxembourg. It's a fun group, so if you want to join us or just to visit us, you're welcome to come to the smallest country in Europe, only 680,000 people, a little city. Probably would be Leganés or so in Madrid. With this, I thank you for your attention.
Thank you very much, Dr. Henneka. It was a great talk, very inspiring. I'm not sure if there is any question in the audience.
Thank you, Michael. That was fascinating. I was wondering, you showed us very clearly that the release of ASC is critical for the propagation, for the initiation of this disease setting, but also that microglia are, one of the professional tasks is to clear A-beta. What leads to the switch? Why do microglia all of a sudden stop performing this professional task? And the second question is, what are the biochemical mechanisms that promote the formation of these structures, these halos, once ASC is released? So intracellularly, it's part of the biochemical mechanism of trying to identifying A-beta as a danger signal. But once that comes out, why is this interaction continuing?
So the first question is probably let me start with the last question. Nature has developed this ASC-spec release phenotype for a good purpose. Because imagine a skin wound. In the skin wound, you have invading Staphylococcus aureus, and usually at 37 degrees, they would double in 20 minutes. Right. So you've seen that mounting the immune response takes hours. So the immune system and the host defense will fall behind the velocity of the bacterial spread. And for that, the ASC-specs can be released, and invading leukocytes can take the ASC-spec up and have a ready-to-fight inflammasome. So they don't have to go through these initiating and licensing steps, which I showed on the schematic. They are much faster, and they can limit the inflammation on the skin wound. And for a host defense, for a real host defense against living bacteria, that makes sense. What we have in Alzheimer's is a completely different situation. It's a sterile type of inflammation. There are no bacteria around. There is something that mimics the bacterial cell wall component. And therefore, we have this continued release of ASC-specs. Now, and I didn't show that, but we showed in 2021 that in the very early phase, at the beginning of this immune encounter, around 2 or 3 decades prior to the cognitive decline, we have an innate immune response of the microglia cells driven by TAM receptors, which is protective. And as stronger this immune reaction is, as more stable is the cognition and more stable is brain volume. Those of us which will not mount a strong TAM receptor response will go down in cognition and will have faster atrophy of the brain than the ones which have a strong, but that's the initial phase. And you know, inflammation is there to protect us, but not chronic inflammation. As for most of the body and organs we have and know of, the chronic type of inflammation becomes damaging. And this is the same thing in the brain.
Wonderful. Thank you very much.
Okay. Thank you very much for this interesting talk. I would like to ask you if this mechanism is general mechanism for other neurodegenerative diseases, or how do you explain it for other diseases, neurodegenerative diseases with intracellular aggregates of proteins?
That's an almost political question. The reason is that I'm a bit in, there is already one trial going on in Parkinson's patients, and with Roche, with a NAB inflammasome inhibitor, and I'm in a constant exchange with them, and I claim that they started too fast. You may have heard that out of my, they claim it worked. They will release the data at the Movement Disorder Conference in September. They claimed it's great. I haven't seen the evidence of a massive inflammasome activation in Parkinson's disease. I might be wrong. I believe more of an adaptive role in Parkinson's disease. And to me, there is, to date, no substantial publication which would show that there is activation of the inflammasome in the substantia nigra pars compacta, which is affected by the disease. For frontotemporal dementia, it's different. In FTD, we see a very similar phenotype that leads to tau phosphorylation and blockage of the NALB3 inflammasome have prevented disease phenotypes in two different models which carry mutations of familiar forms of frontotemporal dementia. For Huntington's disease and other diseases, and ALS, I don't know, because it has not been tested. There are papers here and there, but I think it needs, you know, I want to see an amount of confirmations by various groups in order to comment on that.
Hi, thank you for the great talk. I would like to know your opinion about this last paper showing the prevention of dementia in people that receive the vaccine against herpes virus, and if this has some relation with this mechanism?
Yes and no. Yes and no. So there are two things. So that's complicated. The NALB3 is a canonical sensor of bacterial products until June 2025. So it just came from an innate immune conference where somebody showed the data that actually the NALB inflammasome can sense viral particles, and especially varicella and herpes simplex virus particles as well. We have some more clinical data showing that herpes simplex virus is also likely to drive Alzheimer's disease, like the publication 4 weeks ago in Nature about varicella virus. And so the question is it truly the NALB3 inflammasome? There's another inflammasome where we have a large operation on, the NALB1 inflammasome, which is the classical sensor of viruses in the microglia cell, in the myeloid cells. And we believe there's a strong role for the NALB1 inflammasome. For me, it's still likely that the NALB1 inflammasome which is also belonging to the core signature of human microglia, is a driving force behind that, and and it's also leading to ASC-SPEC release. So you could consider that you have initially a latent virus infection. We experience throughout our life various latent virus infections, including the brain, so that one of these very mild infections spark the initial response, and then the system starts to avalanche through the brain by this ASC-A beta interaction mechanism. When exactly the NAB inflammasome mechanism sets in, I cannot tell you. That's why I'm thinking we need to have more information along the trajectory of the disease. But it may well be that it's not the single or only inflammasome that contributes disease, and that viral particles do the very same. I learned 2 days ago that NAB3 senses viral particles as well. I want to see that in my own hands. But there's another inflammasome, which is the NAB1 inflammasome, who is engineered to do so and has a strong role in the disease. Maybe two. You know, they're more or less two sides of the coin.
Well, thank you very much. Now, thank you, Michael, for your talk and for the questions. Now we have a coffee break downstairs in floor 0. So please be back on time. We will start next session at 11:30. Those attending online also, we will start again at 11:30. Please be here like a couple minutes in advance. Thank you very much.
La innovación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico, estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IS Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación a consumidor. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
La innovación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico, estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IS Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación a consumidor. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
innovación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico, estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IES Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación a consumidores. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
innovación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico, estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IS Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación a consumidor. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
Okay. Hello again. We are going to continue with our program, and the next speaker is Dr. Xavier Aguirre. As you may notice, we have had a change in the original program. Dr. Felipe Prosper cannot be today here with us, and we have a new speaker, that is Dr. Xavier Aguirre, a collaborator of Dr. Prosper. Dr. Xavier Aguirre obtained his PhD at the Department of Genetics at the University of Navarra. After that, he completed his postdoctoral training in the laboratory of Dr. Felipe Prosper at the Center for Applied Medical Research, CIMA, of the University of Navarra. He has also performed several research stays in several international laboratories, such as the laboratory of Professor Rainer Siebert at the Institute of Human Genetics in Germany, and in the laboratory of Dr. Ari Melkit at the Weill Cornell Medical College in New York. From 2004 to date, Dr. Aguirre has been the principal investigator and head of the Cancer Epigenetic Mechanisms Laboratory of CIMA in the oncohematology program. In addition, during several years, he was the director of the genomic unit of the CIMA Lab Diagnostic. Dr. Xavier Aguirre will talk today about his research on the metabolic transcriptome analysis for discovery of novel therapeutic targets and biomarkers in multiple myeloma. Please join me in welcoming Dr. Xavier Aguirre.
Thank you for the introduction, Maria Jesus. Okay, first of all, I would like to thank the organizers for inviting me to this workshop and giving me the opportunity to show you the results of our study about the metabolic transcriptome in multiple myeloma. And multiple myeloma is an aggressive hematologic neoplasm that represents about 10% of human hematologic tumors. It's most frequent in elderly patients with the median age at diagnosis around 65, 70 years. It's characterized by a malignant and clonal transformation of plasma cells in the bone marrow. And this disease is genetically, epigenetically, and clinically a very, very heterogeneous disease. Genetically, we can classify multiple myeloma in two groups, the non-hyperdiploid and hyperdiploid group. In the case of the non-hyperdiploid, we can detect different translocations, like a translocation 414, translocation 814, in all of the cases implicating the immunoglobulin gene, and mutation in some particular genes, like a RAS or TP53. But this genetic alteration is very low frequent among multiple myeloma patients. In the case of epigenetic, it's very similar that genetic alteration. We detect some common alteration and DNA methylation and histone modification level in multiple myeloma patients. But globally, multiple myeloma patients show a very heterogeneous landscape of epigenetic alteration. For this reason, it's very complicated to have a group of patients with the same alteration, more than the same alteration with the same druggable alteration to have a treatment for these patients. And despite the advances made in recent years in therapy of multiple myeloma, this disease remains incurable, with the median survival around 7 years. One of the reasons of this worse prognosis of multiple myeloma patients, at least in part, is this heterogeneity that we have at genetic and epigenetic level, because we don't have, or we don't can identify or define group of patients to have a specific treatment for this patient. As you know, besides genetic or genetic alterations, one of the most important hallmarks of the cancer is the deregulation of cellular metabolism. Some groups of some studies have shown distinct metabolic vulnerabilities in human tumors, especially in solid tumors, showing a clear potential as biomarkers and also as therapeutic targets. But this is not well analyzed in the case of multiple myeloma, because in this disease, we don't have a large scale of metabolomic studies or data to analyze this type of deregulations. And for this reason, in this study, we decided to analyze the metabolisms of the metabolic alterations in multiple myeloma with this idea to identify new metabolic vulnerabilities that could have a very important role in the multiple myeloma cells and could be new therapeutic targets to develop new therapeutic strategies to treat these multiple myeloma patients. Initially, we would like to perform a global metabolomic study, but this is very complicated in the case of the multiple myeloma because it's very difficult to obtain a large number of plasma cells from the bone marrow of multiple myeloma patients, and more to obtain normal plasma cells from the bone marrow of healthy donors to compare the metabolomic alteration between normal and multiple myeloma plasma cells. And at this point, we thought that one of the approaches that we could use to analyze the metabolisms in multiple myeloma could be the study of the transcriptome or genes involved or related with these metabolisms. And in this regard, in our previous studies, in collaboration with Dr. Francis from Tecno Universidad de Navarra in San Sebastián, we developed different computational tools to identify essential metabolic genes in multiple myeloma. These computational methods are based in the synthetic lethal analysis. Synthetic lethality occurs when we need the inhibition of two genes to reduce the viability of the cells of kills these tumoral cells. Because if we inhibit only one of these genes, this inhibition is tolerable with the cell viability. Using this computational model, we identify distinct synthetic lethals, but interestingly, when we add the expression of genes implicated in these synthetic lethals, we were able to identify essential metabolic genes for the multiple myeloma cells, as I showed you in this figure. We found that CTPS1 and CTPS2 conform and synthetic lethal. So if we inhibit the expression of these two genes, we should reduce the cell viability. But when we add the expression of these two genes obtained from different B-cell normal subpopulation and plasma cells from the multiple myeloma, we observed that in this particular patient, CTPS1 was an essential gene because this patient didn't express CTPS2. So in this patient, if we inhibit only CTPS1, we should reduce the cell viability. And we validate this idea using multiple myeloma cell lines. In these two cell lines, without CTPS2 expression, when we inhibit using the CRISPR guides or a specific inhibitor of CTPS1, we observe a significant reduce of cell viability, but not in this cell line with CTPS2 expression, demonstrating that CTPS1 is an essential gene because its inhibition decreases cell viability. These analyses are interesting to identify some synthetic lethal or essential genes, but are limited to synthetic lethals at these specific computational models. We don't detect all the vulnerabilities. And for this reason, we decided to analyze all the metabolomic in multiple myeloma to have more information about the metabolomic or metabolism alteration in multiple myeloma cells. And to do that, we analyzed the expression around 3,000 genes related with the metabolisms in different B-cell normal subpopulations that participate in the terminal B-cell differentiation or humoral immune response from the naive to bone marrow plasma cells, and also bone marrow plasma cells from multiple myeloma patients. And we observed that plasma cells showed different metabolomic transcriptome than the other B cells. And multiple myeloma plasma cells showed a similar metabolic transcriptome than normal bone marrow plasma cell, as we expected, but with a huge heterogeneity among multiple myeloma patients. And we thought that if we analyze more patients or with multiple myeloma, probably we could identify a group of patients with a specific metabolic transcriptome or metabolic vulnerabilities. And to do that, we analyzed the RNA-seq data of 600 patients of COMPASS cohort. And using the expression of these 3,000 genes related with the metabolisms and different computational methods like bimodal index or non-negative matrix factorization, we classify multiple myeloma patients in 6 distinct groups showing a different metabolic transcriptome or metabolic genes profile. Interestingly, these metabolic genes on this profile were associated with specific metabolic pathways or characteristics. Group was associated with inositol metabolism, group 2 with glycan metabolism, group 3 with cholesterol biosynthesis, group 4 with gluconeogenesis, group 5 with fatty acid metabolism, and group 6 with amino acid metabolism. But curiously, when we integrate the cytogenetic data of these multiple myeloma patients, we detect a big correlation between these 6 metabolic groups and well-defined cytogenetic alteration identified in multiple myeloma patients. The first group was also enriched in patients showing translocation with cycling genes, like a translocation 1114, like you can see here. The second group was enriched in patients with translocation 414. The third group with translocation involving MAF transcription factors, like a translocation 1416 or 1420. The fourth group with translocation including the MYC oncogene, like a translocation 814. And the fifth and sixth group showing as a more relevant cytogenetic alteration the hyperdiploidy. This is very interesting because we integrate the genetic and also the metabolomic data in multiple myeloma patients, identifying 6 different well-defined myeloma groups. And we demonstrated we found the same results in another 2 cohorts of multiple myeloma patients. And when we analyze the impact of these metabolic profiles or metabolic transcriptome in the survival of multiple myeloma patients, we observed more or less the expected results. But interestingly, we detect differences between 2 groups showing the hyperdiploidy as a more relevant cytogenetic alteration, group 5 and group 6. Group 5 showing significantly worse survival than the group 6. These 2 groups are equal at cytogenetic levels, and the difference is at metabolic transcriptome level. And we continue analyzing the transcriptome of these 2 genes to know if this transcriptome could define the reason of these differences at survival level. And when we compare the transcriptome of these 2 groups, group 5 and group 6, we observe that patients or plasma cells of patients of group 5 were enriched in myeloid pathways related with myeloid genes or myeloid cells, especially in myeloid progenitor cells, showing higher expression of myeloid genes or myeloid-specific erythroid, monocyte, or neutrophil genes. This is very interesting because multiple myeloma is a lymphoid malignancy. It's not a myeloid malignancy. And why these patients show myeloid characteristics? One explanation could be that probably these plasma cells were dormant plasma cells because the dormant plasma cell show an immune and myeloid gene expression. And when we analyze if these plasma cells from group 5 show this characteristic of dormant cells, effectively, we observe that group 5 plasma cells show higher expression of these dormant cells, demonstrating that group 5, in addition to show a specific transcriptome of metabolic gene, exhibit myeloid progenitor and dormant cell characteristics. We asked if we could have some possible connection between these characteristics, metabolic, myeloid, and dormant characteristics, and we analyzed the regulons, and we detect that myeloid transcription factors, non-lymphoid myeloid transcription factors, regulate the expression of these metabolic, myeloid, and dormant genes in plasma cells of group 5 patients, showing that these genes, myeloid, metabolic, or dormant genes, are more expressed in patients of group 5 in these 3 cohorts of multiple myeloma patients. Here we ask if we could use these characteristics of myeloid or dormant or metabolic genes to define better this group of MM5 patients, like a biomarkers, or if we could use these characteristics as targets for developing new therapeutic strategies. when we analyze the expression of myeloid genes in this patient, we observe that hyperdiploid patient, or patient showing like a hyperdiploid, like a more relevant genetic alteration, with a high expression of CD13 or CD33, two myeloid genes, show a significantly lower survival than patient with low expression of these myeloid genes. And 75% of patients in this group were patients for the group 5, demonstrating that these myeloid genes could be used as biomarkers to identify these MM5 patients. Like a targets, we use the bestatin, is the CD13 inhibitor, to analyze if the treatment with bestatin on myeloma cells could have some effect. And we observe that when we treat CD13 positive myeloma cells with bestatin, this treatment reduced significantly the CD13 positive myeloma cells. And we observe the similar result when we use CD33 CAR T cells. In this experiment, we analyze this cell line, which is an acute myeloid leukemia cell line, as a control, because it's CD33 positive cell lines, like we can see here. One regular multiple myeloma cell line. This cell line don't express CD13 and express PSMA, that is the regular marker in multiple myeloma, and this cell lines expressing CD30, similar to patient of group 5. And we treat these cell lines with two different CAR Ts, CD33 CAR T and BCMA CAR T, which is the CAR T that we use for the treatment of multiple myeloma patients. And we observe in acute myeloid leukemia cell line, only the CD33 CAR T cells reduce the number of leukemia cells. In the case of regular multiple myeloma, only the BCMA CAR T cells reduce these myeloma cell lines, but in this case, with CD33 positive, only the CD33 CAR T cells reduce the number of myeloma cell lines, indicating that these myeloid markers could be an interesting target or to develop new strategies for this patient with the group 5 of multiple myeloma. In the case of metabolic genes, we focus in ACLCL1, which is higher expressed in the group 5, like you can see here in these 3 different cohorts of multiple myeloma. This gene converts free long-chain fatty acids into fatty acid-CoA and play a key role in the lipid biosynthesis and fatty acid degradation. The same, we observe that patients showing the hyperdiploidia as a more relevant cytogenetic alteration with high expression of ACLCL1 showed a significantly lower survival than the patient with low expression of this gene. And in this case, 100% of the patients were patients of the group 5, indicating that this marker is a good marker to identify this patient. When we inhibit the expression of ACLCL1 with CRISPR guides, we observe a significant decrease of cell viability, so this gene is essential in multiple myeloma. And when we analyze the transcriptome after the inhibition of ACLCL1, we observe, as expected, a reduction of fatty acid-CoA biosynthesis and also cell cycle. And interestingly, we also observe a reduction of respiration of mitochondria that we validate using the seahorse. Like here, we can see that after inhibition of ACLCL1, the respiration of mitochondria, the basal and minimal respiration capacity, reduced significantly. This is interesting because we know that in multiple myeloma, this situation is important because in this situation, the anti-apoptotic genes from the family of BCL2 are more accessible. And in this case, the drugs that target these anti-apoptotic genes can have more activity in these multiple myeloma cells. And to demonstrate this, we inhibit ACLCL1 with CRISPR guides or a specific inhibitor, triaxine, and combine this inhibition with venetoclax, that is the BCL2 inhibitor, or this inhibitor of MCL1. And in these two cases, we observed a significant synergy between the combination of the ACLC1 inhibition and treatment with venetoclax or MCL1 inhibitor. I show you here one cell line, but we have the same result in different cell lines, showing that ACLC1 is an essential metabolic gene, and the combination of ACLC1 inhibition with anti-apoptotic inhibitor is a promising therapy for the treatment of MF5 patients. And since we might have a treatment for this patient, we decide to look for the best marker to identify this patient among patients showing the hyperdiploidy as a most relevant cytogenetic alteration. We choose this patient, and we divide this patient as a training set and test set, and we analyze the differentially expressed genes between these two groups, searching those genes with extreme values, with the idea to have a black and white, genes that are highly expressed in the group 5 and not expressed in the group 6. We detect these 3 genes. We analyze the correlation of the expression of this gene with the idea to eliminate those that are totally correlated, because probably we are analyzing the same, and we found only this gene, S100A8, which is implicated in the inflammation, like the best marker to identify this MM5 patient. When we analyze the expression of this gene in the test cell, the analysis of this expression allows us to identify MM5 patient with an accuracy of 90%. And when we analyze the same in the Helsinki multiple myeloma cohort, we were able to detect MM5 patient with a 97% of accuracy, demonstrating that probably the analysis of these unique genes is enough to identify this patient with these myeloid, metabolic, and dormant characteristics. As a summary, we can say that multiple myeloma is heterogeneous at the level of transcriptome profile of the metabolic genes, showing 6 different groups, related with well-defined genetic alteration. We have identified 2 hyperdiploid subgroups with distinct metabolic transcriptome, exhibiting different survival outcomes with notably poorer prognosis observed to MM5 patients. Plasma cells from patients of MM5 exhibit specific metabolic, myeloid, and dormant characteristics regulated by myeloid-specific transcription factors. Myeloid genes are interesting biomarkers and therapeutic targets to MM5 patients, and ACLC1 is an essential metabolic gene, and the combination of ACLC1 with anti-apoptotic inhibitors is a promising therapy for the treatment of this patient. And finally, I would like to thank the people of our group and all the collaborators. Thank you very much.
Thank you very much, Javier, for this excellent presentation of your research. Now we have time for some questions. Somebody in the audience here have some questions?
Thank you, Xavi, for the awesome presentation. One question, what approach do you use to separate the patients regarding the expression, for example, of ACLC1, I think it was, to do the survival plots? Like, what do you use to stratify patients with high expression and low expression?
We use 3 different analyses. We use the median. We use also the quartile separation. And we also use the computational model that separate the groups with the cutoff, with a better cutoff to separate the genes. And we also say that this is significant when we had this signification in these 3 models, right?
Hi, thank you very much for that talk. I was wondering, you showed us that there is this myeloid bias in at least one of the subsets of the plasma cells. Do you think that is in the hematopoietic cascade, the plasma cells arise from the lymphoid lineage? Do you think that somehow during the differentiation from the stem cells, there is an alteration already in the bone marrow where these cells are picking up a phenotype, and whether this is potentially linked with CHIP that's been reported to contribute to many other conditions, including some of the other cancers?
Yeah. It's a good question, but we don't have this data. But something like that. Probably we can have some alteration in this hematopoietic differentiation, and probably we can something in these progenitors that probably then come back to the lymphoid differentiation more than the myeloid differentiation, and probably for this reason, in these cases, we have these characteristics.
Following on from that, is there any reports around changes in the hematopoietic niches within the bone marrow of either experimental settings or in patients? Because the niche is also known to be critical in imprinting the phenotype of the progeny. So is there any evidence for niche alterations in these patients?
We don't have this data. We are analyzing in the group the niche of the multiple myeloma, analyzing some niche subpopulation, but in this patient, we don't have this data to say that. Yeah.
Thank you.
Thank you.
We have time for one more question. No? Okay, then. Thank you very much, Xavi. And we are moving to the next speaker. It's my pleasure to introduce Dr. Rubén Nogueiras. Rubén Nogueiras completed his PhD at the University of Santiago de Compostela. And after finishing his postdoctoral research in different institutions in Germany, in Switzerland, and in USA, he joined again the Spanish Research and Development System as a Ramón y Cajal researcher. In 2014, he became an associate professor, and later he has been a research opportunity professor. Dr. Nogueiras is the PI of the Molecular Metabolism Group at CIMUS, University of Santiago de Compostela. He has led a relevant number of national and European research projects and has published a high number of outstanding scientific papers. His laboratory investigates the molecular mechanisms involved in the development of obesity and its associated disorders such as type 2 diabetes and metabolic dysfunction-associated liver disease, with a particular focus on glucose and fatty acid metabolisms in liver. Please join me in welcoming Dr. Rubén Nogueiras.
Invitation to be here, sorry. It's a real pleasure. So, it is very well known that cell cycle regulators are also involved not only in the proliferation of the cells, but also in the metabolism of the cells. And this has been extensively studied in the field of cancer because this proliferation requires high amounts of energy, and therefore it makes sense that the metabolism is reprogrammed in these cells. But during the last years, we have been interested in studying the potential role of these cell cycle regulators in conditions that have nothing to do with cancer. And when we think about cell cycle regulators, many of us will think about P53. P53 is, as you may know, mutations in this gene are probably the most prevalent mutations in many different types of cancer. It is called the guardian of the genome. It's very relevant for many functions. But P53 is not alone. It's a member of a family composed by three different members. It's P53, P63, and P73. And there are several works suggesting or demonstrating that these family members can have different metabolic actions. And again, these metabolic actions have been mainly studied in cancer cells. But we have translated that knowledge to more physiological conditions and then to other pathophysiological states that are completely different to cancer. So During the next minutes, I will talk about the role of these family members, mainly P53 and P63, in two metabolic diseases. The first one will be muscle D, metabolic-associated steatotic liver disease, and the second one will be type 2 diabetes. So our work concerning the role of these family members in metabolism started with this article published by Begoña a long time ago, suggesting that P53 and P63 have completely opposite actions in the modulation of fatty acid metabolism. So if we inactivate P53, P63 levels will be upregulated. This is going to trigger inflammation, ER stress, and therefore it will favor the storage of fatty acids. Contrary, if we activate P53 only in the liver, the levels of P63 will be downregulated, therefore inflammation, ER stress will be low, and we will improve the lipid storage in the liver. So for this study, we generated different animal models, and during the subsequent years, we analyzed by proteomics the proteome of mice overexpressing and downregulating P63 in hepatocytes. And one of the proteins that caught our attention was ATG3, autophagy-related gene 3. And the reason for that is that it has been well established that autophagy is important in the field of muscle D. There are many alterations in autophagy-related genes in conditions of lipid steatosis, liver steatosis, but nothing was really known about the role of this specific gene. So we carry out some further experiments, first measuring the expression of ATG3 in the liver of patients at different stages of the disease, because muscle D is a progressive disease, starting with steatosis, then inflammation, fibrosis, cirrhosis, and hepatocellular carcinoma. And what we found is that the expression of ATG3 was upregulated when the condition of the liver was more damaged. And actually, there was a positive correlation between the expression of ATG3 and the steatosis rate and the NASH score. We repeated these results with different animal models of muscle D. Therefore, what we did next was to knock down ATG3 in the liver of animals fed with a choline-deficient plus high-fat diet. It's a common diet to produce liver damage, and in addition to that, they are obese, they are insulin resistant, so it's a metabolic syndrome. And what we found is that after some weeks on this diet, the mice with a downregulation of ATG3 were resistant to the development of steatosis, as shown here by the oil red staining, and this was associated with higher levels of some enzymes, all of them involved in fatty acid oxidation. So just to make a long story short, what we found is in the liver, through a pathway involving JANK1, SIRT1, one is going to improve mitochondrial function, and this will reduce the amount of lipids in the liver. So we repeated the same strategy doing proteomics in the liver of mice overexpressing and downregulating P63. Another gene, or another protein that called our attention was MAPS, mitochondrial antiviral signaling protein. So, as the name indicates, this protein is a mitochondrial protein involved in the immune response to viral infection. The metabolic role of this protein is controversial. There are some papers reporting opposite data. The expression of MAPS was proportional to the levels of P63. Again, we tested the potential clinical relevance working with Gemma Frubeck and Amaya here in Pamplona. They provided some liver samples from patients without muscle D and with muscle D. And what we found is that the RNA and protein levels of MAPS were clearly upregulated in patients with muscle D. And this expression was positively correlated with the NASH score. Again, we corroborated this clinical data in different animal models. All of them showed higher levels of MAPS. Therefore, we knocked down MAPS in the liver. We put these animals on a Western diet, again, to produce a metabolic syndrome. And we found that the knockdown of MAPS was protecting the animals against liver steatosis. Circulating levels of transaminases were lower. Even the expression of some fibrotic markers were also reduced, and the oil red staining also displayed a clear reduction in the amount of lipids. So, also to summarize this work, what we found is that the inhibition of MAPS was acting through a pathway involving ERK and inflammation, TNF-alpha and NF-kappa beta, and the reduction of inflammation was responsible for reduction in the lipid content. So, in addition to muscle D and the first stages of the disease, the liver steatosis, we also found that the inhibition of hepatic P63 was able to improve liver fibrosis. Here we did also some proteomics in different animal models, and the results are very diverse because we could find improvement in many different biological functions, most of them usual suspects, like improving mitochondrial OXPHOS, increasing the antioxidant activity, reducing the peroxisome function, all of them very well known to be related with the status of the liver. So, because P63 could, or the inhibition of P63 could improve liver fibrosis, what we wanted was to study much more specifically the role of this gene in different cell types. And the reason for this is that during the progression of the disease, when the patients reach fibrosis, liver fibrosis, it is when the pathologists are really worried about these patients, because it's when the clinical features are more serious. And of course, this fibrosis may develop to cirrhosis and hepatocellular carcinoma. But one critical step for the generation of liver fibrosis is to hepatic stellate cells. These are the main fibrogenic cells in the liver. So these cells are in the space of Disse between endothelial cells and the hepatocytes. Normally, they are quiescent, they are inactive. It is the main place where we store vitamin A. But upon liver injury, these cells get activated. And when they get activated, they change the cell identity. So they transform into myofibroblasts. And this means that they change the properties and they start to proliferate, they acquire contractility, and they also synthesize extracellular matrix. And this is why they produce the stiffness of the liver fibrosis. So what we did first was to check the expression of P63 in isolated hepatic stellate cells from different animal models with liver fibrosis. These are different diets or drug, very well-known models of liver fibrosis. In all of them, the expression of P63 was clearly upregulated. In collaboration with Javier and Paula at the Hospital de Valdecilla in Santander, they provided liver samples from patients at different stages of fibrosis. So patients that did not have fibrosis at all, and then patients that are in F1, F2, F3, F4. So different progression of the disease. And what we found is that the staining of P63 in these hepatic stellate cells was increased according to the progression of the disease. And actually, staining area with the fibrosis stage, NAS score, and circulating levels of transaminases. So we reproduce these results using cells from human donors that we activate with TGF-beta, transforming growth factor. Is a very common activator of these cells. And we did the same with a cell line called LX2. We treat these cells with LX2. We that the expression of the fibrotic markers, ACTA2 and different collagens, is upregulated. And in these conditions, the expression of P63 was also higher. So we generated animals lacking P63 only in hepatic stellate cells and put them in this choline-deficient plus high-fat diet. To generate these animals, we use the P63 LOXP mice together with the LRAT-CRE. LRAT is lecithin acetyl retinol transferase. It's a critical enzyme for the synthesis of vitamin A that is mainly located in hepatic stellate cells. So what we found is that after 20 weeks on this diet, the knockout animals were protected against liver fibrosis, as shown by the lower levels of transaminases and the lower expression of fibrotic markers. The same was found with different diets that we used. And if instead generating a knockout, we overexpress P63 in the liver, what we found is exactly the opposite. Independent of body weight, nothing happened with body weight. What we could found is that circulating levels of transaminases were significantly increased, and also the collagens as indicated by the Sirius red staining. Why is P63 producing this or accelerating this liver fibrosis? We did again proteomics in the liver of these animals, and we found that most of the proteins were involved with metabolism, and more particularly with fatty acid metabolism. And this was quite interesting for us because fatty acid synthesis is, of course, a critical factor for the development of liver fibrosis. And actually, there is one target. It is one enzyme, acetyl coenzyme A carboxylase, ACC, that is a critical step in the synthesis of triglycerides. And there are several clinical trials testing the potential beneficial effect of inhibitors of ACC to treat muscle D. So what we did was to focus exactly in this enzyme among the different proteins involved in the synthesis of fatty acids. And first step was to overexpress P63 in our hepatic stellate cells, check that indeed ACC1 expression and activity were upregulated. And we also corroborated that the overexpression of P63 was inducing the fibrotic markers. However, if we silence ACC1, we found that P63 was not able to activate these cells anymore, meaning that ACC1 looks indeed a critical factor for the effects of this P63. However, we performed different experiments and we were not able to find a direct correlation or a direct effect of P63 on ACC1. And after many options and failures, what we did was to identify one gene that could have potential to be the link between P63 and ACC1. And this gene was the epidermal growth factor receptor 2, HER2, which is very well known in the field of cancer. It is a marker of breast cancer. One subtype of those patients have a strong number of positive HER2 cells. And we found that P63 was indeed activating the promoter activity of HER2. So we check if this HER2 could be of functional relevance for the actions of P63. So again, what we did was to overexpress P63 in hepatic stellate cells. We checked that everything is working as expected. We have more ACC and more expression of the fibrotic markers. And when we silence HER2, we found that we could not see those actions of P63 anymore. So, what is the potential clinical relevance of HER2 in muscle D or fibrosis or cirrhosis? For that, we check a public data set of single-cell RNA sequencing comparing patients without any liver disease with patients with liver cirrhosis. And we found that in hepatic stellate cells, the expression of HER2 was clearly increased in patients with cirrhosis. And we could confirm this when we activate human primary hepatic stellate cells activated with TGF-beta that also show an increased expression of HER2. So basically, what Marcos and Eva proposed in this study is that the increase of P63 could induce the activation of the cells working through an HER2 and ACC1 pathway. This is going to produce changes in glycolysis and mitochondrial function, and this activation of the cells will accelerate liver fibrosis. So, if fatty acid synthesis is important for the activation of the cells, and we are not the only ones showing that, another option could be that fatty acid oxidation could be also relevant, because if these cells are activated and they need to cope with high energy requirements, there is a good way to energy from beta oxidation. So this is what we also tested, whether mitochondrial beta oxidation could contribute to the activation of hepatic stellate cells. And we focus on CPT1, carnitine palmitoyl transferase 1, is a critical enzyme located in the membrane of the mitochondria, responsible of the translocation of fatty acids into the mitochondria to be oxidized. And just to summarize, indeed, the overexpression was sufficient to activate them because they got more ATP, and therefore they were proliferating and they were inducing liver fibrosis. This is completely the opposite to what happens in hepatocytes, where the increase of fatty acid oxidation plays a beneficial role because it removes the fat out of hepatocytes. But P53 family members have nothing to do with this, so I will not explain this in more detail. In the second part of the talk, what I would like to show is to summarize some of our studies suggesting that P53 family members are also important for the modulation of insulin sensitivity and glucose homeostasis, not only for fatty acids and muscle D. And we started with a very simple experiment. We just put some animals on fasting during 6, 12, or 24 hours, and we check the RNA expression or the protein levels of P53. And as you can see, protein levels are rapidly increased upon fasting. Only 6 hours was sufficient to see an increase in protein levels, and this was maintained at further time points. But nothing happens, absolutely nothing, with the RNA expression of p53. So this was telling us that p53 somehow suffered a post-translational modification that was responsible for the stabilization of protein levels during fasting. And here we have some difficulties because one of the characteristics of p53 is that it can suffer so many different post-translational modifications. And these are just some of them: phosphorylation, ubiquitination, methylation, sumylation, acetylation, and much, much more. So it's a very complex. And we failed so many times trying some of those modifications, and none of them work, until we, CHUSA, finally found one of them that could explain these effects. And this was the so-called O-gluconacylation. This process consists in the addition of N-acetylglucosamine, so gluconac, in serine or threonine amino acids in the target protein. This addition is a process that occurs after different nutritional or stress conditions. It's very variable, very heterogeneous. But what CHUSA found is that indeed, under fasting, p53 was O-gluconacylated. And we check whether this post-translational modification could indeed play an important role in the metabolism of glucose. We started, again, with a very simple experiment. We put some hepatocytes in a medium without nutrients, and we find that p53 levels are upregulated, and this was associated with increased protein levels of PCK1. PCK1 is a rate-limiting step for the synthesis of glucose. And next, we put also some hepatocytes that do not express p53 in a medium without nutrients. If there is no p53, PCK1 was not upregulated. So what is the relationship between p53 and PCK1? So with the help of Samuel and Roman at the Thymus in Santiago, they performed some luciferase studies and demonstrated that p53 was indeed promoting or activating the activity of the transcriptional activity of PCK1. And we check the clinical or the potential functional relevance of the O-gluconacylation here. And for that, we generated a mutation in one serine that it's very well established that it is the amino acid where the gluconac is binding. And we compare the effects of the wild-type p53, here in orange, or the mutated p53. And we found that the mutation was not able to induce protein levels of PCK1 nor the activity of the enzyme, while indeed the wild-type was activating this PCK1 activity. And we could reproduce these results doing metabolomics and measuring some of the metabolites that are very important during gluconeogenesis. All of them were upregulated when wild-type p53 was overexpressed, but nothing happened when the mutated p53 was present. What is the situation in vivo? What we did was to generate mice lacking p53 in hepatocytes. And we performed a pyruvate tolerance test. So this test is widely used because pyruvate is used by the liver to produce glucose. So you inject pyruvate, you measure blood glucose levels at different time points, and what we found is that these conditional knockouts display lower levels of glucose during these tests, meaning that hepatic glucose production is likely reduced. And actually, if we put these animals under calorie restriction for 4 days, so the animals only eat 30% of the amount of food that they would like to, what we find is that these animals are not able to maintain blood glucose levels. Actually, we had to rescue many of them because they could die of hypoglycemia. And if we measure hepatic glucose, we'll find that indeed this hepatic glucose production was reduced in the conditional knockouts. What is the clinical situation for P53 and glucose metabolism? So again, together with Gemma and Amaya, we measure the expression of P53 in the liver of people with obesity but normoglycemic versus people with obesity and type 2 diabetes. And what we find is that P53 protein levels were indeed upregulated in the diabetic patients, also PCK1, and there is a clear correlation between these two proteins. So basically, what Chusa proposed in this study is that the O-gluconylation of P53 is activating PCK1, and this is activating the production of glucose in the liver. And we find that this modification of P53 is also important for the actions of gluconeogenic hormones like glucagon, adrenaline, glucocorticoids, and so on. So it plays a wild role in the regulation of glucose metabolism. Next, what is the potential role of P63 in glucose metabolism? So here, again, we start with a very simple experiment. We put the animals during 24 hours under fasting, and then we put the food back again in the cage. Here, the situation is completely different to P53 because RNA expression of P63 is downregulated. And when we put the food back to the animals, this expression is clearly upregulated. And these changes can occur very rapidly because only 6 hours of fasting is enough to see a reduction in the RNA levels of P63, and only 30 minutes of availability of food was sufficient to see an increase in the expression of the gene at transcription level. So here, what we did was to generate mice lacking P63 in hepatocytes and check how is the glucose metabolism of these guys. And again, when we did a pyruvate tolerance test, what we found was completely the opposite to P53. So here you have the knockouts, and you see that these animals have higher levels of glucose during these tests. And this was associated with higher levels of different gluconeogenic enzymes, including PCK1. So next, what we did was to rescue the expression of P63 in hepatocytes of the knockouts. So again, we do a pyruvate tolerance test. We see that in the conditional knockouts, the blood glucose levels are increased. But if we rescue P63 in these animals, the levels of glucose are completely normalized, are exactly the same as the controls. And this was, again, associated with the rescue of the protein levels of gluconeogenic enzymes in the liver. How is this interaction between P63, SIRT, and the gluconeogenic enzymes? So again, together with the help of Roman and Samuel, they did some luciferase assays, and they found that P63 was repressing the activity of SIRT1. SIRT1 is important for glucose production. And what they found is that if they knock down P63, the promoter activity of SIRT was upregulated. However, if they overexpress P63, the promoter activity of SIRT1 is reduced. And we could reproduce the relevance of these results by using our conditional knockouts of P63 in hepatocytes. They have higher levels of glucose during the pyruvate tolerance test. if in these mice we silence SIRT1 in the liver, we again rescue the phenotype. And this is because we can rescue the expression of the protein levels of these gluconogenic proteins. So, what is the potential clinical relevance of P63 in diabetes? Again, together with Amaya and Gemma, we checked the expression of P63 in normoglycemic versus type 2 diabetic patients, and we find that P63 expression was reduced in the liver of diabetic patients, and it is negatively associated with fasting glucose and the HOMA index. So, what Susan and Eva suggest here is that P63 was repressing SIRT1, and therefore SIRT1 was blocking the activity of gluconogenic enzymes and reducing the levels of glucose. And our search on new genes related with P53 family members is still ongoing. And actually, this is one of the genes that we have identified by doing chip sequencing together with Ashwin and Christina, also in Santiago. They found that there is a strong binding of P53 with one receptor, especially in conditions of calorie restriction and fasting. And this receptor is GFRA alpha. I will go to it right now. The thing is that these results with the chip sequencing were quite consistent with public data sets analyzing the expression of genes comparing mice fasted versus fed ad libitum. And the expression of this gene was also upregulated in fasting. So this gene is a receptor, glial cell line derived neurotrophic factor receptor, the receptor of GDNF that is widely known in the field of neuroscience because GDNF is very important for the survival of neurons, in particular dopamine neurons. And it is also known that GDNF can modulate systemic metabolism through central actions. But here we were quite surprised to see, to identify this receptor in the liver, apparently with nothing to do with the central nervous system. So this receptor is working through the red signaling pathway. It's a member of a family of different receptors, all of them in the plasma membrane. But this one is the most specific for GDNF. So first thing that we did was to check the expression of the gene and protein levels in the liver of animals fasted and refed, and we checked the expected recovery when the food is back to the animals. Also, the expression of the gene is increased upon calorie restriction. And if we knock down this receptor only in the liver, what we find is that if we do a pyruvate tolerance test, the glucose levels are reduced, meaning probably reduced hepatic glucose production. If we put these animals under fasting during 24 hours, they are not able to maintain blood glucose levels. And actually, they show a lower expression in the PCK1, in the critical enzyme for gluconeogenesis. And if we put these animals on a high-fat diet, we find similar results. These animals have lower levels of glucose during a pyruvate tolerance test. They have more insulin sensitivity, and this is consistent with the trigger in the phosphorylated levels of AKT at hepatic level. And just to finalize, at clinical level, what we found is that the receptor was indeed upregulated in patients with type 2 diabetes, and there was a positive correlation with fasting glucose and the OMA index. So, in summary, what I have been trying is to convince you that P53 and P63 play an important role at physiological and pathophysiological level in the modulation of glucose and fatty acid metabolism. Do we believe that P53 or P63 can be drug targets to treat muscle D or type 2 diabetes? Honestly, I don't think so. But we can learn, because they are promiscuous transcription factors, and we can identify novel targets that may have some potential. And finally, I just want to thank all the team doing the experiments. They are the ones doing the hard work in the lab, and funding bodies and collaborators, and thank you for the attention.
Thank you, Ruben, for this very excellent presentation. And we have time now for questions. Somebody in the audience have some question?
Thank you very much, Ruben. It was a really nice talk, really clear. Okay, and I have a question regarding these stellated hepatic cells. Okay, with this activation of P63, that is increasing the promoter of HER2. Okay, then we got fibrosis. Have you tried to block this HER2 receptor in this with trastuzumab Herceptin, that are very known drugs for breast cancer subtype?
Yeah, good point. Yes, we are doing that. With trastuzumab, it works quite well. We are able to ameliorate liver fibrosis, but our aim is to be even more specific and to carry the inhibitor, antibody or shRNA, specifically to hepatic stellate cells. And this is what we are trying to do with one expert, in Maria Jose Alonso in Santiago, that she's an expert with nanoparticles, and this is what we are testing, whether targeting specifically, inhibiting specifically HER2 in these cells could be beneficial. We have some preliminary results, but we are in these patent things, and they tell me that I cannot talk about this too much. Sorry.
Thank you.
Time to eat.
I have one question, Ruben. Since these P53 and P63 are very relevant for metabolism, and you can show us the phenotype of the mice, have you tried to do some aging studies to see how is the aging process in these animals, if it's accelerated or is prevented?
Yeah, that's a very good question. So the problem here is that if we maintain these animals lacking P53 or P63 for a long time, they will develop tumors. Okay. So we will not be able to distinguish which part of the metabolism they play independent or dependent of the cancer aspect. So we did all the experiments at short time points just precisely to avoid the development of tumors. We tried to do what you are asking, but with P53. But after, if I remember properly, 5 months, they started to develop some tumors in the liver and sometimes outside the liver as well.
Okay. Some more questions?
Despite being liver-specific knockouts?
Yes. Despite being liver-specific knockouts, sometimes some of them developed tumors outside the liver. We did not study that in detail because we are not experts, but yes.
Both, even with the P and the...
With the P63, we did not wait so long. We tried with P53 because the reviewers asked for that study. We wait, but it was useless to answer the questions that we wanted to answer because the metabolism could be affected by many different aspects.
Okay, then thank you very much. Then we now close the session for the morning, and we have time for lunch now. We will continue with the sessions in the afternoon at 3:30 p.m. Then enjoy the lunch and see you at that time. And thank you very, very much, Ruben, for this excellent presentation.
La innovación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico, estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IS Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster, marketing a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación a consumidor. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
La innovación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico. Estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IS Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación a consumidor. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
innovación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico, estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IES Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación a consumidor. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
innovación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico, estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IES Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación a consumidor. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
innovación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico, estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IES Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación a consumidores. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
formación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico, estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IES Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación a consumidores. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
innovación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico. Estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IS Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación a consumidor. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
innovación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico, estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IES Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación a consumidor. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
innovación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico, estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IES Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación al consumidor. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
innovación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico, estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IES Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación a consumidor. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
innovación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico, estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IS Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación a consumidor. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
innovación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico, estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IS Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación a consumidor. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
innovación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico, estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IS Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación a consumidor. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
innovación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico, estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IES Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación a consumidor. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
innovación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico, estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IS Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación a consumidor. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
innovación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico, estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IS Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación a consumidor. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
innovación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico. Estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IS Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación a consumidores. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
innovación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico, estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IS Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación a consumidor. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
formación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico, estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IES Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación a consumidor. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
innovación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico, estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IS Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación a consumidores. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
innovación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico, estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IS Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación a consumidor. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
innovación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico, estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IS Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación a consumidores. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
innovación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico, estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IES Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación al consumidor. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
innovación es esencial en cualquier empresa, especialmente en el sector alimentario. Necesitamos personas creativas y con pensamiento crítico, estudiantes capaces de liderar iniciativas sostenibles.
Es un programa que abre puertas a un mercado en constante evolución.
Con este máster serán capaces de integrarse en equipos innovadores de la industria alimentaria.
Aprenderás con el método del caso, enfrentándote a situaciones reales del sector desde el primer día.
Cuando un alumno viene con una visión más global de lo que es una empresa, es mucho más fácil.
El programa incluye prácticas de 3 a 6 meses.
Está diseñado junto con el Centro de Investigación en Nutrición de la Universidad de Navarra. De hecho, muchos de nuestros profesores vienen de allí y también del IS Business School.
Yo mismo cursé mis estudios en la Universidad de Navarra y comencé a trabajar en Nestlé. Realmente hay un factor diferencial en la formación de los alumnos que hace que a la hora de incorporarse al mundo laboral aporten un plus que se valora muchísimo en una empresa como Nestlé.
Lo animaría porque va a tener muchas más oportunidades de encontrar trabajo en el mercado.
¿A qué podría dedicarse en una empresa como Nestlé un alumno que curse este máster? Marketing, a ventas, nutrición propiamente dicha ligada al etiquetado de los productos, a la comunicación a consumidor. Las oportunidades que ofrece para mí son amplísimas.
Impulsa tu carrera en la industria de la alimentación. Únete a nosotros.
El mejor sitio.
Hello.
Good afternoon, good evening to everybody. We continue with our workshop, and the next presenter, the next invited speaker is Dr. Jesmond Daly. Yes, Jesmond Daly is a leading figure in the field of molecular pharmacology, internationally recognized for his pioneering research on lipid mediators and their role in inflammation resolution and tissue regeneration. He currently serves as a professor at Barts and the London School of Medicine and Dentistry, and as the director of the Lipid Mediator Unit at the William Harvey Research Institute, and is the chair of a recently funded cost action focused on resolution biology. Congratulations for the cost action.
Thank you.
Following his doctoral studies, he joined the laboratory of Professor Charles Sherrand at Harvard Medical School and the Brigham and Women's Hospital. There, he held a faculty position and served as co-director of the two NIH-funded lipid mediator profiling course. further deepening his expertise in the emerging field of pro-resolutive mediators. Jesmond's research centers on the molecular pathways through which specialized pro-resolving mediators, SPMs, restore tissue homeostasis, regulate inflammation, and promote repair. His work bridges fundamental discovery with translational impact, reflected in his portfolio of patents, several of which are currently licensed for clinical development. He has authored over 180 peer-reviewed publications, holds a H index of 80, and his work has been cited more than 19,500 times. His contributions to science have earned him numerous accolades, including the Eicosanoid Research Foundation Young Investigator Award and fellowship to both the Royal Society of Biology and the British Pharmacological Society. We welcome Jesmond. Thank you so much.
Thank you for the kind introduction. I'd like to take the opportunity for inviting me here. It's always good as a Mediterranean to be in the Mediterranean climate and to leave the Northern European wetter, darker climate behind for a few days at least, and also for the wonderful food. So, having said that, this is our campus, and this is on a nice sunny day. So they do occur, those nice sunny days in the UK. And one may disagree that climate change exists. We know a few people who do, but we're getting a few more of these sunny days in the UK, at least this year. So what I'll be sharing with you today is not my work alone. It's a work of many colleagues, including, for example, Maria Jesus here, and discussions with many colleagues over the years. So I'd like to acknowledge this here, as well as the support of the funders. And we do have fun on occasions, at least. And this is the people, many of the people who have contributed to some of the work that I'll be sharing with you today. So you're all familiar with this very simplistic view. This is our view of the world of inflammation. So you have a challenge that leads to the initiation phase of the inflammatory response. The inflammatory response gets elaborated, and eventually it resolves, at least in an ideal case. And this happens tens of hundreds of times a day in the host. And in most cases, we don't even notice about this because it is part of our defense mechanism, it's highly tuned, and so on. And we know that during this initiation phase, many soluble mediators are produced that largely fall into these three categories. And they're important in directing the host immune response to elaborate, to recruit cells to the site of inflammation, to contribute to the clearance of pathogens, and so on. And for many years, the paradigm was that disease occurred through the overproduction of these molecules, and that leads to chronic inflammation that we know influences many of the diseases that influence our society. And so the therapeutic paradigm has been, let's block aspects of the biology of these molecules, depending on which is our favorite molecule, which is the molecule that we think is important in a particular disease. And the pharma has refined approaches to virtually annihilate the biology of specific molecules. And we know that that is effective, at least in the acute phase. However, in many instances, those approaches don't lead to complete resolution of inflammation, to restoration of organ and tissue function. And many chronic inflammatory diseases require lifelong treatments with associated side effects. And one of the key ones is immune suppression. Now, we've been interested in trying to understand whether there is more to just simply overproduction of these molecules. And indeed, if you look at the simplistic view, there's a hole here. The second part of the inflammatory response, the resolution phase, which for many years was thought to simply occur through the passive dilution of these pro-inflammatory molecules. However, Charlie Serhan and colleagues in Boston early in the 2000s questioned whether this was really passive or whether there was an active process that occurred here. And what they found was that juxtaposed to what is produced during the acute inflammation from a small molecule perspective, where we know that, for example, arachidonic acid, which is an 6-polyunsaturated fatty acid, is converted to molecules that elaborate the inflammatory response. There are mediators that are produced from omega-6 primarily fatty acids, including eicosapentaenoic acid, docosapentaenoic acid, and docosahexaenoic acid, that have defined structures, defined biosynthetic pathways that basically contribute to control this inflammatory response. So they're produced during active self-limited inflammation to reprogram immune cells to prevent the overshooting of the inflammatory response. And we now know through work of many groups around the world that these molecules are produced in humans as well as in other experimental settings at biologically relevant concentrations. And they're produced, interestingly, not only in settings where we think that there is inflammation, so for example, adipose tissue or in synovial fluids from patients with RA, but also in settings like, for example, in milk, where we don't think that there is pathogenic inflammation. Rather, there is a protective response. And milk is critical, as you all know, for infant development. So there's got to be a role for these molecules beyond simply limiting inflammation, which is a question that we're very interested in addressing. From a pharmacological perspective, and this is another point that is critical, much of what we know about the biology of these molecules is from a pharmacological perspective, where we're adding these mediators. These molecules display protective activities in a range of chronic inflammatory settings, at least in experimental settings. So going from COPD, for example, to neuroinflammatory disorders, we heard some great talks this morning about neuroinflammatory disorders, to adipose inflammation, again here. But interestingly, and juxtaposed to what we know about anti-inflammatories, these mediators are also protective in models of viral and bacterial infections. So they do not lead to immune suppression, as we know many of the anti-inflammatories do. They rather promote the clearance of bacteria and facilitate host immune responses through, for example, antibody class switching to viral infections. And they do so through the activation of cognate G protein coupled receptors. This is a list of the GPCRs that we know are important in directing the biology of these molecules. And recently, this paradigm has been challenged, although unfortunately the evidence is very weak to challenge this paradigm that was provided. Because in many cases, for these, the role of these GPCRs has been shown through, for example, knockout and knock-in approaches, whereas the challenging paradigm has been only been demonstrated through recombinant receptor systems. So I think potentially there is an additional role for these mediators to also contribute to the regulation of other receptors through orthostellic regulation of other G protein coupled receptors. However, the evidence so far is weak. What we really think is primarily involved is the activation of these G protein coupled receptors. And so the working model so far, as we think that these mediators are acting, is through, when they engage their cognate receptors, they lead to an epigenetic change in the target cell. This epigenetic change basically changes the way that particular cell perceives its environment. So now, when you get an inflammatory signal, you're not hyperactivating the cell, but rather you have a protective response that leads to a lower production of pro-inflammatory molecules. So you break that feed-forward mechanism that's very critical in many chronic inflammatory conditions. And actually, and I'll share some of the data, what you also tend to get is that you switch the phenotype of the cell to a reparative and a protective phenotype. So, as in any field, there are challenges and open questions, and these are some of the questions that we've been very interested in addressing. So, for example, the first one is the identification of these molecules in biological systems and in tissues. Another one is linked to the concentrations, linking the concentrations that we can measure to the bioaction of these molecules in tissues. A third one is the role of these receptors in human, some of the SPM receptors in human and the link with disease, and then the role in physiology. So we know a lot about the role during active inflammation, but there's clearly a role in maintenance of physiology and in resilience. The first one has actually recently been, strong steps towards addressing. The first one have been recently achieved through an international effort led by the International Lipidomics Society, and indeed some of the groups that have struggled to measure these molecules are now reporting on them. And I will share some of the work that we've been doing around the next two questions. So some years ago, Charlie Serhan's group described the formation of what was at the time a new family of mediators termed the meresin as macrophage mediators in resolving inflammation. And this family is produced via the conversion of the fatty acid docosahexaenoic acid by specific enzymes, lipoxygenase enzymes, through the steps that we've now determined using total organic synthetic approaches that lead to this molecule, meresin 1, as the first member of the family. And meresin 1 has been found in many organ systems and tissues, both in acute and chronic inflammatory settings in both mice and humans. And its pharmacology has been demonstrated to be important in studies ranging from hypertension to chronic rhinosinusitis, for example, and cognitive decline. And Charlie's group, a few years later, demonstrated that this mediator activates the GPCR LGR6, which is a class A GPCR. And they used a whole range of assays to demonstrate the role of this GPCR in mediating the biology, at least in experimental settings, of meresin 1. And again, a number of groups have shown a role of LGR6 in mediating the biology of meresin 1 in models such as, for example, lung injury and viral infections, adipose tissue inflammation, work from Maria Jesus' lab, subarachnoid hemorrhage, bacterial infections, and so on. Now, this is all in experimental settings, but it's very difficult to do a knockout in humans, or at least that's what we thought for a while. But we were lucky that locally we are linked with a cohort of patients called the Genes and Health cohort, which has collected samples from about 60,000 British Bangladeshi and British Pakistani. And for these volunteers, we have linked health data with genetic data. And so we can screen these patients to identify single nucleotide polymorphisms in genes of interest, and then link those to the health records. And the other advantage that we have is that we can recall these patients to obtain biological samples. And this is work that was led by Esteban in our group. And what we found was that within this cohort of patients, there was a very small group of patients that had a single nucleotide polymorphism in the LGR6 receptor at the genome level. And this was predicted to lead to a frame shift mutation, to a loss of function mutation. Interestingly, because obviously biology can't be always very simple, LGR6 has 3 biological isoforms, and at least the prediction was that this loss of function mutation, this frame shift mutation, would only affect 1 of the 3 isoforms. So this is where the loss of function mutation is predicted to occur. So you get a shift in the amino acid sequence of this protein that leads to an early stop codon. And this happens in the N-terminal portion of the protein. So basically, you have no protein. So we were expecting a human knockout of LGR6. So we got very excited here. So what we did was we obtained human samples from patients with this LGR6 variant, or matched as much as we could for the usual variables, sex, age, inflammatory conditions, and so on. And then evaluated the biology of the cells, primarily focusing on phagocytes, because that's our sort of cell family of interest, as well as some of the other biological activities. So what we did was, first of all, we looked to evaluate the expression of LGR6. And we started off with neutrophils. We were very pleased. And when we look at the entire granulocyte population, or the neutrophil specific, you see there's virtually no LGR6 expression in these cells. We then turned to monocytes. Looking at the whole monocyte population, we started to see a bit more of a signal here. And as you can see, there's a subset-dependent loss of LGR6 in monocytes. And the opposite was true in macrophages. So macrophages had higher levels of LGR6 than monocytes did, than neutrophils did. So this started to suggest to us that different immune cells use different isoforms. Unfortunately, the antibody that we had, all the commercially available antibodies, do not distinguish between the 3 isoforms. So we couldn't quite tell whether this hypothesis was true, and actually this is something we're currently pursuing. But this suggests, at least from the overall expression, that different immune cells require or use different isoforms of the receptor. So we focused on neutrophil biology, and we started looking at the phenotype of the neutrophil through what they're producing. So here we're looking at lipid mediators. We're obviously very interested in lipid mediators. And what we found was that when we look at the lipid mediator profile, which was previously shown in macrophages, for example, to reflect the phenotype, neutrophils from the variant population displayed a more inflammatory phenotype, especially through the higher production of leukotriene B4, which is a potent chemoattractant, and lower levels of pro-resolving mediators. And this was also true when we evaluated monocytes. We also found that these cells displayed defective biological responses. So here we're looking at the ability of neutrophils and monocytes to uptake bacteria, and the neutrophils and the monocytes from these patients displayed lower ability to uptake bacteria. And when we evaluated the responses to merism-1 in macrophages, we found that, in neutrophils and monocytes, we found that there was a reduction in their ability to respond to merism-1. So this confirms, first of all, that merism-1 is a ligand for LGR6 because we lose the response. And secondly, that the endogenous loss of LGR6 also feeds forward into a defective phenotype in these cells. So then, in collaboration with one of our colleagues who is an expert in genetics, Patricia Monroe, we wanted to see, okay, we've shown some phenotype at an in vitro level. What does this mean at a population level? So what does the loss of LGR6, at least in a portion of the cells, do to the entire population, knowing that merism-1 has these pro-resolving activities? So we used the UK Biobank, which is a UK-wide cohort of patients like the ESGL, where genetic data is linked with health data. As it happened, the region where our SNP was not sequenced, but there was another region that was on the same part of the chromosome that was in linkage disequilibrium with our SNP, and it was virtually an identical linkage disequilibrium. So this basically was used as a proxy. And what we found was that when we evaluated the incidence of diseases with these SNPs, we found there was a significant signal at a genome-wide significance for viral infections. And as it happened, when we went back to the patients and obtained cells and stimulated them with TLR ligands that are linked with viral responses, we found defective viral responses. So for example, here, these are the neutrophils. There's a defective production of IL-12, interferon gamma, and interferon beta in response to TLR7 and 8. As natural killer cells are the primary cells that are linked with antiviral responses, we went back and evaluated viral responses in NK cells and also LGR6 expression. And as it happens, LGR6 is also lost in NK cells from these patients. And as you can see here, there's also a defect in the ability of these NK cells to respond to TLR ligands, so TLR4, TLR9 primarily, suggesting that indeed there is an immune response, a defective immune response when LGR6 is lost in these patients. And in support of this, it's always good when someone else demonstrates similar findings. Bruce Levy's group in Boston demonstrated that in experimental settings, the MAR1 LGR6 was important in responses to RSV infections in the lung. So this suggests from an experimental settings that the human responses, the genetic data, that was indeed a potential link here. So this basically suggests, so what I've shown you so far is that we've identified this particular SNP that leads to a loss of expression of LGR6 in phagocytes, primarily neutrophil monocytes, and also in NK cells. This is linked with dysregulated immune responses, and at a population level, it exposes the host to viral infections, but potentially other chronic inflammatory conditions. There was a few others that didn't quite make the genome-wide significance, and potentially they're just simply because it is a low abundance SNP, so if we increase the ends, probably these signals would be significant. So another portion of the merism family that we recently identified are these merism conjugates in tissue regeneration. And what we observed was that These mediators, which are peptides, so this is glutathione, cysteine, glycinyl, and glycinyl, conjugated to the DHA backbone, are produced in inflammatory settings, but they're also evolutionary linked. So they're produced in planaria, for example, they're produced in mice, but they're also present in humans. And they display protective responses, regulating host immunity, inflammation, but also promote tissue repair and regeneration. So we wanted to see whether this, first of all, is also linked in humans with potential, so loss of these molecules is linked with disease. And we obtained peripheral blood from DMARD naive. So these are patients that present to the rheumatology clinic before diagnosis and before receiving their first round of treatment. And we evaluated the levels of these mediators in peripheral blood. And what we found that one of these mediators, MCTR3, the peripheral blood levels were correlated with typical markers of inflammation. And loss of MCTR3 was also linked with early signs of erosions in patients with joint inflammation, suggesting that there's a potential link with a defect in the formation of these molecules and joint pathology, in particular joint damage. So we wanted to test this a little bit more in experimental settings. Here what we did was we treated mice at the peak of inflammation with MCTR3. The 3 arrows showed the 3 doses of MCTR3, and then evaluated disease severity as we went along. And what we observed was that there was a significant protection in disease severity when we given MCTR3, and this was through the reprogramming of at least in part, of macrophages in the joint. And here we also observed that there was an improvement in joint protection. So not only do we limit inflammation, do we shift macrophage phenotype, but we also promote cartilage repair within the damaged joint. And this was also true for the bone. So the red regions here are regions where you get bone remodeling. It's called bone callus. And what we observed was that when we treat mice with MCTR3, we reduce this bone remodeling and we improve bone strength. And this was measured here, as well as a reduction in peripheral blood levels of the bone degradation product, CTX1. Now, what we observed was that part of the mechanism was linked through the reprogramming of monocytes in the periphery. So if we obtain arthritic monocytes and treat them with MCTR3 and inject them back to an arthritic mouse, what we could observe, and this was the treatment ex vivo was only for 90 minutes. So what we observed was that those monocytes that were exposed to MCTR3 for that short period of time displayed an enhanced protective activity compared to control monocytes, suggesting that this paradigm where you have ligand receptor engagement and then engagement of downstream signaling was really active here. And we could observe that this replicated some of the joint protective activities where we can see increase in bone and cartilage, collagen 2 levels, which is linked with cartilage protection. Now, because of this idea that we're potentially engaging signaling mechanisms that are long-lasting, we questioned whether there was a role for epigenetic regulation. And so what we did was we incubated the monocyte, we pre-incubated monocytes with this DNA methyltransferase inhibitor prior to exposing them to MCTR3. And if you inhibit these DNA methyltransferases, what you get is a complete reversal. So the white circles and the triangles are overlapping here. Complete reversal of the biology of MCTR3. When we're looking both at the number of swollen joints as well as the degree of swelling, suggesting that indeed epigenetic mechanisms are important in the biology of MCTR3 at a joint level. This was confirmed when we did single-cell RNA sequencing, focusing on macrophages in the joint, where we observed that there was a significant shift in the transcriptome 10 days after transfer of the monocytes. So we collected the macrophages from the joint, and we could see that the transcriptome of these macrophages persisted all the way through to that time point. And indeed, we observed that there was protective pathways upregulated, such as, for example, arginine and proline metabolism. And So given the role of apoptotic cell uptake and clearance in tissue repair and regeneration, we questioned whether this was at least part of the mechanism that we're regulating. So this is work that was led by Duco Kernis. And what Duco did was he posed a very simple question, which was, what happens during the process of apoptotic cell clearance during macrophages, and are these pathways upregulated during this process? And what we observed was that there was a selective upregulation of ALOX12, which is the initiating enzyme in the MCTR biosynthetic pathway. And this was specifically upregulated in those macrophages that had taken up an apoptotic cell. So those macrophages that had a bound apoptotic cell, but it was still on the outside, we didn't see a change. And those macrophages that did not even engage an apoptotic cell were also not engaged. So this suggested that there was crosstalk between the uptake of the apoptotic cell and MCTR biosynthesis. So in order to try and link the two, what Duco did was a very simple experiment. He transfected macrophages with a GFP-labeled ALOX12 and evaluated the uptake of apoptotic cells in these cells. And what you can see is those macrophages that were GFP positive, 8 more. So that suggests that there's an autocrine loop here. Interestingly, macrophages that were GFP negative, but within 50 microns, so adjacent to this GFP positive cell, 8 just as good, potentially even better than the cells that were ALOX12 positive. Those cells that were further away just didn't notice. So this led us to the paradigm that the macrophage that's producing MCTRs can, apart from regulating its own biology, can regulate this biology of cells around it, but this effect doesn't dissipate further. So basically, you have a gradient away from a cell that makes the molecules. And this is, frankly, not too surprising, considering that most of these soluble molecules travel through gradients. The effect goes through gradient. However, it does lead to it does raise the point that the way we currently measure and try and link concentration to function is flawed, simply because what we currently do is we get a piece of tissue, we homogenize the tissue, and then we measure the concentration at a whole tissue level. Many of the tissues, and I'll show some of the data in a couple of slides, only have a subset of cells that are able to produce these molecules. So if, for the sake of argument, we're measuring picomolar concentrations, likely at the site of formation, we're log orders higher in terms of concentration. So this leads to the point that we need to develop better methodologies to be able to link formation to function. And also, as we're thinking about biology, we've got to include the spatial aspect, as we're doing for many other technologies, into the equation. So as you'll see here, this is a mouse model of apoptotic cell clearance, where we administer dexamethasone and evaluate apoptotic cell clearance in the thymus. And what you can see here is that in the cells that are adjacent, so these are the ALOX12-15 positive cells, these are the adjacent cells, you get apoptotic cell clearance. Those cells that are further away, you don't get apoptotic cell clearance. And this is specific to the enzyme, because if we knock out the enzyme in macrophages specifically, you lose the effect completely. So this is not simply a dish phenomenon. This also happens in tissues, in living organisms. And when we evaluated the formation of these mediators in tissue culture during apoptotic cell clearance in macrophages, we observed that there was time-dependent increase in MCTR formation, and the concentrations that we measured at a whole sort of tissue culture level were within bioactive concentration ranges for the molecules. But this obviates for the fact that only a subset of these cells, I'm not sure what's happening here, produce the mediators, and so likely the concentrations are much higher than what we're measuring over here. So this is also true in vivo. If we go back to the dexamethasone model, where you have thymocyte apoptosis, you see a significant increase in MCTR formation that is lost in the global knockout for the ALOX12-15 or in the macrophage-specific knockout. So this is macrophages that are produced MCTRs as part of the feed-forward loop to facilitate apoptotic cell clearance. And it's also true if we look in a tissue culture level, where we see that MCTRs are able to feed forward the clearance of apoptotic cells. So this is a high burden efferocytosis model where we're looking at subsequent uptake, so not the first cell, but the second and third cell. And we can see that the knockout mice display a defect, but we can rescue it with the exogenous MCTR formation. And this is the same experiment in vivo. So how does this happen? We got together with a colleague of ours, Pedro Cutillas, and we looked at the signaling pathways. And what we observed was that there was an important, at the phosphoproteomic level, an important the pathways that came up, some of them were glycolysis and GTPase regulation, and in particular RAC1 regulation. And RAC1 is critical for this on-off mechanism that's linked with the actin assembly and disassembly that's important for the apoptotic cell engulfment. So we did a pharmacological experiment evaluating whether if we use a RAC1 inhibitor or a ROE inhibitor, which could potentially also interact, we see a reversal of the ability of MCTRs to uptake apoptotic, to induce the uptake of apoptotic cells, but not with ROE. And this was also true in one of our favorite experimental models, which is a planaria model. These are flatworms that have retained the ability to regenerate. And when you injure planaria, so if you surgically remove, say, the head, they'll grow a head in 7 days. And a lot of the signaling pathways have been described, and they are parallel to what we have in humans. So what we observed here and there is, upon injury, there is, at the site of damage, there is a high apoptotic cell burden. So they require the clearance of apoptotic cells. And indeed, we can accelerate this clearance if we get MCTRs, but if we inhibit RAC1, we reverse this ability to clear apoptotic cells and to regenerate. And so, because metabolism is also important in this process, we question whether metabolic pathways, and in particular the glycolytic pathway, is regulated. And what we found was that there's an upregulation and phosphorylation of several key steps within the glycolytic pathway. And so when we measured the levels of intermediates within this glycolytic pathway, we observed that there was a selective upregulation of glycolysis, but we did not affect the TCA cycle. And this made complete sense to us, simply because what we think is happening here is that we're shifting the energy production of the macrophage to glycolysis to release the mitochondria to be able to digest the metabolic load that comes from the apoptotic cell. And that facilitates subsequent round of apoptotic cell clearance. And indeed, we observed that we improve glycolytic capacity when we use the seahorse system with all 3 MCTRs, and this was linked with an improvement in glycolytic reserve. As well as if we look at glucose uptake, we see an increase in glucose uptake that was inhibited using the RAC1 inhibitor, and we later found out that this was also inhibited with aldolase, with an aldolase inhibitor, which is one of the key steps in glucose uptake within the cell. And if we evaluate the effect of this in vivo, there's a panel missing. You're gonna believe me here. If we evaluate the effect of this in vivo, we see that if we inhibit metabolism in the planaria, you prevent the tissue regenerative activities of MCTRs in planaria. So this is our working model where MCTRs promote the uptake. So the initial apoptotic cell engagement with the macrophage, and usually it engages with cells that already express ALOX12 at a lower level, promotes the formation of MCTRs, and this acts as a feed-forward loop, whereby MCTRs in an autocrine manner upregulate ALOX12, and ALOX12 is upregulated through the release of DNA from these apoptotic cells that is sensed by TLR9 and through an ALA hydrocarbon receptor mechanism, which I don't have time to share with you, but it's all published. And the MCTRs then regulate the cells around this initially efferocytic cell to propagate the effect to other cells around them. And the last part is we've been interested in the role of identifying common mechanisms in chronic inflammatory diseases. Currently, chronic inflammatory diseases, if you have a joint inflammation, you have arthritis, you go to a rheumatologist. If you have cardiovascular disease, you go to another physician. And many of these conditions are linked. There are potentially linked mechanisms. And so in RA, patients with RA have a higher incidence of cardiovascular disease, and this is actually one of the key factors that leads to the higher incidence of death in patients with RA. So we recently observed that in experimental settings, through leukocyte endothelial interactions, you have the formation of a family of mediators called 13-series resolvins, and these mediators, their formation is upregulated by atorvastatin through the nitrosylation of cyclooxygenase 2. And in trials with atorvastatin, in patients with RA, demonstrated an improved outcomes in both joint inflammation and a reduction in cardiovascular mortality. So we questioned whether these mediators had an important role in regulating both joint and vascular inflammation. So this is work that was primarily done by Mary Walker and Roberta de Matteis. And basically what we did was we set up an experimental model first where we treated mice with Western diet for several weeks. And towards the end, then we challenged with KBN serum that leads to joint inflammation that's primarily driven by the innate immune system. And what we found here was that there was an increase in both joint damage as well as vascular inflammation. And this was negatively correlated with the formation of this mediator RVT4. And when we administered RVT4 to the mouse model, we saw that we could protect the vascular inflammation, so joint lesion area. We reprogrammed and upregulated factors that are linked with an increased protective macrophage phenotype in the vascular, such as, for example, MERTK, and reduced lipid load in the macrophages from the vascular tissue. And this aspect of reducing lipid load caught our attention because this is part of what is thought to lead to dysregulated macrophage responses. And so we sought to characterize how these macrophages, how RVT4 regulates lipid load in macrophages. And we found there was a dose-dependent there's a dose-dependent reprogramming of macrophages and their ability to clear apoptotic cells here. These are lipid-loaded macrophages, and it improves chemotaxis towards classic chemotactic signals, two mechanisms that are known to be dysregulated in vascular lesions and vascular macrophages. We then looked to see whether RVT4 promotes cholesterol efflux, and indeed we found that it did, at least in human monocyte-derived macrophages, as well as in macrophages from ApoE knockout mice, both in vivo and in vitro, where we see a reduction in overall lipid load in the macrophages that was linked with an increase in free cholesterol outside the cell. We then looked to evaluate this mediator was regulating lipid load. We went to evaluate ABCA1 and ABCG1 as two of the classic channels that promote cholesterol efflux, but we observed that if we knock down these two channels, we did not see a reversal of cholesterol efflux in these macrophages. So another channel that has been linked with cholesterol efflux is SRB1. And when we knocked out SRB1, we saw that we got a complete reversal of the ability of macrophage RVT4 to promote cholesterol efflux. So this is lipid load, and this is the cholesterol efflux, which is completely abrogated in SRB1 knockout mouse. We then evaluated the kinetics and found that this SRB1 translocation was very rapid. Within 15 minutes, we get maximal translocation to the plasma membrane. And if we inhibit cytokalasin D, if we use cytokalasin D, we inhibit this translocation, suggesting that this was an active process. And when we evaluated the mechanism in vivo, so here what we did was we repeated the Western diet experiments, but this time in ApoE mice, so a more severe phenotype, and then generated arthritic mice using the KPN serum, and treated for 2 weeks on alternative days with the mediator. And what we observed is that in control mice, we see a significant reduction in both lipid load as well as in lesion size in the mice. But if we knock out SRB1 specifically in the macrophages, or knock it down specifically in the macrophages, we reverse this protective phenotype. both at a gross tissue level as well as when we're looking specifically in the top part of the aorta for necrotic cores. So this is our mechanism. We observe that through this reverse cholesterol transport mechanism that also involves the activation of N-cholesterol esterase hydrolase, we reduce lipid droplet size in the macrophage. There is a release of cholesterol that gets loaded onto HDL. And this reprograms the macrophage to become less inflammatory and more protective. And this facilitates the stabilization of plaques and reduces lesion size and vascular inflammation. And this mechanism, and I didn't have time to share with you, is also protective at the joint level. So we see a reduction in joint disease activity in these animals too. And so this is the overall summary where we have now observed that there's evidence for the role of SPM receptors, in particular LGR6, in humans, and this is linked with incidence of viral infections. MCTRs are locally produced and regulate the clearance of apoptotic cells and facilitate tissue repair, and RVT4 reprograms macrophages to respond to facilitate both lipid efflux as well as restore the macrophage function, and this leads to reduction in joint inflammation and vascular inflammation. And I'd like to leave you with one little pitch here. So as was mentioned earlier, we've just recently received funding from COST to set up a new EU biology, resolution biology network. We have a decent representation, I would say, so far, but we're looking for more enthusiastic colleagues to join and to contribute to our understanding. So if you're interested, please look this up on the COST website or get in touch, and I'll be very happy to direct you. And with that, I'd like to thank you for your attention, and I'll be happy to take any questions.
Thank you very much, Jesmond. It's time for we have time for a few questions.
Very nice presentation, Jesmond. I have one question or one curiosity. In those people that are carrying the mutation in the LGR6 receptor, that they have less effect of myrosin 1 and more susceptibility to viral infection, do you have some phenotypic data, like the metabolic data, if they also have more tendency to suffer metabolic diseases? Or?
So there is in the UK Biobank data set that we had access to. There was a signal, but it didn't reach genome-wide significance, and that just basically means that we didn't have enough samples to power the statistics. But if I remember correctly, and I would need to go back, there was a signal for that, and we have some additional work that's coming up with some new SNPs for LGR6. So this was only one snippet for LGR6.
Okay, thank you.
We're happy to discuss this later.
Yeah, it can be interesting because of the role that LGR6 may have in adipose tissue and in other metabolic tissues.
Absolutely.
Okay. Nice.
Thank you. We have a question from attendants, online attendants. I'll read it for you. Thank you for your presentation. You presented extensive data on MCTR. In your view, is myrosin mainly active in this particular form? As a follow-up question, are these conjugated forms detectable in the circulation? There are 3 questions actually. And if so, do you consider them to be potentially more robust biomarkers of disease than SPMs themselves, which are often challenging to detect? Okay, I go for the first.
The first one. You might repeat.
Is myrosin mainly active in this particular form, in the form of MCTR?
So MCTRs are part of the myrosin family. So they share a biosynthetic intermediate, which is the epoxide intermediate, and it can go into myrosin 1 or the MCTRs or a myrosin 2. This is a family, so it branches out. So the bioactive forms, as far as we can tell, they're all bioactive. They all have different biologies. And this is why I was trying to stress the fact that so far, most of what we know is really pharmacology, simply because a lot of the experiments that have been done by saying the approach that most of us as basic biologists do. I have access to this molecule, I'm going to give it to a mouse or put it on a cell and see what happens, right? And for me, that's pharmacology. Maybe this is just a philosophical argument. For me, it's pharmacology. The way I look at biology is looking at the formation of the molecule in situ in a specific setting and evaluating what's formed, what's not, what happens if I interfere with the formation or activity of one molecule over another. And this type of experiments, we have not had the tools so far to address at this level, to knock in, knock out specific molecules, to be able, within a tissue, within an environment, to be able to say, if we lose this molecule in this specific setting, what's going to happen? We're currently building some of these molecules. So for example, as we've shown, we've generated a macrophage-specific ALOX1215 knockout mouse. We have different knockout mice for different enzymes in different cell types and so on. So I think all the molecules are bioactive. Some of the biologies at a pharmacological level are overlapping, but that's because it's pharmacology. When we start going into biology and measuring local levels of these molecules in situ, we don't see all these molecules being produced. There's one set over another, and that's where the biology lies, that the pathways are activated in a site and cell type specific manner. Going to the second question, whether these mediators are produced in the circulation, yes, I showed some data, maybe I skipped through it very quickly. In patients with RA, we see these molecules, we've measured these molecules in COVID, and we've observed correlation between MCTRs and disease severity. And to the third question, they can be biomarkers. They are part of the SPM families. It's true, measuring SPMs has been challenging, primarily because they're low-level mediators, and now we are starting to understand why the abundance is low. It's likely because they're local mediators. And when we're measuring at a tissue level, because we're spreading out the levels, because we're measuring over the entire piece of tissue rather than within the site of formation, we're diluting the measurement. And hopefully we'll be able to develop, and there's something else that we're currently working on, trying to develop spatial methodologies to measure lipid mediators at in hotspots, to be able to then understand both the relationship at the biological level and also what they're doing within the environment.
And you said you can detect them
in circulation.
In the blood, in circulation.
Absolutely.
Well, I think that's for the person who asked this question. I hope you got the answer.
I should caveat that the mediators are not produced all the time in all settings in the circulation. And so what we've measured so far and where we see higher levels are in disease settings. So where you have an active inflammatory response. In healthy settings, we don't necessarily measure these. We don't they're below our threshold so far, at least. Doesn't mean that if we come in 5 years' time with more sensitive equipment, we won't be able to measure them. But at least so far, they're below the limits of in health. In disease, it's a slightly different story.
All right. Well, I have more questions, but because okay, there, in the back. Good. Over here, yes, good.
I can speak up. Thanks for a very nice presentation. It's so good to see that at the William Harvey there's still work on COX being done. So the question I have is the Metabolism concentration. Do you know any measurements in cerebrospinal fluid? Would the concentration be sufficient to make this detectable?
So we've measured them in patients with meningeal tuberculosis.
Okay.
We have a study with a group in Vietnam with Guy Thwaites looking at TBM, and we do see that there is a loss of MCTR formation, and actually MCTR levels, loss of MCTR is linked with a negative prognosis in patients with TBM.
Yeah, so naturally, neuroimmunologists are like 10 years behind the real immunology. But I was always fantasizing about, roles of IKB alpha and beta and loss along the way, turning an acute protective kind of inflammation into a chronic phase. And this tipping point would be very interesting to look at this system. So thank
Absolutely.
But you know, if you have a setup to measure, I'm happy to send you CSF.
Happy to.
Thank you.
Happy to.
Okay, well, because we are a little bit behind time, we can continue afterwards, and then we move to the next speaker, María Jesús, Dr. Moreno Aliaga. Thank you very much. Dr. Moreno Aliaga is a full professor of human physiology at the University of Navarra and responsible for the molecular nutrition and metabolism area in the Center for Nutrition Research. She has developed her research activity at the University of Navarra and at the University of California, Davis. During the last 25 years, her research has focused mainly on the study of obesity, adipose tissue physiology, and metabolism. Her group has investigated the mechanism linking obesity and aging with inflammation, insulin resistance, and fatty liver. Particularly, her studies have focused in characterizing the potential therapeutic action of omega-3 derived specialized pro-resolving lipid mediators, such as myresin-1, in obesity and related metabolic disorders. Dr. Moreno Aliaga has published around 170 scientific articles and 26 book chapters. She has participated in 30 research projects and contracts, 17 as principal investigator. Currently, she is the principal investigator of a group of the CIBER, Physiopathology of Obesity and Nutrition, of the Carlos III Institute of Health, and of the research group Personalized Nutrition and Metabolic Syndromes of the Navarra Institute for Health Research, IDISNA. Dr. Moreno Aliaga is editor-in-chief of the Journal of Physiology and Biochemistry.
Thank you, Pilar, for the introduction. And it's a pleasure for me to be here with all our invited speakers and to share with you some of our research results in the area of obesity and inflammation and the potential role of therapeutic potential of pro-resolving lipid mediators. Okay, as you know, obesity is a very high prevalence disease in our society, and is a huge problem because it's increasing the risk of the development of another diseases like the type 2 diabetes, liver diseases, heart diseases, even cancer. And adipose tissue plays a very important role in the development of all of these obesity-associated diseases. As you well know, adipose tissue is a complex organ. Traditionally, adipose tissue is divided in white and brown adipose tissue, and even the white adipose tissue can be accumulated in the visceral and subcutaneous depots, and even if they are white adipose tissue depots, they have important metabolic differences, and it's important in which of these depots we accumulate fat. And as you also know, brown adipose tissue is primarily a tissue involved in energy expenditure with a different role to the brown adipose tissue. Okay, therefore, it's not only, of course, the amount of adipose tissue that we have is important, but also is very important the distribution of this adipose tissue, and because this and how this adipose tissue expands. I'm not going to talk about this because we have here an international expert in this, Dr. Vidal Puig, that will be talking later about this, but this is important for the metabolic health in obesity. Okay, I just going to focus in the role of chronic inflammation in obesity and aging. Obesity has been defined as a chronic inflammatory disease, and this inflammation, this chronic lower inflammation, it seems to start within the adipose tissue and finally can affect other tissues and is linking the risk for the development of these diseases. Also, aging has been considered a chronic inflammatory disease. In fact, it has been this chronic inflammation associated to aging is called inflammaging. There are a lot of evidence of this crosstalk between obesity and aging. And there are evidence showing that obesity can be considered as a premature aging phenotype, and in this situation, the inflammation, chronic inflammation plays an important role. And also, there are evidences showing that obesity may accelerate the aging process. And again, there are the hallmarks of aging, and here the inflammation plays also an important role. Okay, focusing in adipose tissue, there are a lot of changes that occur within adipose tissue during obesity, but also in aging. In aging, there are a redistribution of adipose tissue. As we age, we tended to accumulate more fat in the visceral depots than in the subcutaneous depots, and this accumulation in visceral depots is increasing the risk to suffer the cardiovascular diseases and other metabolic diseases associated to obesity. Also, during aging, there are a reduction in the activity of the brown and beige fat, which can also contribute to the development of obesity and the related diseases. And there are also some other alterations in the precursors of the adipocytes and the senescence of these cells. And all of this facilitates this aging adipose tissue, facilitates the ectopic accumulation of lipids in other tissues. There are a dysregulation in the secretory pattern of this adipose tissue, favoring the development of obesity and inflammation. Okay, in fact, there are some articles that they are linking the inflammaging to the adipaging. This inflammation that starts in the adipose tissue may contribute to the inflammation of the other tissues, both in obesity and in aging. Okay, in this slide we can see how this inflammation develops in white adipose tissue in obesity and aging. There are more infiltration in obesity. The hypertrophic adipocytes secretes more pro-inflammatory adipocytokines, and also there are a recruitment of pro-inflammatory macrophages and other immune cells favoring the inflammation within the adipose tissue. As Guillermo has elegantly presented, resolution of inflammation, we know now that it's an active process that involves the production of a series of SPMs, like lipoxin, resolvins, protectins, and myosins, and how can be related. The question is, do these have SPM some role in the inflammation associated to obesity? Okay, there are some studies that are shown that there are an impaired local production of SPMs in white adipose tissue in obesity. Okay, you can see here that there are a reduction in some of the precursors of some of these SPMs, in protectin D1 and other molecules involved in these SPMs productions. That means that these chronic inflammatory situations that we found in adipose tissue in obesity can be also derived from this impaired local production of these SPMs. Okay, after this observation, there were several studies analyzing the potential therapeutic effects of some of these pro-resolving lipid mediators, like resolving E, protecting D, or resolving D1, on the inflammation in adipose tissue. And as you can see here, this is a nice picture showing that these molecules can increase the production of adiponectin, which is an anti-inflammatory adipokine, as is reducing the pro-inflammatory ones, like lectin, TNF-alpha, interleukin-6, or MCP1. So it's reducing the infiltration of macrophages and is switching the M1 macrophages to M2 macrophages. Okay. I'm going to the last year has been mainly focusing the effects of mycin-1 on metabolic diseases, mainly obesity and fatty liver disease. And let me show you some of our data of the effects of mycin-1 in adipose tissue. First, we did some studies in vitro in the 333L1 adipocytes, and what we observed is that treatment with mycin-1 is able to reverse the lipolytic action of TNF-alpha. And this action is mediated through the regulation of the main lipases and proteins involved in the control of the lipolytic process. Then we moved to in vivo studies. We treated diet-induced obese mice with mycin-1, and what we observed, it was a decrease in the macrophage infiltration in the adipose tissue of the mice treated with mycin-1. We did a comparison in obese mice treated with mycin-1. We did a comparative study with DHA, which is the precursor of mycin-1. They had similar effects, but the concentration of mycin-1 was 1,000 lower than the one of DHA. We also observed a reduction of M1 macrophage markers, and in parallel with this reduced inflammation of the adipose tissue, we observed an increase in the phosphorylation of AKT and an increase in the GLUT4 expression, suggesting an improvement in the insulin resistance in this tissue. We observed similar results in the OB mice, which are a genetic model of obesity. And another finding that we observed in these mice treated with mycin-1 was that there are an upregulation of markers of beta adipocytes. We observe increased gene expression of genes involved in mitochondrial biogenesis, fatty acid oxidation, and specific beta adipocytes markers, together with an increase in genes with anti-inflammatory profile and a reduction in the pro-inflammatory ones. Okay, we observe effects in adipocytes, in culture adipocytes from human adipose tissue, suggesting that mycin-1 can induce the browning of white adipose tissue. Okay, this moves us to a study of mycin-1 on brown adipose tissue. As I briefly mentioned before, brown adipose tissue is a thermogenic tissue, but also has very important non-thermogenic roles, like the secretion of pathogens, which can affect the regulation of other metabolic organs, is involved in triglyceride clearance, and there are evidence that activation of brown adipose tissue improves glucose homeostasis and insulin sensitivity, and therefore the targeting brown adipose tissue differentiation and activation has been proposed as a therapy for obesity and type 2 diabetes. What happened with this tissue with aging? I mentioned it before that there are an impairment in brown adipocytes development and function with aging, and also with inflammation. Inflammation impairs the insulin sensitivity and thermogenesis in the brown adipose tissue. For this reason, we think that Maresin-1 could have beneficial effects in keeping the activation of this tissue active during obesity and aging. And there are, in this paper that it was published some years ago, they found that the 12 lipoxygenates, which is involved in the conversions of some of the omega-3 fatty acids, EPA or DHA, into the lipid mediators involved in resolution of inflammation. Okay, these 12 logs is involved in cold adaptation and in glucose metabolism by producing omega-3 lipid, particularly the 12-HIPI from brown fat. Okay, this moves us to characterize what is the lipid mediator signatures in a brown adipose tissue. And we did this study in collaboration with Yesmond. And you can see that we observe here in young mice, at 2-month-old mice, we observe that the most abundant SPMs in brown adipose tissue are the lipoxins, which are derived from the arachidonic acid, and then the maresins and protectins deriving from the DHA. Okay, we questioned, are aging and obesity inducing changes in this SPM signature in brown adipose tissue? And can the supplementation with omega-3 fatty acids restores the alteration induced by aging and obesity on SPM signatures in brown adipose tissue? Okay, for this, to study this, we aged mice with a controlled diet until 18 months old, or with a high-fat diet, and with a high-fat diet with and without omega-3 fatty acids. We replace part of the saturated fat of the high-fat diet with omega-3 fatty acids, and what we observe is that there are, with aging, with controlled diet in aging, there are a reduction in UCP1 levels, protein levels, just by the aging, and this reduction was much higher in the mice fed with the high-fat diet, and that we can reverse this by supplementing the diet with omega-3 fatty acids. We also characterize the SPM's signature in the brown adipose tissue of these aged obese mice. Differences in the SPMs. The lipidomic studies showed differences in the SPMs between the different groups, particularly in the group fed with the omega-3 fatty acids. You can see here that in this graph we can see the sum of pro-resolving lipid mediators, and there are a tendency to decrease in aged mice fed with controlled diet, but is importantly reduced the content of SPMs in the aged obese mice. And we can restore even to higher levels than the young mice if we supplemented the diet with omega-3 fatty acids. Here we analyze each one of this group of the SPMs, and as you can see, the ones that are more affected by the aging and obesity is protectin D1, maresins, and lipoxins, the ones that were higher in this tissue. And there are a huge increase in the omega-3 fatty acid supplemented diet. We found a correlation, a negative correlation between the levels of several of these SPMs and some pro-inflammatory genes, like CCL2, and we measure the activity of the brown adipose tissue using the FGF uptake in the micropet studies, and we observe that clearly there are a reduction in the activation of brown adipose tissue with age, but it's potentiated by obesity, but unexpectedly, even if there are higher levels of SPMs, lower inflammation, we were not able to recover the activity on brown adipose tissue. One thing that we want to analyze is how are the receptors? Are these changes in the number or in the sensitivity of these receptors with aging and obesity? This is something that we are now analyzing in these models, because maybe we are increasing the SPMs, but if the receptors are affected, we cannot observe the effects. Okay, the main stimulus for activating brown adipose tissue is co-exposure. We characterize how are the effects of co-exposure on SPF signature in brown adipose tissue. And yes, when we were analyzing that, this very important paper from the group of Yuhua Chen in Joslin Diabetes Centers came, and they observed that the beneficial effects of cold induced in the resolution of inflammation is mediated by the production of myresin 2 from the brown adipose tissue. Okay, they observed that when they expose mice to cold, the production of myresin 1 is increased, and this is mediating the beneficial effects in the resolution of inflammation. That is showing that all of this, that the adipose tissue, the brown adipose tissue secretes these lipid mediators and that they can modulate systemic metabolism. Okay, we also did some experiments, some lipidomic studies. These are preliminary studies to characterize the SPMs that are mainly affected by cold exposure in brown adipose tissue, and just to show you some data that we published previously. Cold exposure induced an increase in the levels of DHA, EPA mainly, and we observe a huge increase in the myresin 1 levels and in the levels of the precursor of myresin 1. This move also to characterize the effects, the pharmacological effects of treatment with myresin 1 on brown adipocytes and in brown adipose tissue. And what we observe here is that treatment of brown adipocytes with myresin 1 upregulates the expression of genes involved in the activation of brown adipose tissue, UCP1, and other genes involved in glucose uptake by the tissue, fatty acid oxidation, mitochondrial biogenesis, and some other genes that are known to have thermogenic actions. We also characterize the oxygen consumption of these adipocytes treated with myresin 1, and you can see there are an increase in the oxygen consumption rate. There are an increase in the temperature in these brown adipocytes, and in parallel with the changes at the level of gene expression, we did some functional studies, and we observed an increase in glucose uptake by these adipocytes and an increase also in fatty acids uptake and in fatty acid oxidation. Okay, but what is occurring in vivo? We treat mice with myresin 1, and we observe that treatment with myresin 1 was able to reduce the fat accumulation induced by the high-fat diet in the adipose tissue, in parallel with an upregulation of the UCP1 in brown adipose tissue metabolism and function, suggesting that in vivo myresin 1 is also activating brown fat. We did some micropet studies, and we observed an activation of brown adipose tissue. And it is well established that alternatively activated macrophage, M2, are positive regulators in the control of brown adipose tissue thermogenic activities and also in the bad browning. For this reason, we characterize if myrisin-1 can promote the increase of the accumulation of these M2 macrophages in brown adipose tissue, and we observe that there are reduction of some M1 markers, and on the contrary, there are increase in some markers of M2 macrophages, like the IL-4, CD206, at the level of mRNA and also in the immunofluorescence studies. In order to try to establish this connection between macrophages and the action of myrisin-1 in macrophages and the action on brown adipocytes, we treated brown adipocytes with conditioned media of macrophages treated previously with myrisin-1, and we observed that the macrophages previously treated treating brown adipocytes with conditioned media with macrophages treated with myrisin-1 upregulates the expression of UCP1 and other genes that I'm not showing here related with adipose tissue, brown adipose tissue activation. In order to get more insights about the mechanisms involved in this activation, this action of myrisin-1 on brown adipose tissue, we got interested in the IL-6 because IL-6 has been, even if it is considered a pro-inflammatory cytokine, it has been described by thermogenic properties from IL-6, and also it has been involved in mediating the pro-resolving effects or metabolic effects of other SPMs, like sound protectins, and then we treated mice, we treated interleukin-6 knockout mice with myrisin-1, and as you can see here, the myrisin-1 does not stimulate thermogenic genes in brown adipose tissue of the interleukin-6 knockout mice. And that means that the presence of interleukin-6 is necessary for these thermogenic effects of myrisin-1, and we demonstrated this because when we administer recombinant interleukin-6 to this myrisin-1 treated, we administered recombinant IL-6 to the knockout mice, we recover again the effects, the thermogenic effects of myrisin-1. Okay, but what is the mechanism? When we were doing these studies, it was discovered that IL-6 was the receptor mediating the action of myrisin-1. Then we characterize if this receptor is expressed in brown adipocytes and in adipose tissue, and we observe that there are an expression of IL-6 in both brown and white adipose tissue, but it's much higher expressed in brown than in white adipose tissue, and that myrisin-1 induces upregulates the expression of IL-6, and we do some silencing studies using siRNA to silence IL-6 in brown adipocytes, and as you can see here, the silencing of IL-6 totally abrogate the effects of myrisin-1 on the upregulation of UCP1 or PGC1 alpha. That means that IL-6 is necessary for the action of myrisin-1, but we found that it was very important that by silencing IL-6, there are, even in basal conditions, there are a reduction in UCP1, PGC1, and other markers of adipose tissue, brown adipose tissue function. Right now, our ongoing studies are focused in characterizing the role of these receptors in brown adipose tissue, the physiological role of IRCs in brown adipose tissue. Okay, then just to finalize my talk, I'm going to talk about MALS-D and obesity and aging. Ruben has shown this morning very nice studies about this metabolic associated steatotic liver disease. And of course, obesity promotes the development of MALS disease, but also aging. During aging, there are an increase in the risk to develop fatty liver disease, and also the risk to steatohepatitis and the complication of cirrhosis or even cellular carcinoma. And we are studying characterizing the role of these SPMs in as potential therapeutic targets or strategies to combat MALS-D. This is a slide summarizing the effect of some omega-3 fatty acids at some SPMs like resolving D1 or resolving E1, which has been shown to reduce some of the alterations observed in fatty liver disease, or like the ER stress, the autophagy. And, but I want to focus mainly in the effects of myrisin-1. We treated myrisin-1 with different doses in different models of obesity, and what we observed is that treatment with myrisin-1 can reverse the accumulation, ectopic fat accumulation in the liver induced by the high-fat diet, you can see here, and is reducing the triglycerides accumulation in the liver, and this probably is mediated because myrisin-1 is reducing the levels of proteins involved in lipogenesis in the liver. It's increasing the phosphorylation of ACC, and also is controlling the lipophagy in the liver. Similar effects were observed when we treated diet-induced obese mice with myrisin-1. The difference between the previous study and this study is that most in the literature, most of the study were done administering lipid mediators by intraperitoneal injection. We think that if for the potential therapeutic actions of these molecules, it's important to know if they are active by oral GABAs. We don't see reason for the inactivation of myrisin-1 by oral GABAs. We did this study administering by oral GABAs myrisin-1. As you can see, the effects reversing fatty liver are huge. There are a huge decrease in the fat accumulation due in part to this reduction of lipogenesis genes and upregulation of genes involved in fatty acid oxidation. Again, we observe that myrisin-1 promotes autophagy in the liver, which can also contribute to the reduction in the lipid content of the liver, and we observe that phosphorylation and activation of IMPK may be mediating these actions. Our study is not the only one focusing on the effects of myrisin-1 in fatty liver disease, and there are other papers that just came almost at the same time that our paper, showing the capacity of myrisin-1 to prevent the inflammation and progression of steatohepatitis in mice. But all of these studies has been performed in male mice. All the studies in the literature with myrisin-1 has been performed in male mice. And as you know, there are important sex differences in the risk to develop fatty liver disease. In human women, we are protected in comparison with men to the risk to develop fatty liver disease in the reproductive age, but in the postmenopausal age, our risk to develop fatty liver disease increased, even higher than the risk in men. And the same occurs in the animals models of obesity and fatty liver. And right now we developed a model of obese mice by feeding the mice for a long time with a high-fat diet. These mice develop, they are female mice, they develop fatty liver disease. And in this model of high-fat diet and aging-induced fatty liver disease, we treated these mice with Maresin-1. And let me just show you some preliminary data that we have. We observed that it's reducing moderately the content of triglycerides, but we isolate mitochondria from this liver, and we, as you know, mitochondrial dysfunction plays an important role in the in fatty liver disease development and progression. And you can see here that the mice treated with Maresin-1, they have a higher oxygen consumption rate. We are now trying to characterize the mechanisms. And we performed a transcriptomic study of the liver of these mice. And you can see we have observed, we have detected an important number of differential expressed genes. The gene ontology enrichment analysis showed that these differential expressed genes are mainly involved in pathways involved in fatty acid metabolic processes, but there are many of the pathways with genes with differential regulation are those involved in cytokine production, immune response, ER stress. Then, to show you some heat maps, we observe a downregulation of genes involved in the inflammatory response, even in the acute inflammatory response, and also a downregulation of ER stress genes. I hope to be able to show you in future meetings more data about this. And just to conclude, the defect in SPM's production could underlie chronic inflammation and the metabolic alterations associated with obesity and aging, such as the decrease of brown adipose tissue activity. Regarding this tissue, we know that cold exposure modified SPM signatures, supporting that these bioactive lipids could be key determinants of brown adipose tissue thermogenic function. And this highlights the relevance of to further characterize the physiological role and therapeutic potential of specific SPMs and their receptors in brown adipose tissue development and function. Just focusing on Maresin-1, we think it's a novel agent that counteracts obesity-induced inflammation in brown adipose tissue and promote in white adipose tissue and promotes brown adipose tissue activation. We think that the LGR6 receptor is necessary for Maresin-1 effects, but we think that this receptor can be important in the metabolic function of brown adipose tissue, and that Maresin-1 alleviates liver steatosis, inflammation, and ER stress in MALS-D. And in summary, we think that Maresin-1 and potentially other PUFAS-derived SPM may represent promising therapeutic targets to tackle obesity and aging-related comorbidities, including the insulin-resistant type 2 diabetes or fatty liver disease. And thank you for your attention. I want to thank you, the people, current and previous people in our group that were working in these studies, mainly Laura La Iglesia, Xavier Escoté, Elisa Félix Soriano, Neira, Silvia Lorente, Pedro González, and currently Miguel Burgos, Jesus Saborido, and other members like Asun, Paula, and Marta that performed contribute to all of these studies. Of course, our collaborator, Yes Mondali, our collaborators in CIMA, and the University of Barcelona, the group of Francesc Villarroya, Ángela Martínez Valverde, María Collantes, and José Miguel Arbones Mainar, and our funding for different sources. Thank you very much for your attention.
Time for questions, or here?
Congratulations for the fantastic results.
Thank you.
When you treat your obese animals with Maricin 1, do you see any change in food intake, or all the effects are exclusive of white and brown adipose tissue?
Yeah, very good question. We did not observe significant effects of food intake. We did some studies, and there are no big changes in body weight, and there are a small decrease in fat accumulation, but very small. It's more decreasing the inflammation, and is activating the brown adipose tissue without big effects on body composition. They are very moderate.
Have you ever checked the expression of Maricin 1 in the adipose tissue or liver of people with obesity?
Yes. We did this study with Yesmond because it was a thing that we think it was very relevant. How much are the levels that we reach after we treat with Maricin 1? And we did it in the obese mice, these studies, treated without and with Maricin. The levels of Maricin 1 of obese mice without Maricin 1, the levels of Maricin 1 in brown adipose tissue and in liver were very low, and after the treatment it was increases. So we measure also in young males, in young mice, and the levels were higher than the obese mice. Yeah.
Thanks for a great presentation. I have a question to the receptor side,
Yes.
to LGR6. So many of the resolving receptors have a very quick desensitization. Like TREM2 is one of the receptors that rapidly invaginates and turns over upon ligation. So is it known how quickly the receptor desensitizes or is turned over? Because I was wondering if you would develop a pharmacological drug to maintain that kind of suppression, how effective would that be? Or would you see a similar receptor desensitization like people have now seen with TREM2 and other targets?
Yeah, this is a very, very interesting question. Because we think that it can be sound, as we saw in the aging study, we are now characterizing if there are a loss of the function or the activity of these receptors by obesity and aging, but also it's very important if there are some desensitization of these receptors after chronic treatment with the SPN, something that we have to analyze and characterize. Yes, very interesting.
Thank you. That was fantastic, Maria Jesus. So my question was linked with where do you think the strongest or the main part of the biology, whether this is synergistic, is it in the regulation of the immune response, the inflammatory component, or is it directly through the regulation of the adipocyte biology, or is it both?
We think this is a combination of both, because we have observed changes in the markers of the M2 macrophages. Right now we are going to do a new study treating mice with Maricin 1, and we want to characterize, to isolate of the stroma vascular fraction of the brown adipose tissue and the white adipose tissue, and to characterize the immune cells that are there and how are they affected. But we think that there are direct effects also on the adipocytes. Yeah, and we are planning to do some studies just deleting the receptor of myrisin-1 exclusively in adipocytes just to see if it's in vivo, to see if the effects of myrisin-1 still keep there or not.
Thank you.
More questions? Thank you very much, Maria Jesus, for the wonderful talk. And now we have the honor of having the closing conference by Antonio Vidal-Puig. Some presentation, as a presentation of Dr. Antonio Vidal-Puig. He obtained his medical degree from Valencia Medical School before training in clinical endocrinology at Granada Medical School, where he obtained his PhD, based on clinical and physiological studies of the relationship between insulin resistance and hyperandrogenism. The award of the Paul Dudley White Fellowship from the American Heart Association funded his postdoctoral training at Harvard University, and in the year 2000 he moved to the University of Cambridge, where he developed the Obesity, Diabetes and Cardiometabolic Complications program. Professor Vidal-Puig is currently the Professor of Molecular Nutrition and Metabolism at Cambridge University and Honorary Consultant in Metabolic Medicine at the Addenbrooke's Hospital in Cambridge. He's also Deputy Director of the Wellcome Trust MRC Institute of Metabolic Science and Director of the Cambridge Genomic Center, among other appointments. Dr. Vidal-Puig maintains a close relationship with Spain as a researcher at the Príncipe Felipe Research Center in Valencia, Spain, and Great Juan Carlos University collaborator. He directs the biomedicine program at the Center for Technology and Innovation at the University of Cambridge in Nanjing, China. Is also a concurrent professor at Nanjing University, Shokou University in China, and Chiba University in Japan. Dr. Vidal-Puig is a member of the Center for Artificial Intelligence Technology of Humankind at the National University of Singapore Business School. His research program focuses on the molecular mechanisms of lipid-induced insulin-resistant and lipotoxicity modulating fatty acid oxidation and thermogenic mechanisms. Dr. Vidal-Puig is the author of 260 scientific articles and has trained many PhDs and postdoctoral students, many of them established in Spain. He has received important awards such as the International Hippocrates Award for Research in Nutrition, Phelps National Lecture Medal of the British Endocrinology Society, among many, many others. Finally, Dr. Vidal-Puig is interested in innovative strategies for management in research-intensive academic institutions, and for that he has graduated with an MBA from the Judge Business School of the University of Cambridge, and is part of a research group aiming to translate the knowledge available in other business-related areas to academic management. We welcome Antonio Vidal-Puig.
Okay.
What happens?
I don't know. Controlling this.
Please, the slides. Sorry? Aceptar. No. Cancelar, cancel. But they should do it from there. All right. Sorry about that. Sorry. There you go. Thank you.
Okay. So thank you very much for the invitation here in Pamplona.
It was a long time overdue, and I'm happy to be here. And I'm going to provide an overview about our program. Okay, our main problem is to try to understand why obese people develop cardiometabolic complications. And the hypothesis that is driving the whole program is the concept that is the adipose tissue failure either to expand or to function, which results in peripheral lipotoxicity and cardiometabolic complications. So you are familiarized with heart failure and liver failure and kidney failure. So I want you to think about the concept of adipose tissue failure as a key factor determining when an obese individual will develop complications. So the concept of healthy obesity and unhealthy obesity is very controversial. Whenever I bring it up, there's someone saying, there is no healthy obesity. Fine, I agree with that. The concept here is, when do you develop the complications? So I would rather get my complications later on in life than early in life. So I will define the healthy obesity, some of these individuals that are relatively resilient to the toxicity that obesity determines on them, so that they can get to a relatively old age without having these complications. The way we think about this is that the main difference between the healthy obesity and unhealthy obesity from this perspective is A, the redistribution. The obese and healthy people tend to lose the subcutaneous and accumulate intra-abdominally. And the other thing is that because the subcutaneous is an important depot, if it's dysfunctional and if it is failing, then the fat is going to go to other organs, including the muscle, the heart, the liver, so on. The issue is that not everyone develops everything, or in this respect, that suggests that you may have certain degree of vulnerability so that your genes in your specific organs may not be that well prepared to deal with excess of nutrition. So that's why the variability comes. You get an excess of nutrients, which is a toxic asset, and then your genes in your organs will try to defend themselves, and some of them will be more resilient than others. And that doesn't mean that over time, as these organs start to fail, there's a knock-on effect in other organs, so that at the end you get the whole combo. Maybe at the beginning you may identify your main vulnerability, but by the end of the process, many of your organs have failed simply because what we call the allostatic load of the system has been taking over the resilience. So that's the concept that we have. So when you think about this, we start from the point of view of the adipose tissue, the concept of obesity plus aging. This is the perfect storm. So we are asking the adipose tissue to perform more when you are getting older, when your muscle is not performing well, when they are not oxidizing well. Then is when you're asking more from your adipose tissue. It's like when you're asking the grandparents to take care of your children when they are late in life and they kind of cope with that. But this is the same concept. The other thing is that these lipids that cannot be accumulated in the adipose tissue, they end up in the muscle, in the liver, in the heart, in the vessels. But one thing that we learn is that there is an intermediate stop, which is the immune cells in the adipose tissue. And in this respect, now when we focus in the adipose tissue itself, we focus in the adipocyte, but also in the immune cells, and the possibility that the lipid metabolism in these immune cells get dysfunctional as the adipose tissue fails. So when we talk about lipotoxicity, which is the key concept, that refers to the topic accumulation of lipids outside the adipose tissue in organs that are not designed to accumulate that. But then when you accumulate the lipids there, you may have different degree of toxicity. And that results, the phenomena that you observe are the ones that we have discussed today. Related to inflammation, ER stress, ROS, signaling defects. For us, it's very important the changes in the membrane fluidity, because these lipids are going to be affected in the context of lipotoxicity. Also, the bioenergetic system will be defective, and also your system of extracellular or communication through vesicles may be affected by having the wrong address and the wrong messages signaling through the body. But we think that before this is the toxicity of the overnutrition. And we prefer to focus in the first mover than when the whole thing gets out of control. It's more difficult to control when things get absolutely losing their homeostatic control. And by focusing in the early stages, you may prevent the fire from getting in. So that is the way how we conceive that. So the way how the program is organized around lipotoxicity, focus in 3 different levels, and I will try to cover some of this. The first thing is the concept that if you keep the adipose tissue as a healthy adipose tissue, then you will be able to accumulate the fat in the adipose tissue, and this will not leak, and you will prevent lipotoxicity. That means the lab is interested in learning how you can get a healthy adipose tissue, or how can you recover the adipose tissue. The second aspect is, if your adipose tissue is an adipose tissue failure, then you need something to clean up. You need an organ that will clean up the muscle and the liver and the heart. And the vacuum cleaner for us is the brown fat. The main function of the brown fat, obviously, is to make heat, and in the small mammals is to maintain the temperature. But for us, the brown fat is an organ that will consume the excess of nutrients in the form of lipids or in the form of glucose, and will help the liver and the muscle and the heart to, in some way, be protected from the excess of the nutrients and prevent toxicity. And the third approach is that if your adipose tissue is failing, if you are not able to burn the excess of energy, or you are not exercising, or you cannot exercise, for example, then the third approach that we propose is a palliative approach, which is to try to understand the lipid networks, try to understand the ones that are more toxic and less toxic, and trying to manipulate these networks to move from the most toxic to the less toxic. So and for this, we focus in the liver, because the liver is an interesting organ to study lipid metabolism. We think it's the first barrier of defense when you get lipotoxic is the liver. And in some way, the liver is the one that protects your muscle and your beta cells. So I think understanding here as a model can help us to devise other strategies for other organs. So my lab is, if we are good at something, was mouse genetics. And I remember working with animal models, and some animal models were very obese and relatively healthy, and some other animal models that we have engineered to limit the adipose tissue, they were obese, less obese, and extremely unhealthy. So that led us to think that maybe the fat mass was not the main determinant of the disease, but actually the uncoupling between your positive energy balance demands, so what you need to store, and what your adipose tissue is able to store at any given time. It's a relative concept. So that could explain why there are some people in the obesity clinic that are 140 kilograms and they are not even glucose intolerant, and why there are people in a lipid clinic that are 85 kilograms and we blame them that they need to lose weight because their insulin resistance, diabetes, dyslipidemia, and second, prevention from a heart attack can be justified because they are only 85 kilograms. This suggests that these people are inappropriately unhealthy for the degree of obesity. So the way we conceive that then is the concept that even though in the obesity clinic you think the adipose tissue expansion is infinite, we think your genes and epigenetics are going to define how far you can go. You cannot become as fat, as obese as you want. You may be as obese as your adipose tissue will allow you to be at one time. So in this respect, what we think is that from the point of view of the complications, it is more important the remaining capacity to expand of your adipose tissue. This is a more important determinant of the complications, the absolute amount of fat. This is a key concept that allows you to understand your patient in front of you and what you may expect from them with respect to improve the treatments. And we think that there is a point of maximal expansion. Before that, you may get an adipose tissue which is dysfunctional that determines when this adipose tissue is not going to have good capacity to retain the fat and where they will be leaking the fat and your macrophages will be just facing with the load. So when we focus on the problem of a healthy adipose tissue, in some way, the lab has been focused on some of these questions. Obviously, this will depend on your pre-adipocytes and your program of adipogenesis. We think that the immune system in the adipose tissue has a physiological role, which is to promote the remodeling and allow the expansion, but under toxic conditions, you may have inflammatory changes. We have the vasculogenesis, because the function of the adipose tissue is not only to store fat, it's to mobilize the fat. You need to get fat in and fat out. If you don't have proper vascularization, you're going to become hypoxic and pro-inflammatory. A key factor is the remodeling of the connective adipose tissue. If you cannot remodel your adipose tissue, you cannot expand, and that is going to limit. A key factor that people don't focus enough is the lipid membrane composition. Probably it's not as friendly as other type of problems, but we think in an obese individual, changes in lipid composition of the membranes may determine the characteristics of the adipocytes. And the last one, and the one that we are going to be focusing more in the future, is this one, metabolic flexibility. It's very interesting because all the 6, you can see them. You get a biopsy and you see something there. This one, you don't see it. This is about the speed. The speed about how the lipids get into the adipose tissue and the speed at which the lipids get out of the adipose tissue. And we have evidence that this is probably defects in metabolic flexibility from this concept is one of the first ones that appear dysfunctional in an obese individual whose adipose tissue is poised to become dysfunctional. So in this respect, our laboratory has contributed to identify some of the key players for adipose tissue differentiation. But one of the points that for me is very important is the concept of titration. So when you have the adipose tissue, you always have this paradox between the hypertrophic and hyperplastic. And it's the concept of when your adipocytes are full enough to decide that they need recruitment of new adipocytes, because you cannot conceive a situation where, under specific signals, you will have an inappropriate increase of differentiation of adipocytes that will be probably bioenergetically not efficient and will lead to a hyperplastic phenotype. Or when your adipocytes will be taking fat and not being able to signal, we need some help for recruitment. And for this, we were focused in aspects of the Wnt signaling with respect to the crosstalk between preadipocytes and mature adipocytes to determine the moment when your preadipocytes can be titrated. The second aspect is related to the adipose tissue macrophage and inflammation. When you look at the literature, you get the perception that the macrophages in the adipose tissue are bad people. They are there just to cause inflammation and problems. But one of the first things that we learn from these macrophages in the adipose tissue is that in a healthy state, they have transcriptional programs that are fundamental for the remodeling of the extracellular matrix. However, in the adipose tissue of an unhealthy insulin-resistant and inflamed mouse, this program disappears. And what you get is a paradoxical increase in the pro-inflammatory changes, and you wipe out the program of the remodeling. However, when you look at the macrophages of the adipose tissue in an obese model, what you see is this. This looks like a foam cell. It's full of fat. It's the first impression that you get from this is thinking, well, these macrophages are macrophages, but they are full of fat, and that must not be too healthy or too good, even though there are situations where the macrophages accumulate fat and it's healthy. But then we focus a bit more on this, and we came out with 2 conclusions, or 2 outcomes. One of them was to focus in the de novo lipogenesis in the macrophages in the adipose tissue. And one of the things that we learned is that there is an increase in de novo lipogenesis in these macrophages, and we thought that this was relevant from the point of view of the lipid that had been generated through this process. But we failed to show that the increase of de novo lipids in the macrophages were important for the functionality of the macrophages. What we learned instead of this is that the process of fatty acid biosynthesis is using cofactors like NADPH. And NADPH is also utilized for glutathione synthesis in a reduced state, which is fundamental for the levels of ROS and the alternative activation. So from this paper, what we learned is that in a healthy state, the activation of the STREP1 fatty acid de novo by hijacking that allows the system to really produce ROS by decreasing the scavenger and facilitate alternative activation. And conversely, in states of insulin resistance, you may block this, preventing the accumulation of ROS and alternative activation. In some way, you have a housekeeping system that is involving the biosynthesis of the lipids, that is controlling this type of redox situation and the activity of the macrophages. The second thing that we learned was about the question that we were asking is how the inflammation in the adipose tissue is initiated. And here we were looking at the macrophages from the point of view of the membranes. And if you look at the biosynthesis of phosphocholine, what we could see is that there was an induction of the enzymes of the phosphocholine biosynthesis, particularly in animal models that were obese, like an OVOV. So by looking at this, we learned that in an obese model, there is an increase in the biosynthesis of phosphocholine. There is an induction in the enzyme. And also you can see that there is an increase in the induction of this enzyme in parallel with the body mass index, and at the same time with the production of lysopesis. And you could say, well, is the problem the increase in the phosphocholine? Well, what we learned was that in obesity, there is an increase in the turnover of de novo production of phosphocholine, but that this accelerated turnover resulted in the lung cycle, which is the one that is replacing a specific fatty acid long chains in the phospholipid, didn't have enough time to replace them from the membranes, which means these membranes were paradoxically depleted of some of the long chain fatty acid. And what we learned from this is that this contributed, so changes in the remodeling of the membranes contributed to increase the pro-inflammatory changes capacity of these macrophages. Okay, so this has been the adipocytes and the macrophages. And here we talk about the vascularization. As I said before, it's very important for an adipose tissue to be healthy to have proper vascularization. And it's important because the nutrients need to get in and the nutrients need to get out. And this structure, if it's not functional, you will decrease the supply of oxygen to the adipose tissue, and that will result in facilitating pro-inflammatory problems. What we learned from here is that we learned about this molecule. BMP-8 has been one of the key molecules of our laboratory. We learned about this, about thermogenesis. Then we learned about the influence in white adipose tissue, and finally in the progression of NASH. But the key thing about this is this. So BMP-8 is a molecule that is secreted by an adipocyte. It's the center of the universe is in the adipocyte. And this molecule, when secreted, promotes a pro-angiogenic portfolio of molecules and decreases the anti-angiogenic molecules, resulting that a molecule secreted from the adipocyte can control the niche and the vascularization to supply enough oxygen based on the demands of this type of cells. So we learned that BMP-8 facilitates this protein, vascularized protein, and we learned that this was mediated through the ALK7 receptor that we may block using pharmacological intervention. For me, the key thing about this is that when you think on the adipose tissue, you are not only thinking in the adipocytes and the macrophages, you're also looking at the vascularization and the innervation. And we learned that BMP-8, a molecule secreted from the adipocytes, can control the network and the niche of vascularization and innervation, facilitating the homeostasis of the adipose tissue. Okay, so we are getting now in the territory of the fibrosis. And here I would like to comment in what is our direction at this present moment, okay? So we have been working on adipose tissue dysfunction or failure, and considering this from the point of view of an adipocyte, which is dysfunctional because it's overloaded and then is coupled with inflammatory changes, okay? Activity of an immune cell. Then we have confirmed and we have data showing that the regression of the brown fat in obesity and with aging depends again on the cell injury-based defects on this, and then communication with immune cells resulting in a pro-inflammatory change and fibrosis. It's fibro-inflammatory changes. If you look at the liver, we have the same thing. I mean, when you go from the transition from steatosis to steatohepatitis, then the key thing is a hepatocyte that is injured with an excess of nutrients, and then the progression to an immune cell that need to sense that and get activated and produce an inflammatory response. And the fourth organ that we are considering for that is the heart, is the HFpEF, which is the heart failure in obesity and diabetes, where you may have cardiomyocytes suffering an injury from overnutrition and triggering a pro-inflammatory response. So our point of view is that in these four situations, and maybe now in the brain, if we could elucidate the factor that uncouple the cell injury from the fibroinflammatory response, we may in some way prevent the progression of these diseases into more important pathological states. So you think in the fibrosis of the adipose tissue, what you find is that the more fibrosis you have, the more insulin resistant you are at systemic level. If you look at the adipose tissue, fibrosis itself is sufficient to prevent expansion, and you will have inflammation, insulin resistance, and metabolic alterations. and this is very important. If you have fibrosis in your adipose tissue, you are obese, and you have bariatric surgery, and you think you're going to lose weight, you are very naive. Your fibrosis will not allow you to mobilize the fat, and you will be one of these failures of the bariatric surgery because you will not be able to mobilize the fat. So our approach to studying fibrosis, well, to study the problem of the uncoupling of obesity from complication is based on genetics. So we are looking at genes and pathways that decouple obesity from the metabolic complications. When you're working in obesity, traditionally you get obese and complications. You want to see A causes B. The way we look at this is we want to see A uncoupled from B. What are the factors that determine that these two things will not go together? And we think that if we find why they don't go together, we may learn a new type of relationship that may be useful from the point of view of treatments or even biomarkers of progression to disease. So we went to genetics, and we found variants in this gene, PEPD, is prolidase protein. And this is, these two variants have some interesting characteristics. Either they were associated with a low BMI, but in this case, there was an increased susceptibility to diabetes, triglycerides, and insulin resistance. Or other variants in this gene, or SNPs, were the opposite. You have a high BMI, but your HDL is high, your adiponectin is high, and you're insulin sensitive. This is not something that you're expecting to go hand in hand. So what we learned about PEPD is that PEPD has two roles. One is an enzymatic role that happens inside the cell and is important for the collagen turnover. When you are degrading your collagen with metalloproteinases, then you get to D-peptides with acetylproline, and then they enter in the cells, and then PEPD will contribute to the final degradation. And some of these will get recycled, some of them will enter in signaling. The bottom line of this is that PEPD is very important for the turnover of collagen, and if this doesn't work, the expectation will be collagen accumulating. The other thing that we learned is that PEPD, besides being an enzyme, is in blood, is extracellular, can be secreted from the cell that has the PEPD. And when it is circulating, PEPD binds the EGFR receptor and signals through the EGFR receptor. Imagine a molecule, PEPD, is released from the adipose tissue and signaling through EGFR receptor. This could be important in process of EGFR control, cell proliferation, cancer, and so on. Thinking a signal from the adipose tissue is functional with potential to promote cancer development. And the third thing that we are pursuing is the role that, it's a pity that Ruben is not here, but I prefer not to see him this time, is the role of PEPD and the control of P53 and senescence, which is something that we are actively following in the lab at the present moment. The key person for this is Vanessa, who has been instrumental in finding, following, and developing this program. So what we know about PEPD as an enzyme is that you get less activity of the enzyme in humans in the visceral adipose tissue, that the activity of the prolidase predicts diabetes, so that means the increased prolidase activity is associated with diabetes. And in animal models, when you put them on a high-fat diet, again, you get a decrease in the activity of prolidase, a bit less in the liver, mainly in the adipocytes. And then this is associated with the fibrosis in the animal model on a diet. So you can see the fibrosis when PEPD is not being active. If you look at the PEPD circulating, what you have is that in an obese person, non-diabetic and diabetic is increased, and when you compare the non-diabetic and diabetic, it's even more increased, suggesting that this, you can find less activity of the enzyme in the tissue, but more protein circulating in blood in this individual. And we know that the amount of PEPD in circulation is a biomarker of type 2 diabetes. Okay, so we cannot work with a PEPD knockout mouse because the mouse is fibrotic, it's tiny, it's runty, it's a whole fibrosis evolving. What we have learned is that if you work with 50% PEPD activity, which is the 50% that you may get in a diabetic individual, what you have is a decrease in activity, an increase in fibrosis, and this is associated with a decrease in insulin sensitivity and an increase in the amount of fat in the liver. That means your adipose tissue gets constrained, you become insulin resistant, and the fat goes to your liver, which is a lean, linear and healthy phenotype. However, when you look at the extracellular, the one circulating, one thing we learn is, first of all, in the adipose tissue, this type of cells that have PEPD are mainly the macrophages. The adipocytes, they may have it, but the macrophages are the ones that have more. Second, if you treat these macrophages with LPS, as in a human adipose tissue, you will have a decrease in the activity of PEPD, okay? But you will have an increase in the release. We have documented that the decrease in the activity and the release are coupled, okay? And strategies that inhibit activity pharmacologically are coupled with an increase in the secretion of this. The other thing that we have here is that PEPD by itself is able to activate an FKBP and promote inflammation, but if you use an EGFR inhibitor, you can block it, suggesting that PEPD enzymatic action contributes more to the collagen and fibrosis, but the PEPD circulating through the EGFR receptor contributes more to inflammation. And that means that even though in obese people we see fibrosis and inflammation, doesn't mean that these two things are coupled. The same molecule with two different functions may contribute to uncouple these two. And we have done with animal models, in an animal model with no PEPD, you don't have the secreted and you don't have the functional. And we can analyze that. Anyway, so this model, what happens is in obese individual, you have a decrease in the PEPD enzymatic activity, you have an increase in the PEPD release. That results in fibrosis, and this results in inflammation. And this is how, in these models, you couple obesity, you couple inflammation and fibrosis, but not necessarily through the same mechanism. The other thing that we have learned is that PEPD is also relevant in the brown adipose tissue. So they contribute to the regression of the brown fat, which is one of the key problems that we observe in obese individuals. And in some way, we can see that they have more fibrosis, the brown fat is more white, it's in periadipogenesis, and you have an alteration in the extracellular matrix. This is suggesting that the PEPD secreted may have not only effects in the adipose tissue, maybe in other organs like the brown fat, maybe in the heart, maybe in the liver. Okay, so we have been adipocytes, macrophages, vascularization, and fibrosis, and so on. I will not touch the membranes because it's too complicated, but I would like to go through this one, which is the metabolic flexibility of the speed of lipid mobilization. And the two key functions are storage and mobilization. And we learned that one of the isoforms of PPAR gamma 2, not PPAR gamma, only this isoform, which is very interesting because it's the only one that is increased with the feeding and decreased with the fasting. And we were always worried about an adipogenic gene that changes with feeding and fasting because this is not how you regulate adipogenesis itself. So this isoform is adipogenic, but this probably is most important. So what we learn is this. Okay, a bit of metabolic flexibility. So this is related to the RQ, which is related to the specific consumption of carbohydrates and lipids. If you have an RQ which is close to 1, then that means your body is consuming carbohydrates. And this has to do with the amount of oxygen that you need to oxidize it. And if you are consuming lipids, the RQ will be close to 0.7. So there's a difference of 0.3 in the system, okay? So here what you have is that you get a lean, healthy individual. When they are in fasting, they are using mainly lipids, which means their RQ is around 0.8. And when this individual is feeding or is on insulin, they will be using preferentially glucose, and then the RQ goes up. You have a big excursion. This is an obese individual, meaning in the fasted state, the RQ is higher than you expect, and in the fed state, it's lower. I mean, it's not moving, like there's no change. And if you are obese and you lose weight, then you start in the fasting, you don't see differences, and you recover part of the effect of insulin with respect to the RQ. So in some way, we are interested in the concept of the delta RER as a measure for the metabolic flexibility of the system, okay? And then in the PPAR gamma model, this is gamma 2 model, this is what we observe. This is the RQ, RER, and then this is during the day when the animal is not eating, is fasting, and then the RQ is low. And this is when, in the dark hours, when the animal is eating. So that means the RQ should be higher. In black, you have the wild type. In white, you have the PPAR gamma 2. And then, sorry, you look at this, you can see that the wild type has a larger delta RER than the knockout mouse. And then this difference in delta RER are a global estimate of systemic usage of lipids or carbohydrates in the context of the metabolic control. So we are very interested in this parameter with respect to identify mechanisms that determine metabolic inflexibility. One of the key things that we learned from these animals is that the PPAR gamma 2 is glucose tolerant. Inappropriately, we didn't expect this to be glucose tolerant because PPAR gamma is supposed to be a target for insulin sensitizer, am I right? But it takes time for these animals to become glucose intolerant, if at all. But if you do a lipid tolerant test, this is what you get. These animals don't get fat in, and they don't get fat out. And if you give back garbage to them, the lipids accumulate in blood for a longer period of time. So, Hema, I need you to go to the clinical research and do lipid tolerant tests to people from your lipid clinic with familial combined dyslipidemia to show that they have an enrichment in problems with metabolic flexibility and an increase in discussion of the lipid profile. Very early on, even if they are not glucose intolerant, because we never test the ones that are not glucose intolerant because we assume they are okay. But that is uncoupled, okay? So then what we have done, which is the most exciting part of our future, is we have gone through the IMPC and do the delta RER of 5,000 animals. And we have got the distribution of areas for the low and the high, which in some ways is telling us the ones that are high metabolic flexibility and the low metabolic flexibility. And now we have the first genetic screening in vivo for genes that are important for metabolic flexibility. That doesn't mean that all of them are going to be adipose tissue, maybe related to the muscle, to the liver, to other ones, and that opened up a new type of research program based on genetic basis of metabolic flexibility, whatever the mechanisms they are. So that has been the first part of the story. I mean, this is the strongest part of the program. I want to mention this. This is the implications of the adipose tissue expandability concept. So if you talk to an epidemiologist, they will tell you the fatter you are, the more insulin resistant you are. So this is true. We know epidemiologically this is consistent. However, the way we see this for the individual is a sigmoid curve. These are people that put on weight, nothing happened. They are no more insulin resistant until one day they reach a specific weight and they crash. And then you will tell me, how do you go from this sigmoid curve to the linear curve? Okay, this is the explanation. I mean, these ones are the ones from maybe from the lipid clinic. These ones are the ones from the obesity clinic. The one from the obesity clinic has been putting weight forever until a moment where they crash. And I agree with you that being obese is never healthy until you reach your threshold. This one is someone who put a bit of weight and they crash. If you look at the probability frequency of these ones and these ones, it's much less than the one in the middle, which represents the majority. And if you integrate this, you have a linear function. So we think this concept is highly applicable to the Asians, the Chinese, which we think are going to the left. These are people who may have problems with respect to expansion and metabolic complications. And we say this based on this concept, which is the Asian diabetes phenotype. People in Asia have low BMIs, a bit more visceral than subcutaneous, but in absolute terms, they have less fat. They have all these other issues that may contribute, but they have increased cardiovascular risk, increased renal disease, increased cancer prevalence of that. And we think that this type of concept may be very relevant to try to understand the increased susceptibility of this population to metabolic stress. The other thing that Maria Jesus mentioned is the fatty liver and the paradox and the concept that there is lean people with fatty liver. Look at this list of countries: India, China, Japan, Korea, Hong Kong, Japan, Canada. So we are talking an Asian phenotype with 15% of these people being lean and having fatty liver. I think it's overrepresented. And with respect to, I mean, we have a project here in Spain now, which is Metabolism, funded by Acacia. And then you have this sequence that Maria Jesus and Ruben have been presenting with respect to healthy steatosis. So this is our point of interest. We are very interested in what needs to happen from a steatotic liver full of fat to promote the development of inflammation. And that's similar concept to the adipose tissue, the liver, the heart, and so on. In this program, we have focused initially in two concepts. One of them is the clinical heterogeneity. You can have a fatty liver for 20 years, and you could be okay. You can have a fatty liver for 3 years. You have an accelerated progression that will get you into steatohepatitis, very severe, and be a fast progression. And we don't have a clue how that happened. The second thing that I decided to focus is in the concept of transferability from preclinical models to clinical models. In some way, because we have been always accused that the mouse is not relevant to model human disease. And we think that in some way is determined by your approach to the phenotyping and your capacity to humanize the mice so that they recapitulate better some of the conditions of the humans. And I think putting them at thermoneutrality may not be a waste of time. So in this regard, what we have done is to make, to establish some kind of ranking of animal models that recapitulate the human phenotype. For this, we have been working now with 42 different models provided by academics and industry. Many of them are based on dietary changes, some of them based on genetics. We have been having access to the clinical phenotype of the animals, to the histology and to the transcriptome, and we have compared this data with data from human data from the litmus cohort to try to determine first what we call proximity scores with respect to the histology, to the molecular events, and to the phenotyping, and combine them in a score that allows you to do a ranking. So this ranking was supposed to be, the objective was to find the best mouse model to model fatty liver disease and steatohepatitis. We found a few of them. I think the GAN diet is definitely one that I would recommend, and the diamond diet was also, diamond model was good because it works in metabolic part and in fibrosis. However, we realized that the metabolic ranking and the inflammatory and fibrotic ranking was slightly different. If you want to use these animals to study some antifibrotic, you need to focus in this list. If you want to understand the early stages of metabolic dysfunction in the liver, you will lean more towards this type of classification. So the bottom line is that there is not a single model that will do it, but actually this information may help you to prioritize the model that you want to use according to your objective. With respect to the trajectories of the human being, what we have been doing is to develop a new model. We want to go from the 4 stages, traditional stages in the classification of the fatty liver, to apply a different system which is based on pseudo-ordering and creating a trajectory interference. Each one of these points is a patient, and each one of these points we have RNA-seq from these individuals, and we have ordered them in what will be a trajectory from early stages to later stages based on what we call sliding windows. That allows a huge granularity for the progression of the disease. And in some way, we are very interested in each one of these stages. It's trying to determine the mechanisms that facilitate the progression, as well as other type of mechanisms that can be identified for diagnostic and prognostic factors, as well as to direct the specific type of treatments. This is very interesting because, for example, with the new type of treatments for the thyroid hormone receptor beta, we know that not every patient will respond to that. But if you look in this trajectory, where we find the receptors is at the very beginning, but we lose later on, meaning that maybe with this trajectory, you may identify a subset of individuals where this treatment will become relevant. And you can understand why later on in the progression you may not find this. From this data also, we are developing now the networks for NAFO. Then we are related this to the histological phenotype. We didn't put the histological phenotype a priori. We are doing reverse. And we have documented the correlation with the different type of phenotypes. And the final part at this present moment is the convolution of the transcriptomes on the basis of the specific type of cells that you may have during the progression. Because during the progression, you may change the transcriptome, but you may change the relative type of cells that you get there, and even inside the type of cells, and for this single cell is very relevant. Not all the macrophages are equal. As you progress, you may have high level of macrophages by different types. So this is, I think, is very relevant from our point of view. And then relating this to the sliding window. So we are now evolving into spatial, trying to identify some of these changes in different regions. And through the integration of that, what we aim is to define who may be a progressor from the beginning and second, to devise some specific treatments that may cover, depending on the slight window where these individuals are at the beginning, to try to modify the progression of the disease. One thing that we have done, which is very interesting, is to take the data from the human trajectory and to match this with the different models of the ranking of animals that we have. And we could say that you can identify amongst the animals probably different stages of the progression of the disease. It's not a one-to-one, but you can place your animal in a specific moment of the progression, which in some way gives us a sense that the heterogeneity is not that the animals have not worked, it's that you may get different windows of opportunity in your model. So the final thing, just to finish, is because Maria Jesus has been talking about the brown fat. Well, I think our experience, I'm not going to talk about this. Our experience on this, over the years we have been working with mice and rats and mice and rats. And we know probably everything you need to know about brown fat from a rodent point of view. But at some point, we thought that if this is going to become relevant for humans, we need to have a model, which is a human model where we could identify differences and also we could conform pathways and we could use this. So that's why we move into a different strategy, which is based on human pluripotent stem cells. So there are two ways how you can do this. You can hammer the cells with some genetic perturbation and some ligands and so make a brown fat going like crazy. But we didn't want to do that. What we wanted to do is to follow the normal process of an embryological development of the brown fat cells, stage by stage by having the specific. The reason for this is the following. If you have an obese person who doesn't have brown fat, you cannot activate what you don't have. You're not going to do transplantation, because that's not. However, if you take the obese person, you put them in the cold, they will make their own brown fat cells, meaning that they should have somewhere in between of this process, the system is blocked. So by understanding how this process occurs and how we can push them forward, then we may go back to this. First, to promote the differentiation, and second, you go to the activation. Honestly, I think it's very, this has a clinical opportunity at the end, but we are very interested in understanding the progression through this process to understand not only the transcriptome, but the proteome and different levels of granularity. At this present moment, we are characterizing the effects on the lipid droppers, the mitochondria, shifts in some of the lipids, and we need to talk about this because it's relevant for that. And then aspects of the extracellular matrix. So I think the model is human, is quite advanced, and now we are in the phase where we are deciding how to manipulate it to advance, to accelerate, and to understand how the system works. So this is in some way part of the activities that we are talking on this. Opportunistically, Sonia, who is the one leading the project, she is going to focus on lipid remodeling because in some way we are going back to the in-circle to the projects to integrate the key background that we have, which is relevant in the context of the brown fat. And then also some aspects of mechanotransduction that we think may be quite particularly relevant. I like that. Some people talk about competitive advantage, right? You have a competitive advantage for your project, and you get the feeling that to succeed, you need to compete and then be successful. I have changed this concept. I like the collaborative advantage. You have a collaborative advantage, then you may be successful. And it's very clear that given the current climate of competitiveness, you go alone, you don't go anywhere. You need to have a proper network of people working with you, provide all the credit that you need to provide for everyone to. And this is our current network. We have in Cambridge and in Príncipe Felipe, and we have some links in Seville particularly, and this is data-driven, so data are very important. Then we have people from computing sciences in Cambridge, which are very relevant. Then companies that may help us to reposition drugs in the process. And then we have the Chinese contingent that are very relevant for mice and some of the in vitro developments. We deal with the stem cells and so on. And human genetics, because if you're going to work in something and you have three things that they are cool, choose the one that these guys will tell you may be more important. And then you do something that you're interested in is relevant, but at the same time give you an opportunity to work on something that may have the possibility of being relevant. So this is the lab we have in Cambridge. It's very international with different, I don't think we have that many Brits in here. I think key people for the project that I have shown is Vanessa, and Vanessa's here, Sonia is here, Guillaume is here. There's macrophages, brown fat, and fibrosis. Each one of them we have a common topic, but each one have their own expertise. So that the day that they leave, they take the project, and we have a global network of people working lipotoxicity and complications, each one with their own expertise. So this is the Cambridge group. We are opening a lab in Valencia, and Stefania is my partner, who is allowing me to land there safely. She's brilliant, and then we are growing this group, and I guess this is part of the aging restructuring. Then this is the Chinese Center from the University of Cambridge. These are more offices than it, but these are the team that we are working with. It's very interesting. We are getting people from Pakistan to go to China for training. And I realized that my team in China now speak English with a Pakistani accent, which is contributing to the culture. It is good. Okay, sorry about this. And this is our funders and and I think that's it. Thank you.
Thank you very much, Tony, for this fascinating talk, which really has opened our eyes to a new concept in obesity and metabolism, for sure, at least for me, and I believe for many in the audience, on-site and online. And now we have some time for questions. Gemma?
Thank you, Tony, for a fascinating talk. It's always so comprehensive and so insightful. I was wondering, do you believe there is that when we talk about ectopic fat deposition, that the liver is always the first site? Or are there possibilities in this heterogeneity that the thresholds for other organs might be different in several people?
Okay. That's a good question. So for me, the liver is the next system of control. Probably quantitatively is the main place where you can store as much as you can. And I think being able to have a fatty liver protects you. So you cannot store in the fatty liver because there is a defect that prevents selectively to get the fat there. It's more likely that if you have an excess of nutrients, they may end up in the muscle getting insulin resistant. But my main worry would be the beta cells, because the beta cells are not a good territory. They are not good with oxidative stress. They use oxidative stress for insulin secretion, and this is a regulated process and so on. But if you overload the system, it's quite likely that they will not recover from that. Whereas if you have the liver, you can hold the fat there, and then assuming that you don't make it chronic forever, you still have the capacity of storing this in an efficient way. And eventually, if you unload it, it's quite likely that your liver will be relatively healthy. Whereas in other places like the tsunami, you get fat in, but when the fat gets out, it's not as it was before. So I think that is what I think is statistically probably the most important. However, you then have the individuals that may have genetic vulnerability that can make their liver not able to store appropriately, or their PNL-PL3 retains all the fat there that you could imagine they get more than anything. I don't know. But that you have different probably. And the final thing is, you can get to a metabolic syndrome from every organ. But when you think about the biggest gate is the adipose tissue, it doesn't mean that you cannot do it. If you have a primary defect in muscle, you will become obese and you will get defects. And if you have an effect in the liver, you could from there guide your metabolic syndrome. But it's a matter of probability and frequency. And I think in obese people, the most common problem is going to be adipose tissue failure at one point when your system of resilience allow that to happen.
Thank you. More questions?
Yes, thank you very much for a super nice talk, and also for the organizers for an amazing workshop throughout the day. So something that I've been missing a little bit in the workshop is that the body is not alone without the brain. So in most of the talk, the brain wasn't present. And then I cannot understand peripheral metabolism without brain signals. And actually there was in one of your slices, the innervation. And indeed the brown adipose tissue activation comes from adrenergic innervation through beta-3 receptor. So if you block the beta-3 receptor, there is no brown adipose tissue activation, in PET scans at least. So yeah, and which is the role of the brain in
why,
all this?
Why I don't think there is brain here? Because the brain is so important that as soon as they get here, there is nothing else. I can see that the people who have been invited represent people with expertise in complications associated with obesity that may suggest that you want to reserve some space to hear from some of these topics. That doesn't mean that the brain is not, I think the brain is very important. In fact, if we were going to go, for example, to exercise, I would say I'm more convinced that you may be exercising with your right leg goes to the brain and you take care of the left one. So you need a central mechanism to cut the signal. And the work we have done on the brain, I cannot take credit for that. I always depend on some very smart people who thought, I know about the brain, I may want to go to this lab because I can combine what they know with what they do. And we have had very satisfactory visitors that came with a brain perspective and left with a brain perspective but the other perspective, which I think allows them to do integrative physiology at a high level. Yeah, but I think it's not neglect, it's incompetency.
Thank you so much.
Interesting, thank you. Maria Jesus, you want to ask? No? Yes.
Just to congratulate Tony for the excellent talk and showing us very nice results, but especially I want to thank you also to share your philosophy of science and how science can keep growing through collaborations. I think it's a very, very important issue. And related, well, the question of Maite, I think is very important, all the brain connections, like the gut-brain axis, the connection with the liver, with the adipose tissue.
What she's asking is for a 2-day meeting.
Okay, yes. The next one, we have to do that. Okay, we have already
neurosciences.
We have already the area for the next meeting.
More questions? Thank you, Maria Jesus.
Well, first of all, thank you very much, Tony, for the presentation. I know you started several years ago with this theory, and you have been able to increase and to improve all the topics within it. You have started in your present, well, not started, but at the beginning with the controversial concept of healthy obesity. I would like to continue on that controversy.
You couldn't go beyond the first slide, I can see that.
A few slides after that, you have shown the different factors that could be considered in terms of expandability, and the first one was the pre-adipocytes. And I imagine that was also connected with an article published by Arner's group about the number of adipocytes that we can increase after a certain age. So in relation with that, we know that we have right now a problem of obesity throughout the world, but we have probably even a worse problem in terms of infancy obesity or juvenile obesity. So connecting these concepts, can we think that maybe the future is not that bad because they have started with more pre-adipocytes and therefore they can have a better metabolism?
Okay, I follow that. I will also consider what happens before you deliver the baby. Okay. And malnutrition. So that's another one. I think there is evidence that if the babies are born and they are not too thin, they are a bit, well, that is not bad. Okay, so because at the beginning they may be recruiting the adipocytes and be prepared for the next life. The key problem is when you have intrauterine malnutrition and they are born with less, probably, and with a brain rewire to eat more.
It's okay, out brain.
Okay, so that means that they have a crossover because the system may centrally may be rewiring the nutrients and I need to eat it or not, and peripherally you will dispose the adipocytes because your body may think, well, I'd rather protect the heart and the brain than protect my future adipocytes that I don't even know if I'm going to need them. So that is some of the key parameters that I would consider. And then you have a healthy baby that is born and is a bit, I will not be too worried. I will be too worried if you just feed the baby saying the bigger the better. No, it's not this, but I think having a good repertoire of the cells may be relevant. But that doesn't mean that this is a good factor that you should incentivize to make them fatter later on in life. I mean, I think also there are some data suggesting that is depending on the early stages may have an impact on, so the ones that are relatively well-fed, they may evolve to be being healthier than the ones that at the beginning, and they will have a redistribution when they put on weight. So I think this, but I will not explain that to the lay public because that will be a mess. And I don't think this is something that we are interested in trying to promote at policy level, okay? So just keep it for yourself.
Thinking it not a last future, a close future, because we know already that obesity is related with, or obesity influences the development of cancer. With your concept of obesity or obese tissue, fibrotic obese tissue versus obese tissue in obese people without fibrosis, not fibrotic, could you oversee if that could impact or could have a role in the development of cancer? This is a...
Well, one of the areas that we are pushing on this project specifically is whether by obesity leading to an amplifying some of these inflammatory may accelerate something that is going to happen from the point of view of genetic predisposition to cancer. But I think this is one of the potential links that you may establish. Then is the insulin. I mean, insulin, the IGF-1 has been suggested and the estrogen has been suggested. But in the context of obesity, I will be interested not only in PEP-D, but all the molecules coming from the remodeling of the extracellular matrix of an adipose tissue, which is inflamed, signaling, and finding the right place where to contribute to accelerate the process. In a change, one order of magnitude change can make the difference from dying from a heart attack or having enough time to get a cancer. So that's how I see it. I mean, I think, because if you don't die from the heart attack, the cancer will come. But if you accelerate this, you may get it before the other one. And I think I will not say that the cancer comes with Nova, but maybe you will get it faster or earlier in your lifetime and you may see it. Okay?
Thank you. Any other question? Okay, so I would like thank you very much. I would like to thank all the speakers for the fantastic presentations, the audience, on-site, online. I'm sure we have learned a lot and we have found the answer to many of our questions we brought with here. And I encourage, I know there are students, some already left. In the morning there were many students and young scientists who are enthusiastic with science and probably attending this workshop help you to keep this and increasing your enthusiasm to do your research, your future research. And because this, well, I will talk about physiology and human physiology and mechanisms. We have learned a lot about the mechanisms which are in the basis of human physiology, pathology, in the function dysfunction, and we found that doing this kind of research we can find targets to treat diseases. So I encourage you to keep or start your research career if you are attracted with that. And finally, I invite you to submit your work to the Journal of Physiology and Biochemistry. We will be very happy to consider your work for publication. It's a peer-reviewed journal, of course. And I hope I'll see you in the next edition of this Frontiers workshop. Thank you very much to all of you.